So a much debated, a much focused, and very needed topic which we have kept as a theme for this international conference on advances and future outlook in biotechnology and crop improvement for sustainable productivity. So today I welcome you all the participants, all the distinguished guests, speakers for this international e-conference. So we are privileged. I take great pride in welcoming Dr. S.R. Bhatt sir, who worked as principal scientist at NRCPB and presently a great eminent emeritus scientist at ICR NIPB, New Delhi. Sir, welcome you, sir, to serve as chairman for this session. And I also welcome Dr. S.I. Katagiri, sir, to serve as co-chairman for this session. So to introduce Dr. S.R. Bhatt, sir, we all know him, he's pioneer, he's instrumental in carrying out transgenic research in mustard in India. His primary interest is molecular biology of male sterility, and he has developed technology platforms for hybrid seed production in mustard. Sir is instrumental in cloning and characterization of genes related to sterility in mustard. Sir, we welcome you, sir.
Thank you.
We welcome for this session, sir. Sir, the male sterility system you have developed in Brassica has been extensively used for hybrid seed production in India. Sir, I request you to chair the session, sir. I also welcome S.I. Katagiri, sir, editor US, Dharwad, who served in his various capacities in the university to co-chair the session. And the first keynote address is by another eminent Professor Dr. C.S. Prakash, sir, who is Dean at Tuskegee University, USA. Sir, I request the chairman and co-chairman to take on the session, and I request the speaker keynote to give his keynote address, sir. Thank you. Thank you, sir.
Thank you.
Hello?
Now? Hello? Audible?
Hello?
Am I audible? Hello?
Hello, sir?
Am I audible now?
Yes, sir, you are audible.
Am I audible?
You are audible, sir.
Okay.
We can hear you.
Okay. Yeah. Because I cannot see the organizers. Good morning and welcome to you all for this first session of this second day proceedings. First of all, I thank the organizers, Dr. Indiresh, the Vice Chancellor of the Horticulture University at Bagalkot, and Dr. Fakhruddin. In fact, when Dr. Fakhruddin called me for chairing this session, I felt this is an undeserved honor for an ordinary scientist like me. Because when persons like Dr. Prakash, Dr. Vishwanathan, Dr. Sarma, and are lined up for speakers, I think my choice to be the chairing the session, I think it's in the spirit of giving voice to the common man. And in that spirit, I am taking it. With that, I think it's a really great honor to be here this morning. A few words about horticulture revolution. Horticulture revolution is a silent revolution. It is unlike the cereal revolutions, which was limited to a few crops. Horticulture revolution has been across the crops, as well as across various subdivisions of science. Both minor innovations and major innovations have contributed to it. And also, it has also touched a wider sections of society. That way, horticulture revolution is a silent revolution, which is also combating another problem, a hidden hunger, nutrition. I think horticulture scientists deserve great applause for this major success in a very silent way. Uh, Now, coming to the present, we are in the midst of a crisis, but every crisis could also be converted into an opportunity. Uh, the way we have taken up to this internet mode of communication, the seamlessness and the swift and smooth transition to this sort of technology is a testimony that our common people can adopt and accept technologies, which is a really heartening thing. Uh, Because if this crisis was not going to be there, I think it would have taken years of training of whatnot to persuade people to go online for classes. Now, even the primary school teachers and students have taken it up. This also gives a great opportunity. As we can see, Dr. C.S. Prakash is joining us at a very unusual hour for him. It's morning for him, for us, and it's evening for him. In fact, other day we had a similar conference where the speaker was in the midnight at 2 a.m. So it's also taking our toll on uh, our time schedule. But nevertheless, it's a great opportunity, I feel. Uh, while the crisis of COVID has shown that how we are looking forward to science and technology to provide solutions to the crisis. The way the vaccine development, accelerated development of vaccine that we are witnessing, could not have been possible but for the dedicated effort of scientists. Uh, Yet, I foresee a problem. Although the vaccines are likely to be available very shortly, there is a large opposition to vaccination. Uh, See, this science denial is very, very rampant. As we can see that people are not listening to scientists and healthcare workers to follow the norms. While on the one hand, we appreciate science and look forward to solutions from science, yet at the same time, we seem to deny science at another level. So science communication is extremely important in this context. Uh, Today's topic is transgenics and genome editing. Uh, in fact, genome editing is a revolutionary technology. We are, I think, on the cusp of a revolution, particularly in horticulture. Uh, Perennial crops, which are very, very difficult to breed. Now there is an opportunity to take it to the level of other crops, the crops that could be bred through regular breeding. Uh, we have today, this morning, the first speaker, the keynote speaker, Dr. C.S. Prakash, uh, who is the Dean of College of Arts and Sciences of the Tuskegee University, USA. He is a 2015 Borlaug Community Award winner. And I know Dr. Prakash since long time. He is one of the, I would say, the crusaders of this anti-GM lobby. I think he has done a great service to all of us biotechnologists across the globe through his constant and uh, very effective, you know, uh, rebuttal of the anti-GM lobby. Uh, and he has been well recognized for this effort, both in US as well as across the globe. And I'm sure he will continue to provide that spirit uh, in years to come. Uh, I'm also glad that he is a alumni of UAS Bangalore, uh, and he has also done his PhD from Australia, Australian National University, and then moved on to US, where he has been working on transgenics. I still remember a talk that he gave long time back at our institute on sweet potato transgenics. I don't know whether he remembers it. I'm really grateful and honored to have him as the first keynote speaker of this morning session. Uh, I look forward to a very brilliant talk by Dr. Prakash and hand over to him. And in this session, I have the good fortune of having Dr. Katagiri, my friend, as a co-chair, and Drs. Nishani and Dr. Jayappa as co-reporters. Now I hand over to you, Dr. Prakash. The floor is yours.
Thank you very much, Dr. Butt. Can you hear me okay now? Okay.
You're fine.
Thank you very much. It was a very nice of you to remember me, sir. I clearly remember when I spoke at IRI, although it was many, many years ago. And it's really proud for me to speak at this forum that is hosted by UAS. I am, as you said, I'm a graduate of University of Agricultural Science. I did my Bachelor of in BSc Agriculture, and then also I did my MSc Agriculture with Professor Shiv Shankar in Genetics and Plant Breeding. And I did almost 3 years of my PhD also, where I worked part of the time with Dr. Mahadevappa on rice breeding, but I did not complete my research because I got an opportunity at the time to go to Australia, as you said. And then, and so again, it's a matter of a lot of pride for me to speak here today. And thank you very much, Dr. Butt, and thank you, Dr. Katagiri, for chairing this session. And the organizers, including Dr. Fakhruddin, for inviting me to speak here. And so what I would do is I would just go ahead and share my screen in a way that that I will go ahead and use my slides. And so what I want to do today is to essentially to share with you some knowledge that I have on GM crops and gene editing, and give a very broad overview of these two technologies, and then explore as to how this could be of relevance to Indian agriculture, including Indian horticulture. And so one upfront, I just wanted to acknowledge that, as Dr. Butt said, that horticulture has gone through a silent revolution, and India actually is one of the largest producers of many horticultural crops. But of course, the most dramatic agricultural increase that we have seen was been in wheat and rice, and then followed by pulses and oilseeds. We have not had as much a dramatic increase in the productivity of these crops, unlike wheat and rice. Nevertheless, what we need to acknowledge is that while the population grew tremendously in the past 50 or 60 years, the pace of agricultural production has kept up with that population growth, and is one of the reasons why that we have such high, a relatively higher standard of living in not just in advanced countries, Western countries, but also developed countries, and especially in India. And so India's prosperity today as the global economic power, it is in terms of the purchasing parity, India is number 3 after China and United States. And in actual terms, it's about number 5 or number 6 in the economic powerhouse. And that level of economic prosperity would not have occurred if not for the tremendous advances we had in agricultural productivity in India. And for that, the land grant system of universities, including UAS system that we have in Karnataka, can be rightfully be proud of that. And so when I'm talking about plant breeding, I just wanted to remind our audience that we, especially those who are not very familiar with plant breeding, I know there's a lot of agricultural scientists here, is that we have been modifying our food ever since we invented agriculture, perhaps 6,000 or 8,000 years ago. in different parts of the world. And all our food crops were once wild species. Wild species like this I see here in maize, which was a geosynchron. And in the over over 4, 5, 6,000 years, by slow process of domestication, every crop plant that we grow today has undergone substantial change that you can see again very dramatically shown in the case of maize. And this kind of changes occurred in every plant, including all the horticulture crops. The most important one that I illustrate here is banana. Again, as Dr. Butt said, especially with horticulture crops, it is not easy to breed these crops. But nevertheless, crossbreeding has been one of the most important tools of plant breeding in the past 100 years, 120 years since the discovery of genetics. But in addition to crossbreeding, we have used mutagens. We have used chemical mutagenesis, irradiation, to bring about, create variation when such variation did not exist. And we also used other techniques such as polyploidy to multiply the chromosomes and tissue culture methods. But the focus of this meeting is on transgenesis and genome editing. And so I'm just going to illustrate those two techniques, and primarily also because of the controversial nature of it. It doesn't again mean that these are one of the only two techniques in biotechnology. Many others, especially the genomics and the ability to select for traits through the genomic selection, has literally transforming agriculture in every place, including India. But I am not going to go into that because my expertise is primarily in trying to talk about the GM technology and the genome editing, and I'll just stick to that. So genetically modified crops were first introduced in the United States in 1996 and in India in 2002. And today we have cotton, corn or maize, soybean, three crops where most of these crops that are grown around the world, the content of that is primarily genetically modified. In addition, we also have sugar beets in the United States, canola, which is of course the cousin of mustard that Dr. Katagiri is an expert of. Is also much of that canola grown around the world is genetically modified, and a small amount of papaya, alfalfa, and other crops are also genetically modified. And when you look at what kind of traits that we grow GM crops, most of them tend to be either insect resistant, like the Bt cotton, or the herbicide tolerant, or the few other traits like male sterile genes that again that was developed here in mustard, but was not approved. You can also see that. And slowly but surely, there are few traits of much relevance to consumers. Again, most of the traits that are grown today are of interest to farmers, but slowly traits and products that consumers can relate to are being released. And when we look at the statistics of GM crops, we can see almost about 200 million hectares every year are planted with these GM crops in about 28 countries. This is a dated slide. In about 28 countries and 17 million farmers, including about 7 million farmers in India who grow this crop, who grow biotech crop in the form of cotton that we saw, which again Dharwad, UAS Dharwad played an important role when this Bt cotton was being tested. And almost 95% of Indian cotton grown today is Bt cotton. And so if you ask me why farmers in India like Bt cotton so much, again, this slide is a good illustration of that answer, although this slide is from Australia, but it illustrates very well the value the Bt technology brings to cotton, because you can see it's almost like day and night in terms of the difference between the cotton. The one on the left is GM or transgenic cotton. One on the right is a regular cotton on which no pesticides were sprayed. And you can see the remarkable difference in that. And that's the reason almost when the Bt cotton was released in 2002, there has been a gradual increase in its acceptance, so much so that now practically 100% of the cotton I think in India that is grown is BT cotton, including almost 1 million hectares of uh, of herbicide-tolerant trait added to BT cotton, although that is not even approved by the government of India and our regulatory board. And the impact of BT cotton in Indian cotton production has been remarkable. And then this graph here shows that India is to produce a kilogram per hectare, um, about 300 kilograms per hectare when BT cotton was introduced in 2002, and today it's about more than 550 kilograms per hectare. Similarly, the amount of insecticide that is used on cotton fields have also come down dramatically, although in the past 3-4 years, uh, because of the advent of pink bollworm, that insecticide use has gone up a little bit, and again, it's coming down because of the use of integrated pest management measures to control the pink bollworm. And while BT cotton is very popular in India, in United States, Argentina, Brazil, and many other places, it's the BT corn that is very popular. And the reason why BT corn is popular again is shown it's a kind of a dramatic slide showing the extremes here. This is a farmer in Honduras who's growing this, and my friend Wayne Parrott took this picture. And the regular corn that is not genetically engineered, and if it is not sprayed with pesticide, it would look like this. And again, you can see with one gene that it can confer tremendous tolerance to that pest in corn or maize that you can see here. Similarly, this is another slide showing how a same BT gene that is introduced into soybean can make a remarkable difference in a laboratory trial here, where soybean looper, a caterpillar, was left reared in the laboratory and released into this controlled environment, where you can see almost all of them are eating the regular soybean, whereas a GM or transgenic soybean is completely untouched. And again, and this is a safe gene, despite the protein being highly toxic to the caterpillars. It has known toxicity at these levels to the human beings, um, in that form, in the transgenic form, primarily because the BT protein becomes toxic. It get the toxin is released only under alkaline conditions of the midgut that we see in the insect, and also because we do not have the receptor for this particular protein, it does not bind into the midgut, into the humans or vertebrate animals, and that's why it is not toxic to us, and it's one of the safest proteins that one could see. And the same technology was developed here in India, including US Dharwad played a role in that, in developing an eggplant with the same gene. But sadly, this was not approved in 2010, although the biosafety protocol showed that there was absolutely no problem with this eggplant, but it was not approved by, at that time, the Environment Minister of India, Mr. Jairam Ramesh. And just to show another example of biotechnology is this. A friend of mine, Dr. Kigundu, sent this to me from Uganda, where they are testing some potatoes for late blight resistance. And you can see in this field in Uganda, every one of this potato that's grown here, you see, is transgenic or GM. The disease pressure, the late blight, is so high that it has killed every regular untransformed potato. And again, the gene for this comes from a wild potato. You can see from Dr. Jonathan Jones, who's holding this small wild potato. And you can, by cross hybridization, you can transfer this gene, but it brings in a lot of undesirable traits. And so by GM, if you put this gene into potato, you can see a dramatic resistance that it provides. Similarly, one other important trait that is increasingly going to be relevant is drought tolerance. Again, because of the global climate change and a lot of change, then drought is an increasing fact of life. And you can see again one single gene put into maize here compared to the regular maize, how it can make a big difference in transgenic. And one other example of application of biotechnology is also the development of flood-tolerant rice by identifying flood-tolerant genes in rice and then introducing them into the regular rice. Uh, Another dimension of a most important application of biotechnology, as Dr. S.R. Butt rightly pointed out, is the hidden hunger, is because the micronutrient deficiencies, and India has very big problem of micronutrient deficiency, um, despite the increase in horticultural crop production that we have seen. And so one way to address that is to introduce these genes for high nutrition into our food crops that we eat regularly. And golden rice is one famous example of that. But you also see here how they've increased the vitamin A content, or provitamin A content here in maize and soybean, and here in banana. And in fact, currently feeding trials, the nutrition efficacy of banana that has been transformed, this vitamin A genes is going on here in the United States. But this banana was developed in Australia. And so, so a few other examples here: a tomato with high lycopene content, and a lycopene, now we can get this pineapple now in United States. It is available in the market, and this has a high lycopene content, but it's really more of a cosmetic nature of pineapple. It simply looks so much prettier than the regular pineapple. And here is another example where in the lime they've introduced again genes to improve the anti-oxidation factors that have improved the color in the lime that you see here. And also in the United States, we can get this potato where the genes for browning have been silenced using the silent sRNA approach. And we can also get these apples, which are which do not brown, and yet, so if you cut the apple and pack it in the lunchbox for your child, when she eats that in the afternoon, it would not have turned like this. It would still look fresh. And another big example of biotechnology is also how it reduces the toxin content in our food, like the BT corn that has very little of this fusarium growing on it because it's not been damaged by the insects before. It also makes our food much safer. Another example is how we can also produce therapeutic proteins in plants, in the GM plants. And this is an example where the Ebola antibodies were, when they were deployed 5 years ago, were all grown in tobacco. And even today, against coronavirus, against COVID-19, a company in Canada called Medicago has transformed tobaccos with antigenic genes from the coronavirus and testing them in the clinical trials. And again, as you can see, embracing GM technology will also be helpful from a medicinal point of view. And of course, horticulture, one of the biggest segment of horticulture is ornamental crops. And again, we can buy blue rose here in the United States. It was developed by scientists in Japan and grown in Colombia and exported to Europe. And so, while I've given you a few illustrations of application of GM crops, one of the biggest problems with this, and again, Dr. Butt alluded to, is the public acceptance of it or the lack of acceptance. Not necessarily public, it is mostly the reluctance of the policymakers, including in India, where despite the success of cotton, no other GM crop has been approved. Although there is a tremendous safety record of these GM crops that are as safe or even safer than conventional food. We have thousands and thousands of products containing GM that is available in the supermarkets around the world, and more than 2,000 peer-reviewed papers attest to the safety of this technology. But nevertheless, continued skepticism and doubts and fear of this technology persists, also because of tremendous activism and opposition to that by certain anti-development groups. And just, I believe, to a certain extent, the advent of this newer technology called gene editing might help overcome some of the fears and also help reduce the burden of regulation, because GM crops go through tremendous requires tremendous regulatory testing, costs a lot of money. And that's one of the reasons why big companies like Monsanto or now Bayer are able to bring about GM crops, and universities like UAS may not have the resources. But on the other hand, gene editing is much more cheaper, it's more precise, and most importantly, it does not entail transfer of foreign genes. It is essentially similar to the mutagenesis that I talked about, where we simply change a few letters in the DNA of the crops, like the mutation. But unlike the traditional mutagenesis, here it's a targeted mutation. In that way, you only change the gene that you want to change, and there is not much background unknown genomic changes. And so, because of that, in the United States, the gene edited crops are not regulated. And so, the couple of companies like KLX and Cybus have already released some of the products like potato that's acrylamide free, canola that's low in saturated fats, so that this canola oil can be stored at a room temperature without the need for hydrogenation like dalda that we do in India. And also, they're working on reducing gluten in wheat, because many people are allergic to gluten they're intolerant to gluten. And so few other examples that I list here. And again, there are many other products that have been developed in the United States that are being communicated to USDA and waiting for the regulatory or non-regulatory status of that, saying from USDA that this does not need any approval. And so I've listed several examples, and many of them, these are horticultural crops, and so of relevance to India. Non-browning in avocado, a better tasting pea, and canola with, again, better oil quantity, low phytate canola, so that it is many of the nutrients are easily available. And then disease resistance in tomato and lettuce and maize. And I just want to show you some few examples of that in the next few slides. And so you can see there are a range of traits, almost anything that we had been earlier working with genetic modification. By using gene editing, we can bring about some of the changes. And here is a tomato, for instance, that the size of the tomato was increased. And so gene editing in the beginning was, we are really working on mostly qualitative traits. Now we also recognize many of the quantitative traits can also be improved using gene editing. And this is one other example, whereas in rice, they were able to bring about, through gene editing, make rice more resistant to bacterial blight. Bacterial blight is a very important disease in India, where a lot of rice breeding around India, in addition to blast, they're also trying to improve resistance to this Xanthomonas bacterial blight. And again, by gene editing, they were able to to bring about resistance to this bacterial blight in a recent paper. One other example is how, by altering the transcription factor, they were increased the shelf life in tomato, and by slowing down the ripening of tomato fruits. And almost all of you, I'm sure, would be familiar with the tomato, that if you don't store it in the refrigeration under regular ambient conditions, tomato has a short shelf life. And so it's very common in India that during the glut of tomato season, farmers don't get much returns for their, I think the tomato prices will come down, and it's often you see they just throw those tomatoes into the highway. But on the other hand, if we can come up with this low ripening technology, then these tomatoes can be stored for a long time, and they can also be transported to larger distances. In that farmer growing tomato, say in Bagalkot, may be able to send it to a market in Bangalore or Madras or wherever, in a way that to wherever she or he would get a good price for their produce. And so you can see this kind of technology can be adapted to every climatic fruit and vegetable, in a way that we can slow down the ripening and able to improve the shelf life of our fruits and vegetables in India. In India today, 50% of all our fruits and vegetables that are harvested get perished because of lack of good infrastructure and lack of adequate processing facilities for these. And so much of that wastage can be saved if we can come up with varieties that have a larger shelf life, and also the gene editing technology lends itself to improving the flavor and improving the taste of many of these horticultural products, and gives an ability to add tremendous value to the products. And another example here is I wanted to share with you is a recent paper that showed how they were able to increase the lycopene content in tomato. Lycopene is the chemical precursor to vitamin A, actually, that helps make the tomato the red color that we see in the tomato. And by increasing the lycopene content gradually through gene editing, you can see how a greater intensity of redness is produced. And so this is not just of a cosmetic value, because studies have shown that high lycopene content is very good, especially for men, because it can reduce the prostate cancer incidence. And a few other examples of application of gene editing in crops. They've been able to increase the grain size and just able to increase the yield uh, in in cereals, and then there's also a good example where plant architecture can be changed. And here again, in a tomato, they were able to change a vine type of growing tomato that grows in a vine into make it into a dwarf architecture. And as you can imagine, as you know, the Green Revolution was made possible both in wheat and rice, primarily by incorporation of dwarf genes into these, so that short-statured varieties helped uh, reduce the lodging and thus increase the yield in both of these crops. And so by gene editing, you can bring that element of dwarfness into almost any crop that you're working with. And one other important role of gene editing has been to take literally a wild tomato, um, like the Peruvianum, and then modify that into a cultivated plant within a span of 1 year. So the evolution that took 8,000 years, now we can just do that in a couple of years. And that also helps us to bring many elements of wild traits into the cultivated traits, especially the disease and pest resistance that we see, and then the drought tolerance. And again, one other example in increasing the rice growth and productivity by mutations, gene editing of the subfamily of the abscisic acid receptor genes. And again, some of these are providing a lot of insights into the biology of these plants. And so the gene editing helps us not only to alter the gene directly, but also to modulate the gene expression by alteration of the coding sequences. We can repress the genes, but we can also altering the regulatory sequence of the genes. We like the transcription activation that I showed you. We can activate some of the genes that are repressed in the plant. But the most important thing to remember is when you use gene editing, especially like a technique like CRISPR, for which the inventors of this technique got the Nobel Prize this year, as you know, is that it is indistinguishable from a mutation, induced mutation. If you use, like for instance, if you have a crop that is edited with CRISPR on one side, and the other side, if you have the same crop where you have the mutant through either chemical mutagenesis or radiation mutagenesis, or even a natural mutant, there's no way you can tell which one is which. And so there is really no scientific reason to regulate one, such as CRISPR, and not regulate your chemical mutagenesis or radiation mutagenesis or natural mutants. And we have more than 3,000 varieties of food crops that have been approved for cultivation that through induced mutation with a very safe history. And so there is no reason why those CRISPR mutants are not going to be any different. I am quite confident they would be as safe. And of course, we can always bring some element of testing to ensure their safety, but there is no need to be scared of them like we have been with the GMOs. And there is no need to bring in a lot of regulation that it takes so many years for them to test. For instance, the genetic engineering today, many of them, including BT cotton, went through about 10, 15 years of testing before they're brought into the market. And it takes many years for them to be even and to develop. On the other hand, gene editing can be accomplished much more faster, and with a limited amount of testing, we can bring them into the market in a much faster manner. So, in summary, gene editing can remove and add traits, and just like conventional breeding does, but it's more precise and it's more faster. Um, and so for India, I believe gene editing technology can bring in, especially in horticulture crops, it can help improve the nutrient levels by increasing the vitamins and antioxidants. And we can develop our crops to be more climate resilient by increasing stress tolerance, heat tolerance, drought tolerance, and also make them disease resistance, pest resistance. We can thus improve our food safety and the food quality, because there is a tremendous price that we pay in India by compromising our food safety and food quality, especially in horticultural crops. And we can improve that dramatically with them by embracing this technology. We can increase the shelf life of fruits, vegetables, and flowers. We can improve the oil quality of practically all the oil that we grow, that we produce in India, whether it's groundnut oil or sunflower oil or mustard oil. And we can reduce or eliminate toxins in many of our food. Like in castor oil, we can reduce or make the ricin content zero, cyanide in cassava, neurotoxins in veg pea, and also reduce the phytate in rice and corn, so that they would be better palatable, especially when you feed it to animals. So that has been my talk. And if anyone wants to get in touch with me, this is my email address. And if any of you are, especially on Twitter and the social media, if you would like to follow me, I keep talking about many new innovations coming along in crop gene editing and other related technologies, and especially as it relates to societal acceptance and what we as scientists, we need to bring greater awareness in the public, so that this technology can be adapted and India can benefit from that. Again, thank you very much for giving me this opportunity to speak.
Thank you, Dr. Prakash, for a wonderful presentation. We have a couple of questions, quick questions. We are just slightly behind schedule. I will specify them and then you can respond. One question from Nirmalya Chakrabarti from Calcutta University is, what's the main problem with BT brinjal acceptance? Because there has been moratorium about BT brinjal. I'll list out three questions and then you can respond together.
Certainly.
The other one is, what are the strategies that we can adopt for acceptance of GM crops in India? The other one is, what are the consequences of transgenic crops on ecology, biodiversity, and food chain? Um, the last question by Srinivas Sabale is, why are you spending money on GM research when India is not releasing anything other than cotton? So these are the bunch of questions, if you can take.
Yeah, certainly. And in the interest of time, I'll be very brief. The first question on what specifically was, what are the problems with BT brinjal, is it? The first
Yeah,
question?
yeah,
Okay. Technically, there is really no scientific problem because, as you know, Bangladesh adapted this BT brinjal, and we have like a twin study. You can just look at across the border and see every year the number of farmers who grow BT brinjal in Bangladesh has been increasing. The markets are full of those brinjal, and I also know that BT brinjal, because the border is so porous, has come into India, and literally hundreds of thousands of Indian farmers are also growing BT brinjal. And technically, we have not seen any problem because we don't expect any, because this is a very protein that is one of the most intensively studied protein in the history of food technology, the BT protein. And we know that it doesn't cause any problem as far as the food safety is concerned. And in 2010, when Jairam Ramesh put a moratorium on that, it was based on a flawed opinion that he got from a French scientist who was visiting India, Seralini, who said that if that he did some studies with BT corn and that he fed it to rats and rat developed cancer. And we know that was a bogus study today, and that paper that Seralini claims has been withdrawn. And so there is no scientific basis for that. And much of that is the fear of unknown that led to the moratorium on this. And sadly, because of that, India has paid a huge price for that. And so, what are the strategies to adapt GM crops in India? I believe the most important strategy is for scientists to speak about this technology to the best of your knowledge, to learn about this, and then to speak whenever you have an opportunity to help improve public understanding and public acceptance. One of the problems we did have in 2010 when Jairam Ramesh went around the country, like almost like a circus, trying to solicit public and opinion on this. People who are opposed to this technology brought in lots of people into the meetings. He held meetings in 6 places, and in most cases, the scientific community did not show up. And in fact, in some places, they were asked even not to speak up about. And so the information that ministry got was practically, primarily one-sided, and it was very biased. And so I believe scientific community must continue to speak up against it and show the science-based evidence for the safety of this. And but beyond that, I really don't have any magic answer to that, because if I had, we would have had a greater acceptance of GM crops around the world. But what I do believe is, with the gene editing technology, some of the newer products that are coming along that people can relate to and see that if it appeals to people and they can readily recognize the value that it brings to them. And then also, one thing you must remember is many of the earlier GM crops, almost all of them were grown by multinational companies like Monsanto. And so people are very, very apprehensive of these big multinational companies. And again, universities like yours, UAS Horticulture in Bagalkot, when you develop a product, the farmers and consumers are more likely to trust your product. And so hopefully, I would think I am hoping in gene editing, when it becomes much more prevalent in India and we find a lot of products coming along, hopefully there will be greater approval of that. And another question, final question was, what are the consequences on ecology, biodiversity, and food chain? That's a very important question. And And unlike traditional varieties, GM crops, we asked these questions when BT cotton was developed. These were exactly the same questions were asked. What is going to be the effect of BT cotton on the ecology? And so the developers had to provide with the data on literally hundreds of tests that they had done, looking at its effects on earthworms, effects on butterflies, and its effects on when you feed it to the cattle, what's going to happen. And so, and again, when you look back and in India itself and see 7 million cotton farmers are growing this BT cotton, almost in all the studies that have been done, there has only been a positive impact of this technology, because when you have less pesticide, that's a positive impact on the ecology. And many of the herbicide tolerant crops, for instance, it promotes zero tillage. So when you don't till the land, that is one of the biggest positive impact you can have on the soil ecology of that. Biodiversity, and again, all of this varies on a case by case basis. What is with happens with one particular trait when one crop may not be the same with the others, and that is why some of this regulatory testing is done. And food chain too. These are all very big questions, but my short answer to this is this. Look, in almost 2000 to 2500 research papers that have come about, that have looked at asking many of these questions on food safety and environmental safety of GM crops, we have not had any evidence that the GM technology itself is any riskier or brings about any unique risks compared to traditional technologies. In other words, almost any fear or any risk factor that you can define using this GM technology, we can also see the same with traditional farming too, or traditional breeding. Because when you take a tomato and cross it to the wild species of tomato, you have no idea what kind of genes you're bringing in to cultivate tomato. You may be bringing the allergen genes, you may be bringing in genes that would have impact on the ecology and biodiversity. And the similar risk exists with the GM crops. But unlike traditional breeding with GM crops, much of the testing is done to ask these questions. And if in any case, if there is a negative impact, then you don't have to approve these crops. And also we learn as we go along. And my final, it's a good question. Why waste money on developing GM crops when it's not approved? That's a very good question. And that is the reason why you don't have almost no commercial GM research in India. Almost all the companies like Monsanto, which is now Bayer, Syngenta, BASF, they all had laboratories conducting GM research, and all of them have closed the shop and left. Uh, they only focus on genomics, traditional breeding, and perhaps now they maybe have started working on gene editing. And that is really sad, because the GM technology, has tremendous potential to offer to India. And India was one of the vibrant research labs all across India. There was vibrant research going, dynamic research that was going on. Now that many of the academic labs do not work on this anymore, and so that I think is is a matter of loss for India. I do hope that it has a course correction and picks up with the gene editing. And 5 years from now, we may be talking other technologies. So science improves all the time, so much so. Just look at the case of coronavirus. 1 year ago today, around this time when the coronavirus started in China, and when it started in January, no one even knew what virus was causing. It didn't even have a name. And 11 months later, we have two vaccines approved in India, one in China, one in Russia. I'm sorry, two vaccines approved in the United States. And two vaccine may be going in India into the clinical phase. And by the way, the one of the vaccines from Biotech is my classmate. He's a graduate of UAS Bangalore. He did his Dr. Yella Krishna, who is the CEO of Bharat Biotech. He's an agricultural graduate from Andhra Pradesh, and then he did his master's in plant pathology. And so what I'm trying to say is, you got to have a scientific ecosystem, a science labs, a dynamic science labs going on, so that we may be able to use many of the newer technologies as they come along. A lot of times you don't know where the application is going to take you to. And so India, is a global scientific power in many, many areas, but sadly, it is languishing in the area of genetic biotechnology in agriculture. Thank you.
Thank you, Dr. Prakash, for a wonderful presentation and discussion. We are running behind schedule, so I will now invite Dr. Vidyut Sharma, who is currently the ICR National Professor and also Director of DBT, Assam Agriculture University Center. Dr. Sharma has been most active in the area of transgenics, particularly BT chickpea, which he has developed and also is given out to private companies for commercialization. Without taking much time, I request Dr. Sharma to talk about, who will be talking about gene technology for improvement of croplands with special reference to chickpea. And I request him to keep to time so that there is some room for questions and answers. Thank you, Dr. Sharma.
Thank you, Professor Bhatt. I'm pretty happy to be here with you all. First of all, I would like to thank the organizer, especially Dr. Fakhruddin. He's a very good friend of mine. And because of his request, I immediately accepted the invitation to give a brief talk today. And I'm happy that I heard Dr. Prakash, who has been advocating the GM crop since quite long, and then he has been always trying to promote the GM crops in India also. So without spending much time, I would like to go through my presentation here. Whether my presentation is visible to all of you?
Yes, sir. Visible.
Okay. Hi, Fakhruddin. And then I have also seen Dr. Katagiri, okay, who is my collaborator now. So I'm happy to see you as well, Dr. Katagiri. Okay, I please forgive me a little bit because my voice is a little bit, down today because of cough. Of course, I'm sure it's not Corona. Anyway, because of the climate change here. So my topic is, gene technology for the improvement of crop plants, with special reference to chickpea, because, we have been working with this particular grain legume to modify it genetically for the last more than 20 years now. And the questions that will be addressed today, that whether genetically modified chickpeas are protected against insect pests, because our major emphasis is on insect pest resistance, and whether the G crops are accepted globally, because this part now I'm not going to discuss in details because already you have heard Dr. Prakash. So very quickly I'll go through that part. And what are the challenges ahead? Actually, Dr. Prakash has also discussed. I didn't know that before me, Dr. Prakash will talk, because that's why I thought, apart from the chickpea gene technology, I would like to discuss, I thought of discussing these two portions as well, but now I'll be reducing those two portions. Anyway, so first part of my talk is the gene technology for the development of insect-resistant chickpea. Now, why? Why did you think of adopting adopting gene technology for developing insect resistance in chickpea? Because in chickpea, there are two very serious insect pests. One in field, that is spot borers, it's a lepidopteran pest, which damage, chickpeas almost 100% sometimes in certain areas. And another one is the storage grain pest. This is a kind of bruchid kind of pest, and it damages chickpeas during the storage condition. especially when they lay eggs, and then adults go inside the seed, and then larvae grow into the adult, and then adults, when they come out of the seeds, then they make such, holes. That is why I always ask, when I present this, presentation, I always ask if there are any, pure vegetarian, because it's very difficult to say, because, inside the chickpea, there are larvae present. And because of that, a lot of pesticides are being applied just to get rid of this sort of, insects. Anyway, these are two major pests. And then so far, there are no insect resistant varieties in the field, completely protected varieties in the field for these two insect pests in chickpea. Well, when we thought of, say solving these two problems, we had to develop a transformation protocol, because which is very much, urgent, which was very much urgent at that time. And then, because chickpea was considered to be a recalcitrant crop, but today in our hands, we have developed a very robust, genetic transformation protocol. We use a cotyledonary explants with half embryogenic axis, and then put them into selection and regeneration medium. We use the NPT2 as a selection marker gene, and then we we go for the further selection, by subculturing them, and then we select the explants with the multiple shoots and the healthy shoots. And then this was a technique we developed, that instead of, rooting them directly in the tissue culture, we do go for in vitro grafting. That is, the transgenic shoot was grafted into non-transgenic, root stock. So, and then we established them in the glass house. So this was, in brief, the protocol that we developed for the, chickpea, and now many labs, they are following this protocol. And then our first target was to develop chickpeas for storage grain paste, because, being from Assam, we worship here in Assam, we worship our God with chickpea. And that's why, in large gathering of, religious gathering, we feed the, people with the chickpeas, and then these are uncooked chickpeas, and then, sorry, uh, I shall go to the next previous slide. Hang on. Yes, uncooked chickpea. So what happens, because of the, if the chickpeas are not washed properly, then there are even sometimes, there is food poisoning. So this is why I took up this problem first. And, during that time, a group in Australia, Dr. T.J. Higgins, and one in US, they cloned a gene from Rajma already. They identified a gene in Rajma. There's a common bean, which is which showed the resistance against the storage paste, that is buckets, And then, I collaborated with them, and then I started developing a transformation protocol, as I said. And then I started transferring this, gene from Rajma, because, common bean is fully resistant against, storage paste. And I transferred this gene into chickpea. So, and instead of going to very details of the technology, I'll just simply show you, this was the tDNA that we incorporated into chickpea. And you can see here that, alpha amylase inhibitor gene was, driven by phytohemagglutinin, a seed-specific promoter, and also a seed-specific terminator from common bean. And we used NPT2, that is the neomycin phosphotransferase gene, as selection marker gene. And this is a southern blot showing that, this is the control chickpea, and these are the transgenic chickpea lines. And you can see here, most of my lines, they contain the single copy of the genes. Of course, this one contained the double copy of the genes, and these two genes were, not having the integration of the gene. And then we showed by Western blotting that, the genes were the lines were the transgene integrated. They also expressed the alpha amylase inhibitor protein from common bean, and this is the control where there was no expression. And these two lines where there was no integration, there was no expression as well. And we looked for some of the lines, which were high expressing lines. We looked for the Mendelian segregation, and what we found that, those lines, where in high expression was there was also a Mendelian segregation of 3:1 ratio, and then they also contain single copy of the transgene. So with this, then we looked for the insect bioassay. We did the bioassay several times in the lab, and then finally we got that these two lines, which were completely protected against, storage space. So this was the first success story from our lab. And when we got this success, then Department of Biotechnology, Government of India approached us that we should do some work with the pod borer resistance in chickpea. And then international organizations like SDC from Switzerland, they also approached us. So under the Indo-Swiss collaboration program, we started working with chickpea for resistance against pod borers, that is Lepidopteran pest or Helicoverpa armigera. So for my next story will be that developing chickpeas resistant to pod borers. You can see here that at that time, there was a controversy that whether we can put NPT2, whether we can keep NPT2 as selection marker gene in the plant itself, GM plant itself. That is why we thought of using a twin tDNA binary vector. You can see here, there are two tDNAs, left border, right border, and then left border, right border. In one tDNA, we put NPT2 as selection marker gene, and in other tDNA, we put BT, because that time we saw the success of BT gene in cotton and corn. That is why we thought of using the BT gene for conferring resistance against pod borers in chickpea. And then, this particular BT gene we expressed with a green tissue specific promoter from, Arabidopsis, that is small subunit promoter of Rubisco. So that because, pod borers infest mostly in the green portions, like, green leaves, green stems, immature cotyledons, etc. So that is why we use a green tissue specific promoter here. And then purpose of putting the two genes in two different tDNA was to segregate out these two genes in segregating generations so that we can get rid of NPT2 gene in our transgenic lines. Unfortunately, we didn't get rid of course, because we found that both genes, of course, co-transformed and that co-transmitted to the next generations. But now we know that there is no problem of having NPT2 in GM crops. European Union, even they have accepted this. That is why we went ahead with this, and then we use Cry2AA as well as Cry1AC. These are two different BT genes, and we transform chickpeas with these two different BT genes independently. And then just one story here with the Cry2AA gene. You can see here, these are my transgenic lines. Initial PCR confirmed that there was presence of the transgenes, and these are control lines. And then we expressed the BT genes with the Arabidopsis small subunit promoter, as I said. And then this is a BT cotton we took as the positive control, and this is a chickpea negative control, untransformed chickpeas. And you can see here, expression varied. Some of my lines had very high level of expressions, some of my lines had very low level of expressions, and some of my lines had medium level of expressions. So with the high expressing lines, we confirmed that these lines had expression in all different tissues where we actually wanted to have the expression, like young leaves, old leaves, sepals, petals, pod walls, immature seeds, etc. And in dry seeds, very minimum expressions. So this confirmed that that the things moved as per our expectations. And by Southern analysis, in one of my high expressing lines, we did Southern. Southern is a very cumbersome process especially in legumes, in chickpea. It took a lot of time, for optimizing a Southern in chickpea, but this is a very good Southern we got finally. And you can see here that some of my high expression lines like 6H, 6L, they contain single copy of the transgenes. So we were happy with this, and then we look for the segregation, those lines wherein we got the single copy of integration, like 6H, 6L, 6L, and we look for their segregation, and we found that the transgene segregated at a 3:1 ratio. So we did the bioassay, and this is the final slide, that we were quite happy seeing this bioassay, because this is a lab bioassay, of course. You can see here is a parental line. These are the null segregants. where we got, a lot of, insects when there's leaves were, insects were fed with the leaves, non-transgenic leaves and null leaves. There was, a lot of damage. But when insects were fed with the transgenic lines, leaves of the transgenic lines, then there was no damage at all. The leaves were quite healthy and green. So then when we obtained this, then we showcased our technology, because DBT wanted to showcase the technology. And then, that Maharashtra Hybrid Seed Company and then many other companies, actually, they put their indents. And then, finally, Maharashtra Hybrid Seed Company own. And then, we transferred our technology to Maharashtra Hybrid Seed Company. And later on, the technology was transferred to the Sangro Seeds, a sister concern of Maharashtra Hybrid Seed, because of political reason only. And at the same day, we also transferred our technology to US Dharwar. I'm very happy that Dr. Katagiri is now taking up, because a lot of several years, it was just remain as such, because there was no permission, no objection certificate they didn't get. But after Dr. Katagiri took over, they have got the permission from, Karnataka government, and now the things are moving there. Why we transfer the technology? Because, now we wanted to transfer the BT gene into the popular cultivars. So from private sector, Maharashtra Hybrid Seed Company took up the things, and then from public sector, initially US Dharwar took up, and later on we also transferred to, ICRISAT, as well as, IIPR Kanpur and Punjab Agricultural University. IIPR Kanpur has now stopped, of course, because of certain regions, but, Punjab Agricultural University has moved quite, ahead. And you can see here the first field test. We did the 2 years of field test, using our backcross derivatives of our chickpea lines, Cry2AA lines. In 2014, 15, and 15, 16, we did 2 field tests in Andhra Pradesh, and there was 25% yield advantage we got. But after that, there was no more field tests, because, because of certain reasons, because of, as already Dr. Prakash has discussed, that, in India, except, BT cotton, nothing has been moved further. So that is why, Maharashtra Hybrid Seed Company is not taking that much of interest now. But of course, we applied, for field tests in many other states, but we didn't get. But anyway, we thought, as it was already expressed by Dr. Prakash also, that, public sector should take lead now. That is why, as I said, we transferred the technology to Punjab Agricultural University, as well as, US Dharwar is also now moving. So I'm happy that Punjab Agricultural University has now already BC3 F2 generations, and then, within a year or so, they will be getting a line ready for, field test now at Punjab Agricultural University. And US Dharwar is also moving, and I'm hoping within a couple of years, Dharwar is also coming up with good backcross derivatives, so that, we can approach RCGM for allowing us to go for field test using our lines. Anyway, we did the biosafety assays also. My lab also engaged in biosafety assays, and we confirmed that there was no vector backbone present in the transgenic lines. And now we have also identified, the chromosomes. It has been integrated. So that this data I have not presented here, but now we know where this particular, BT gene has gone into. And of course, some nutritional studies also we did, and compositional analysis, and we found there was no difference in control chickpeas as well as transgenic chickpea lines. We cannot wait, because we do not know finally that what the regulators will finally approve. They may not approve single BT gene lines. That is why we have started developing chickpeas with 2 different BT genes in a single line. That is why we have started expressing or developing transgenic lines with the 2 different BT genes, 1 AC and then 2 AA lines. And we have got now some new lines with 2 different BT genes in one single plant. So this is going on in my laboratory, and then I'm hopeful that within a couple of years we'll be able to get homozygous lines using 2 BT genes. I'm happy to tell you all that recently Bangladesh has expressed their interest in our GM chickpeas, because Dr. Prakash has said in BT cotton, which was not allowed in India, that has been allowed in BT brinjal, has been allowed in Bangladesh, and that is Bangladesh has now expressed their interest in our GM chickpeas, especially the storage paste, and also in BT. And then we recently visited, last year we visited Bangladesh, and then we talked to the Bangladesh farmers, and they said, pot borer is the major problem there, as well as in the storage space, storage condition, storage space are problem. That is why we are now talking, we are negotiating. My collaborator in Australia, because whole thing we did in collaboration with Australia, Dr. TJ Higgins. Dr. Prakash must be knowing him very well, and many of you know him. And so, we visited that, and now Australia, Assam Agriculture University, and Bangladesh Agricultural Research Institute, we are making a joint program for developing, lines for Bangladesh. So should we accept, GM crops? Yes, I'm not devoting much time now here, because already Dr. Prakash has said, I do not say GMO, because, I would love to say them as biotech crop, because, this is a technology. Because whether we do go for genome engineering, that is also genome modifications. So, whether transgenics, whether cisgenics, these are all genetic modifications. And, genome engineering, genome editing cannot be an alternative, because, for example, in chickpea, we do not have a good gene in the wild or in chickpea gene pool yet. So that is why, in this case, definitely that we have to go for the transgenic approach. So these things already Dr. Prakash has showed, I'm just skipping off, and then as you know that now in cotton, worldwide 80% cotton is in occupied by the GM crops. Soybean 77%, canola 30%, and maize 32% is occupied by GM crops. And India is occupying the fifth position because of BT cotton. And then what are the challenges? As I said, because there are certain statements which are vague statements, like potential risks to the human health, allergy, because allergenicity can be obtained from many other food plants, and then there is, as Dr. Prakash has said, there is no farm evidence that this particular allergy is because of the GM technology as such. So then horizontal gene transfer, even in the cotton field, there is no super which have been discovered. So horizontal gene transfer also, there is no direct evidence like that. And organic is the future. Because of this, many people, they are objecting, but I advocate that if we have to go for organic agriculture, definitely we have to reduce the pesticide applications. And in that case, definitely we need to have the insect resistant lines or disease resistant lines. So if we have to reduce the spray, etc. And so pesticide spray. That is why I would advocate that there should be marriage between organic agriculture and then GM technology. And then many people, they say GM seeds are costly. This is again a vague statement. Unless it is a hybrid, there is no question of the initial production may be a costlier technology, but once it is released as a variety, for example in chickpea, we are going to release this as a variety, so there is no question of higher cost or something like that. So farmers definitely can maintain those lines unless it is a hybrid. So but of course, the consumer perceptions, for that, I would always advocate that, right from the beginning, we should have the social scientists along with us. There, someone asked about the strategy of how we can popularize this technology in India, or how we can go for accepting this technology in India. I would advocate that we do not give much emphasis in social science, but this is very, very important. The economics as well as societal acceptance, we have to definitely advocate for all these things. So other challenges are IPR issues. Of course, there are, yes, issues, IPR issues. Definitely, I must agree to that. And development of marker-free transgenics, this was a very challenge, very much challenge, And then, of course, my group, did through various approaches like, Kellogg's technology we use, and then, twin binary vector system we used. But, in crop, it depends on crops. In tobacco, we got very good success. We published already, because of through Kellogg's system also we could develop marker-free transgenics in tobacco. But it depends on the cytoplasmic background sometimes, genomic background. So in chickpea we have not yet been successful. And process of deregulations, as I said, field test itself is a cumbersome process, because we have been trying to do the field test in different states, but still getting a no objection in India itself is a big problem. So this is another big challenge. And health hazard, many people say about the health hazard, but you see the WHO has already accepted this. They have already publicized that this has gone through so many tests, so there is no question of that. And then the GM food crops grown in US have been evaluated through food and drug testing. So since we have been taking all these measures, unless we take all these measures, GM crops will not be released in the field. The maximum, I think, care has been taken for the GM crops so far. So as Dr. Prakash has said, even in the mutation building, we do not know what the deleterious genes, have been, brought into the plant background. We are not bothered for that. But in the GM crops, because of the regulations, we do each and every test. That is why. And from the health hazard point of view, as the question came in, I would like to show you one slide. This is the one final slide, perhaps, that people talked about the BT brinjal, and then when I visited Bangladesh, I thought of first visiting the sites in Bangladesh, and I was amazed to see where the BT brinjal grew, in a large scale. Those were very much neighboring to neighboring to India, very close to India, and there was no basically there was no protections for even the people to move. So we do not know actually if illegally BT brinjals are being grown here. And I said I would taste. So I became a guinea pig in Bangladesh while visiting. This is just for fun, I'm saying. But question is that both myself and TJ Higgins we tasted BT brinjal for several days. 4 days we stayed in Bangladesh. Anyway, I'm still healthy, no problem. And finally, I conclude that genetically engineered chickpeas resistant to insect pests are developed at Assam Agriculture University, Jorhat, in collaboration with other public and private partners, and expected to bring in major change in pulse production in the country. And genetically engineered crops, that is biotech crops, have been globally accepted for food and nutritional security. And that is why I say we should say yes to G crops for the benefit of mankind. Thank you very much.
Thank you,
With
Dr.
this,
Sharma.
I must acknowledge the Department of Biotechnology, then ICR, Government of India, and Indo-Swiss collaboration in biotechnology, Australia India Strategic Research Fund, McKnight Foundation. Actually, with this McKnight Foundation, I started genetic engineering work at Assam Agricultural University, and of course my parent institute, and all my collaborating organizations. So here are some of my collaborators here. You can see Dr. Higgins, Dr. Anand Kumar, and then Dr. A.C. Sharma from ICRISAT, and Dr. Chaur. Of course, Katagiri is already there, and then Dr. Sumita Acharji, my assistant, she is an assistant professor who has been collaborating with me for quite long now. And this is my whole group here. And thank you very much. This is my center. This is Center of Excellence. Because of our chickpea work, Department of Biotechnology, Government of India has given us this Center of Excellence. And this is the Northeast Center for Agricultural Biotechnology at Assam Agricultural University now. Thank you. Thank you very much.
Thank you, Dr. Sharma, for a wonderful talk. And perhaps with your chickpea going further, I think the question asked last time regarding whether we will have any other crop, especially the edible crop, GM crop in India. Wish you good luck with this.
Thank you. Thank you.
And there is one question which is specifically asking on Dr. Dandapani, could you get marker-free transgenics? Because you had two independent co-transformations. What do you think?
Yes. I think I answered already that question, because marker-free transgenics. We tried in two different approaches. One is the Krelock system. I'm not going to discuss that in details, but there is a Krelock system that we tried in chickpea. But this technology was, very much successful in tobacco. When we transformed tobacco, we could get, marker-free transgenics. We have published one paper in 2008 itself. You can go through that paper in tobacco. And then, but in chickpea, finally, with that technology, we didn't get. Then we tried through two tDNA. This is a twin tDNA vector system. It is a co-transformation system. And, we got a marker-free line. But under one out of about, say 100, perhaps in one, we got marker-free transgenic lines. But, this is not a very good frequency. But, that line was not very good expressors. But today, we do not bother because if we transform it with the NPT to gene, then there is no problem. Of course, with the bar there is IPR issues. So I would advocate that you transform with specific marker gene, or even nowadays people are trying to implant transformations without a selection marker gene. So these technologies are coming up. So if it is NPT to gene, we should not bother about that. Thank you.
Thank you. With that, we conclude this talk. We will move on to the next speaker.
Thank you very much.
Thank you. Thank you, Dr. Sharma.
Yeah.
We move on to the next speaker, Dr. D. Sudhakar. He's a professor at the Tamil Nadu Agriculture University who has done extensive work on transgenics in rice. Today, he will be speaking on CRISPR-mediated genome editing for disease resistance in rice. Dr. Sudhakar, again, I request you to limit your talk, leaving some margin for questions. Thank you.
Thank you, sir. I just share my screen first. I hope you people are able to see my slide screen.
Yes, we can see, sir.
Thank you, Fakhruddin. Okay. Respected Chairman and dear participants, good morning to everyone. It's a great pleasure and privilege to be a part of this key conference organized by University of Agricultural Sciences, Bagalkot, and talk to you people on a topic genome editing for disease resistance in rice. At the outset, I like to thank my friend and Dr. Fakhruddin and other organizers for giving me this opportunity to speak on a subject on which we are currently working. So in the introductory part of this talk, I'm going to discuss a few examples in which gene editing tools have been used to develop disease-resistant rice lines. And then in the second part of the talk, I will discuss one of the work which we have been doing in our lab with a view to develop virus-resistant rice line using genome editing tools. As you all know, one of the major breeding objectives in crop improvement programs, irrespective of crop, is the development of disease-resistant varieties. Conventional plant breeding approaches have been quite successful in developing a large number of resistant varieties. All these approaches, exploit naturally occurring genetic variation. However, when you don't have sufficient variability, as Dr. Vidyut Sharma said in his lecture, we have no other option but to use transgenesis breeding as a final option or the only option. Molecular biologists have adopted a number of strategies for imparting or engineering resistance to diseases in rice. They include detoxification of pathogen virulence factors. Some of the pathogens produce toxins, and here comes the detoxification strategy. And then there are genes called R genes, resistant genes, and you can overexpress this resistant gene. This is another strategy. And one of the some of the popular examples of overexpressing pathogenesis-related genes like chitinases, vulcanases, chromatin-like protein, and others, osmatin-like proteins. And also another approach is to modifying modification of different signaling pathways. So these are all some of the strategies by which people have developed transgenic disease-resistant lines in rice. At the same time, plant pathologists and molecular biologists made extensive genetic and genomic studies to understand rice innate immunity and they have identified a large number of targets for control of disease pathogens and as well as inhibition of pathogen infection. So, plant have evolved resistance genes, we call R genes, that can neutralize pathogens, derive virulence proteins, or effectors, and activate effector triggered immunity, we shortly call ETI. So, besides, plants also do have susceptibility genes, which are essentially involved in successful pathogenic infection. So, in the context of genome editing, the primary strategy for disease resistance involves knocking out these susceptibility genes through the error-prone non-homologous end joining pathway-based repair of target DNA. One of the key in genome editing technologies to create a double-stranded break, site-specific double-stranded break. Once a double-stranded break is created, the cell takes over to repair the abnormality. When it repairs, it uses non-homologous end joining pathway, which is error prone. Though the repair is done and the repair is not perfect, as a result, you have indels. So this is one of the primary strategy people have used to develop disease-resistant lines in several crops, including rice. Besides, it is also possible that possible to introduce resistance genes through homology-dependent repair pathway to introduce the gene into a specific site of interest. And as well, and also another important factor is that the alleles of certain R genes and S genes, susceptibility genes, vary at single nucleotide level. So by using base editing or prime editing, it is also possible that we can generate new alleles for disease resistance. Targeted mutation can be achieved through genome editing, which conventional plant breeding always develops the mutant varieties, but there the mutation is largely mainly random, and it is a tedious process. However, with the advent of this technology, it is now possible to effectively target a specific gene or a specific nucleotide can be targeted now with the genome editing tools. And such genome edited plants are called SDN1 type modification, and they are considered transgene free. Particularly in some countries, including US, this genome edited mutants fall outside the regulation, which was well described by the earlier speaker. So one of the good example for imparting disease resistance in rice through genome editing technologies, bacterial blight resistance. Bacterial blight is a devastating rice disease, and many of the popular cultivars are highly resistant to this disease. And this disease is caused by a pathogen called Xanthomonas oryzae petavar oryzae. And this pathogen produces a protein called tail, transcription activator like effector proteins. And this protein is secreted by the pathogen, and it enters into the cell, right, plant cell, host cell, and then enters into the nucleus, and then binds to DNA, specific DNA sequences. And these sequences are called effector binding elements that are usually present in promoter region. And then once they bind it, they drive the expression of susceptibility genes. Susceptibility genes then overexpress to produce its protein, and which facilitate the infection process and support the compatibility with the pathogens. In short, the susceptibility gene expression gives a conducive atmosphere for the pathogen to survive. That's the reason why we call this a susceptibility genes. In rice, there are several sweet genes are available. 5 sweet genes, 11, 12, 13, 14, and 15 are considered to be a susceptibility gene because they support the virulence of Xanthomonas oryzae and activate there. And these genes are activated by different tail proteins. Not by the same tail protein secreted by Xanthomonas. Different strains secrete different tail proteins, and then they bind to different EBE elements in the promoter. So that means different strains are responsible for expression of different tails, and these tails bind to different, part of the promoter of susceptibility genes. In this case, sweet gene. So if you want to have a broad spectrum, resistance, or if you want to have a rice line which is highly resistant to bacterial leaf blight, and then you have to disrupt all the EBE or uh, effector binding elements. So to this point, Zhu and co-workers recently have generated rice lines with broad spectrum bacterial blight resistance by simultaneously disrupting variable chain binding elements of three susceptibility genes, whereas OS sweet 11, 13, and 14 in rice, and then they have reported that their the rice line developed is resistant to almost all the known strains of Xanthomonas. So this is one good example, and another example is on blast resistance. Plant ethylene responsive factors are transcription factors that are involved in modulation of multiple stress tolerance, and various studies have shown that overexpression of some of the ethylene responsive factors contribute to susceptibility to diseases. So that means these ERFs are negative regulators. One such ERF is Rhiza sativa, ethylene responsive factor 922. Its overexpression, made the plant highly susceptible to blast disease, and then Wang and co-workers in one of their studies, they mutated this ERF 922 through genome editing tools, and then they have shown that the edited mutants are resistant to blast. So, based on encouraged by the potential of this technology in knocking out susceptibility genes to impart disease resistance, we attempted to engineer resistance to rice to grow through editing an susceptibility gene in rice. So, rice tumor is one of the major viral diseases in rice, and this occurs in most of the rice growing areas. And under severe conditions, it devastates the rice crop, leading to significant loss of yield. And this disease is caused by two viruses, a combination of two viruses. One is DNA virus, RTBV, rice tumor bacilli virus, and another RNA virus, which is the rice tumor spherical virus. Both are transmitted by leafhopper and hepatotic viruses. And RTSV acts as a helper virus, assisting transmission of RTBV. But infection by both the virus is very, very important for symptom development. Symptom development. So RTSV resistant cultivars has the potential to reduce rice tumor disease in the field. RNA viruses generally use host factors, host proteins that is produced by the plant cell, such as translation initiation factors for their survival. And these host factors are not only important for virus translation, it is also important for plant translation. So mutation in one of the genes of the host factors can interfere with the virus replication, but at the same time, we need to make sure that this doesn't create problem with the plant survival. So genome editing of host factor genes have been widely used in number of crops to generate virus resistant plants by several earlier workers. Taking a cue from this, we wanted to edit develop RTD resistance in one of the varieties popularly grown in Tamil Nadu. And RTSV resistance is actually available naturally in rice germplasm. It's not that this is not available in the germplasm, but this resistance is a recessive trait controlled by the translation initiation factor for gamma gene, EIF4 gene shortly. And this was reported, as early as in 2010 by a group of workers And this protein has essential roles in the assembly of basal translation initiation factor at the 5' end of the mRNA, as well as in the cap independent translation of viral RNA genome. So this protein is very important for translation of plant genes as well as viral genes. You know, this is a part of the gene EIF4G gene. I invite your attention to this portion of the gene, and it is in exon 7 of indica rice. You know, there is a mutation. There is a mutation which is from valine to alanine at 1060th position, and this confers resistance to spherical virus as per the study conducted by Lee and co-workers. And all these resistant genotypes are, they have mutations in this T residue. That is, this valine is converted into alanine. In one case, TKM6, which is, Tamil Nadu, spread in Tamil Nadu, this has, mutation deletion of 3 nucleotides. There is no valine residue here. So this confers resistance to spherical virus. So taking cue from this, we have attempted to develop rice Chungro disease resistant line using genome editing tool. We wanted to target this region. We wanted to target specifically this region or region surrounding this B residue to impart disease resistance in AST16. And this variety is one of the popular varieties of the state, and this is highly susceptible to rice Chungro disease. And though it is possible to introgress this gene from the naturally available resistant sources, but this has a number of limitations. So we have attempted to introduce this mutation through genome editing tools. What we have done is we have using software, we have identified guide RNAs, to target the YVV residue, the residue which I have shown you in the previous slide. We have targeted sequences in the close proximity to the V residue, and then we have ended up designing four constructs, and 2 of them based on the sense strand, and 2 of them based on anti-sense strand. So as you know, this depends upon the availability of PAM sequence, and we exploited the presence of PAM sequence to generate 2 different constructs using upper and lower strand. And we have used agrobacter mediated. We have cloned this in a vector PRGB32, which has Cas9 gene driven by ubiquitin promoter. It also has a hygromycin phosphotransferase gene, which is used as a plant selectable marker gene in this study. And we have regenerated plants, and then these regenerated plants were screened for the presence of hygromycin Cas9 beside screening the target region. So we have a robust rice transformation protocol, and this protocol based on immature embryo, which is infected by agrobacterium culture, and we use hygromycin selection. And this system is so perfect, and then we don't get any escapes from this. And we have generated over 180 events, edited events. And then these events were screened by PCR as well as Sanger sequencing, and the sequences were then analyzed with the softwares like TST code and CRISPR ID. So using these constructs, we have generated, as I said, more than 180 events, and out of after all of them, I'm sorry about that.
Participants, please don't mark and don't disturb. Participants, please.
I don't know if I have done this. I don't know how to remove this also. I'm sorry about that. How do I remove? I don't know.
Sir, it's okay, sir. Once
Okay.
you...
Right. Yeah. So we have generated large number of lines, and the lines were tested for hygromycin phosphotransferase gene as well as Cas9. And as I said, the system is so effective selection system, and we didn't get any escape. But we have seen only 89 mutants out of this 180 plus events, and the mutation frequency ranged from 32% to 62%. And a majority of the mutations, based on our analysis, were monoallelic, and of course we have seen biallelic mutants also, and we have seen both deletions, insertions, as well as substitutions. But deletions were most frequent. But in our observation, when this T0 lines went to T1 generation, most of the insertions and substitutions have gone missing, and then finally we had only deletion events. And these plants were morphologically normal. Most of the mutants are frame shift mutations, and resulting in truncated, 4G protein in one of the alleles. We did, we predicted the possible amino acid sequence, and in case of frame shift mutation, generally they result in truncated protein. So these are all some of the, this is a table, this is a representative of some of the lines which we have generated. You see more monoallelic and biallelic mutants here. And then this is the, again, T0 events, some of the T0 events where I wanted to show you this how, the mutant lines appear sometimes. Rarely you get biallelic, mutations. And even in you normally you don't get homozygous mutations here, but though it is theoretically possible, we didn't get any homozygous deletions or insertion, but we got monoallelic and biallelic in even in biallelic, the mutations are different in both the strands. And then you see here, most of them are frame shift mutations. As a result, the translation. This is a prediction. Actually, the translation is truncated here because these lines are, in monoallelic condition. These plants could survive. Probably, if this plant had this mutation in biallelic condition, this plant would not have survived. The near normal EIF 4G gene, 4G protein is essential for plant survival. And then we have forwarded this to T1 and T2 generation. And in T1 generation, we have identified, we have not forwarded actually all of them. We have selected only 35 in-frame mutations. We have removed all frame shift mutations, and we used only in-frame mutations, and we had 35 in-frame mutations out of 89 total mutations. And out of them, so far we have identified 20 homozygous mutants, and this we are planning to conduct other studies on these plants. And these plants are homozygous mutations, and many of them are transgene free. Many of them are transgene free because in T1 generation, through segregation, we have identified only homozygous mutants which are free from hygromycin phosphotransferase as well as Cas9. So theoretically, these plants are not transgene free. And this is, this is some of the 18 of the 20 homozygous lines which we have identified so far. And here the mutation, most of them are, almost all of them are deletions. Deletions ranging from 24 nucleotides to just 3 nucleotides. And this is the predicted amino acid sequence. And in all the cases, we have near normal EIF4G gene. That is the reason probably all the plants survived. All the plant looked very normal in T1 generation, and we did not see any abnormality in terms of morphology, in terms of fertility. Okay, so this is the predicted amino acid sequence, and we have, as I said, we have 20 homozygous mutant lines, and the Lee and co-workers have implicated YVV residue, deletion of YVV residue for resistance to RTSV, and we have one line P43-1, this particular line, which is which has deletion of I think more than 8 amino acids. that includes this B residue, and we expect that this line could be a resistant line. Of course, we have not done bioassay yet. We are planning to do bioassay on this, and in recently in IRRI also they have used this genome editing tools to generate mutant lines for RTSV resistance. And they have, apart from YVV, they have also suggested that the NL residue, which is just upstream of YVV residue, maybe any mutation in NL residue also could give you resistant lines. That is the suggestion they provided. And when we analyze our plants, and we have 11 mutants, and these mutants have mutations in this NL residue, and we assume that these are all the promising mutations, and we expect that these mutations are resistant to RTSV. And we are currently we are planning to do insect bioassay. Sorry, this is assay assay in bio studies in transgenic greenhouse. In conclusion, I want to make a few points. EF5G is essential for plant growth and fertility. So the mutation should be in such a way that this doesn't affect the plant growth. At the same time, it interferes with the multiplication of the virus. So presence of an allele encoding nearly full length of for EF5G with the localized mutations seems to be sufficient for plant survival. In addition to YVB residue, the NL residue within this amino acid sequence of EF5-4G are also critical to determine the resistance to RTSV. So McCovey and suggests that this residue in the EF5-4G, which is located between EF4E binding motif and MIF4G domain, which are essential for the function of EFI4G. We assume that any mutation in this sequence will significantly interfere the interaction of RTSV protein with the EIF4G gene. That will severely limit the translation of viral proteins. So of the 4 constructs we have used in our study, P3 and P4 gave promising results with mutants which affect both YBB residue as well as NL residue. And these homozygous mutations are being evaluated for resistance to RTD in the bioassay studies. So besides, we have also initiated a experiment in which we wanted to target specific nucleotides very close to YVV residue, within YVV residue as well as region very close to the YVV residue using base editing approach. And we have also generated few plants, and then the results are also promising. And we haven't completed the analysis, so I'm not going to discuss the results of this experiment. And with this, I acknowledge the funding by NASF, my colleagues in the lab, and Dr. Reddy and Dr. Viswanathan for their support in the project. Thank you very much. If you have any questions, I will be happy to answer your questions. Thank you.
Thank you, Dr. Sudhakar. In fact, there were a couple of questions which you have answered in the course of subsequent slides. So we do not actually have any outstanding questions. Okay. But I compliment you for your success in genome editing in rice. And I'm sure some of these lines are going to come for commercialization soon. Okay, with that, I thank you once again and move to the next speaker, thank Dr. Viswanathan, a former colleague of mine and a friend, a very illustrious scientist at IARI. He is the head of plant physiology in IARI and one of the most productive scientists who has publications in the most prestigious journals like Nature, Science, and the like. And nowadays he is taking on the generation of genome edited crop, particularly rice, where along with Dr. Sudhakar and others, he has been making a great progress. I request now Dr. Vishwanathan to take the floor and try to, limit the talk uh, as short as possible, uh, without really compromising on the content. I know he is capable of doing it. That's why I'm requesting, because we are running greatly behind schedule.
Good morning, sir. Good morning, respected Chair, Dr. S.R. Bhatt, Emeritus Scientist, NRCPP, our former uh, professor of NRCPP, and also our teacher. Good afternoon, good morning to all the colleagues who are present here. First of all, I thank Dr. Fakhruddin for giving me an opportunity to present before you. Today, I'll be talking about some of the work which we are doing in our lab on genetic engineering and genome editing for improving abiotic stress tolerance of rice. So everybody knows that we have to produce 56% more food by 2050 without having any adverse impact on the environment, and also we don't have much land to expand. So we have to increase the productivity, and we have to increase the resilience of the crops, so we can grow crops in resource poor environment, and also sustain yield in the uh, well environment which are going to suffer from various effect of climate change. So these are the four major crops that contribute to 2/3 of the global agriculture calories: maize, rice, wheat, and soybean. Of which we have selected rice as a crop for work with. For various reasons, because rice is one of the major staple food crop of our country, which contribute to about 40% of the food grain production. And if you see the global freshwater scenarios, by 2050, India is expected to experience severe water scarcity level. And how to overcome the water scarcity and produce more. If you want to do this, we have to reduce the water use in agriculture, especially the crop rice, which uses about 50% of the water used in the agriculture. So how to proceed is a major challenge. As we know, water is the key input for the agriculture productivity. So we have selected the ABA dependent and ABA independent pathway. Try to engineer the rice crop with the genes from ABA dependent and independent pathway, and analyze ABA stress tolerance to see if we can reduce the water requirement and increase the abiotic stress tolerance of rice. We used both the genetic engineering as well as genome editing approaches. Why rice, or in fact the crops need so much water? Although water has several functions in the plant, most of the water taken by the plant is used by transpiration, and transpiration is the process through which plants takes carbon dioxide inside, and during this process, because it has to keep the stomata open for taking the carbon dioxide inside, it loses water to the environment. So if we how to make precisely manipulate the stomatal conductance so that it is not affecting the carbon dioxide inside, at the same time it is not losing too much water, for which is a major challenge. This is an important equation given by Pasiero, which says crop yield is equal to transpiration into water use efficiency into harvest index. So if you want to reduce the transpiration, we have to increase the water use efficiency and the harvest index so that we can increase maintain the crop yield. But the important factor is how to regulate the stomata. The answer came in about 50 years back. The scientists have identified a hormone called abscisic acid, which is the plant stress hormone. It was capable of closing the stomata. This discovery was made in 1969. And at the same time, scientists have also found that this hormone increases as the plant experiences stresses. And soon, within few years, it was shown that the plants which are defective in the biosynthesis of the abscisic acid are highly sensitive to drought and other abiotic stresses, and they cannot survive. So over the 50 years, several roles of ABA have been unraveled. It is key for stomatal closure that if the plant is impaired in abscisic acid biosynthesis, they cannot close the stomata and they can build very quickly, even if mild water deficit is there. ABA is important for inducing the root growth under drought stress, and it can provide extreme stress tolerance. That is, these plants can survive where the plants, wild type plants cannot survive. And it is also important for development. That is, if ABA is less, the plants are going to be stunted, and the other associated problems such as seed development and dormancy problem will be there. It is also regulating the biotic stress tolerance, but in this case, in case of necrotropic pathogen, ABA has been shown to regulate positively. That is, in the presence of ABA, the plant is more tolerant, whereas in case of biotrop, it has been shown that if ABA is more, the plant is going to be more sensitive. So it is very complex hormone, and precise manipulation of this hormone only under stress condition can help in the stress tolerance. Besides, ABA is also important for human beings. So this hormone is also synthesized by human beings, and it plays important role in the insulin release from the pancreatic beta cell, as well as it also inhibits the cancer cells. So a lot of search for over the 50 years led to the discovery of the abscisic acid receptor. 14 gene family members identified from Arabidopsis. So this has a ABA binding packet. Upon binding to the ABA, it changes its conformation, and then this results in sequestration of negative regulator of ABA signaling, and then this leads to the stomatal closure and stress responsive gene expression. So it can regulate both the stress responsive gene expression under stress condition, and it can regulate the genes for energy metabolism, and it can regulate the proteins which are the ion channel proteins present on the stomata, so that it can quickly regulate stomatal closure without need for gene expression. So the ABA signaling pathway thus play multiple roles in stomatal regulation as well as gene expression for stress responses. So once the receptors were discovered, a lot of effort is being made to use this receptor for enhancing the drought tolerance and water use efficiency. The simplest approach is to overexpress the ABA receptor and under stress conditions, so that we can enhance the stress tolerance. The other approaches are we can develop orthogonal receptors which can bind to the different off-the-shelf agrochemicals, and which can lead to stress tolerance, or we can identify a cheap agonist that specifically activates certain group of abscisic acid receptor. Unlike the other hormone receptors, the abscisic acid receptor family was too large. Like if you see the other plant hormones, maximum ethylene has 5 receptors, but the ABA has about 14 receptors in Arabidopsis. We analyzed the ABA receptors in rice. We found about 13 receptors are there, and these receptors can be grouped into 3 subgroups. And of the 13 receptors, we identified there are 2 receptors which are non-functional because of the lack of either C-terminal or the N-terminal domain. Remaining all the 11 receptors are functional. So to understand whether all the receptors, their function is redundant or non-redundant, we initially analyzed the expression pattern of these receptors under in the different tissues, and we can see the specific regulation of certain receptors in seeds. And some receptors, they are highly expressed in the panicle and some are expressed in the root. So they have different tissue specific expression during the development. And also if you see the stress response in response to drought and heat, these receptors are at least induced in one of the tissues in one or more stress conditions. So this suggested that these receptors are not redundant, then they may have diverse role. And studies from Arabidopsis also suggest that it has a diverse role, but still the individual receptor role is being investigated. So we used the functional genomic approach to validate the function of the receptor by using the stress inducible overexpression, that is by using RD29A promoter to overexpress the receptor, or we used the constitutive promoter to overexpress the AB receptor in rice, and we also tried the RNAi approach. So we developed transgenic for all the receptors. So I'll be presenting two results for the two of the receptors with contrasting results. One is the PIL6 receptor, which we have confirmed by southern blotting and as well as the real-time RT-PCR. The transgenic line produces more transcript in the drought stress, and it also has relatively more transcript under non-stress condition, as our later analysis found that the RT29A promoter can express at 32 degrees Celsius, and the RNAi lines produced less transcript. And then we analyzed the abscisic acid content, and we found that the abscisic acid content was higher in all the transgenic lines, which overproduces the receptors. And then further we analyzed the causes for this one, and we found that the rate limiting enzyme coded by 9,6-epoxycarotenoid dioxinase gene family is highly upregulated in these transgenics. Interestingly, we also found that when the RNAi lines were also producing high amount of abscisic acid, and that suggested that the certain level of PIL6 is important for the homeostasis of ABA level in the cell. And we examined the ABA induced root growth, that is under mild stress, ABA induces root growth in the plants. So we examined whether the transgenic plant produces higher amount of root. 3 of the 5 event, it produced significantly higher amount of root in the transgenic lines. And then that helps in the water mining under stress. So we also examined the stomatal conductance. So wild type plant, non-transgenic plant, they had higher transpiration rate and thus the higher excess leaf water loss. The RNAi lines, these two lines followed by the wild type plant, they also had the higher water loss rate, but slightly less than that of wild type plant. However, the transgenic lines, they had a less amount of water loss. The number of stomata were almost similar in the transgenic plants overexpressing ABA receptor PIL6 as well as the wild type plant. However, if we see the number of closed stomata under stress condition at about -50 kilopascal soil metric potential, the number of closed stomata were higher in the transgenic plants. Then we assess the whole plant water loss over a period of 50 days. And then this indicated that the amount of water that used by the wild type plant in 30 days is sufficient for the transgenic plant for 50 days. And this difference need to be analyzed in terms of per unit amount of leaf area, because that determines the amount of water used by the plant. In the per unit leaf area basis also, the transgenic plant used about 22 to 27% less water under the greenhouse condition, although the differences may be higher under the open field condition where the VPD is high, which need to be examined. And then we examined the stress tolerance at various stages. This is at the seedling stage, and these transgenic plants, which are shown in the right side, they have better recovery when compared to the wild type plant, and this graph shows the survival rate. So the transgenic plants overproducing the AB receptor, they had better survival rate when compared to the wild type plant, and the RNA lines behaved similar to that of the wild type plant. We also examined the various genes which are important for stress response that are regulated by ABA signaling pathway, and we found that many of the genes are upregulated higher than that of the wild type plant in the transgenic plant, and in the RNAi line these genes were downregulated. We also analyzed the stress tolerance at reproductive stage under greenhouse condition. This slide, it shows that it has better stress tolerance in terms of relative water content or the membrane stability, less injury to the membrane. However, after recovery, when we analyzed the yield, we found that the transgenic plant that overexpressed this receptor, they produce significantly less yield and biomass under control condition, whereas under stress condition, no advantage in the yield was observed. And when we analyzed the overexpression lines, we found that they produce less number of grains, mainly because of the spikelet sterility in these lines. So within the same family of the subfamily of the two, we also analyzed another receptor transgenic. Again, it can provide high level of stress tolerance, but it produces a lot of spikelet with sterility. So this indicates that this subfamily manipulation is difficult to stabilize the yield under stress while maintaining the yield under non-stress condition. So we also analyzed the transgenics of subfamily 1, which showed that we confirmed these transgenic by Southern and qPCR analysis. And again, we analyzed the same ABA level. Here also the ABA levels were high, but it is not as high as the other transgenics. And this provide tolerance to cold stress. That is, the wild type plant cannot survive the cold stress, as rice is highly sensitive, whereas the transgenic plant can survive the cold stress. And then we also analyzed various genes for cold tolerance, and the transgenic plants had higher expression level of these genes. We also analyzed the stress tolerance, drought stress tolerance of these plants. And we found that some of the events, they can produce higher yield under drought stress while using the less amount of water. This is done only under the greenhouse condition. So currently we are planning to do it in the confined, the transgenic net house, which we have recently developed. And again, the gene expression analysis clearly showed that the ABA signaling pathway genes are upregulated in these transgenics. So similarly, the other members of this family were analyzed, and they also showed enhanced drought tolerance, and they produced a significant amount of higher roots and without any yield compromise. With this, like I conclude the ABA receptor part, and we are also working with the another gene which is known as inducer of CBF expression 1, which regulate cold tolerance through different pathways. So transgenic rice plant overexpressing the ICE1 was generated, and it was analyzed molecularly. And we confirmed that this transgenic plant expresses higher amount of transcripts of the ICE1 target genes. And we analyzed the cold tolerance of the AT-ICE1, and it clearly showed that these plants are more cold tolerant as compared with the wild type plants. And at adult plant stage also, these plants produce, two of the even produce significantly higher yield. In case of salt and drought stresses, these three events which we analyzed, they produce higher harvest index and higher grain yield, both under salt stress as well as under drought stress. And interestingly, under non-stress condition as compared to the ABA receptors, which had some impact on the yield, depending upon the receptor used, these transgenic, they produce significantly higher yield in 3 different experiments. And then we also are working on the genome editing. As Professor Sudhakar explained the basis of genome editing, I need not explain it again. So one of the genes which is known as drought and salt tolerance, which was identified in 2009 by using EMS mutagenesis in Japonica background. And this gene encodes zinc finger transcription factor, which can confer both drought and salt tolerance. So we also looked for this mutant in the Nagina 22 mutant population, which the NRCPB has generated under the leadership of our Director General, Dr. Mahapatra. So he tried to identify a similar mutant, but we could not get a similar mutant, although we got one mutant which was better in tolerance, but this mutant had some problem with the yield rate. So we could not use it further. So then when the genome editing technology came and the NASF funded the project for us, so we attempted to engineer or edit the DST gene as a test case. So the DST gene has a zinc finger domain here, and it has two this ELF motifs, and we try to edit this gene by removing the C-terminal part of it, because this part is required for interaction with various other proteins. So we developed two guide RNAs, and these two guide RNAs were cloned in the vector, and we generated large number of plant and confirmed the mutation in the T1 generation. And then in the T2 generation, we identified the homozygous line. Four different alleles of this mutant was selected, and we selected one of the mutants for characterization, where in T3 these mutants they lack tDNA insertion, and it has a larger deletion in the C-terminal part, which is expected to give the stress tolerance. So as expected, similar to that of the EMS mutant reported in 2009, this mutant produces broader leaf, and it used a less amount of water. That is, when compared to the wild type plant, the transgenic, the genome edited lines, it used less amount of water. And we also examined the stomatal number. We found that the stomatal number was significantly less in the genome edited DST rice line. And then we also analyzed genes involved in the development of the stomata, and we found that three genes, speech, mute, and ICE1, these were significantly less in the DST1 mutant plant. Since DST1 is a transcription factor, we analyzed the DST binding site in this promoter, and we found that in case of the ICE1 as well as in speech1, we found the DST binding site. So further needs to be validated. So under seedling stage, we examined the stress tolerance to the osmotic stress and to the salt stress, and we found better results with the salt stress. So the survival rate after stress was significantly higher in the salt stress condition as compared to wild type plant. And after recovery from the salt stress, the recovery rate was also better in this one. So currently these plants, we are evaluating the stress tolerance of, for drought and salt under the adult plant stage in the transgenic, transgenic greenhouse condition. So to conclude that the ABA receptors have non-redundant role in stress response and plant development. So ABA receptor engineering can enhance the water use efficiency and tolerance to drought, cold, and salt stresses, but we have to precisely regulate in the vegetative tissues only, as in the reproductive tissues it causes sterility and other associated issues. And the ICE1 can be useful transcription factor as it enhances the multiple stress tolerance, and also it enhances the yield under non-stress condition. CRISPR-Cas9 mutagenesis is a useful tool for creating stress-tolerant plant, and testing of the developed transgenic and genome edited lines under field condition is necessary, because under greenhouse condition the VPD is less, and also the real environment test is necessary to understand the stress tolerance of these plants. So to conclude, I thank the funding agencies, ICR, NASF, who funded this project. Our chairman was also one of the committee members for selecting this project. So I thank all the committee members and the NASF for providing fund for this genome editing project. And also some of the transgenic developed in this presented in this presentation was funded by ICR, NASF, as well as DBT Government of India. I thank my students Santosh, Shashank, Rakesh, Pragya for helping in for doing the actual work of developing transgenic, and Suchitra for all the technical assistance. And I also thank my co-PAs, Dr. Archana, Shivani, and Nagaraj. Finally, I thank Dr. Fakhruddin for giving me an opportunity to present before you. Thank you very much for your patient listening.
Thank you, Dr. Vishwanathan, for an excellent presentation. This is what happens when a fundamental research on ABA signaling and ICE1 discoveries where Dr. Vishwanathan was the pioneer. He has taken it to practical applications. I'm really glad that the project on genome editing that we gave as a case under the National Fund is bearing fruits now. And with these sort of transgenics or non-GM, whatever you call, I think some of the major oppositions to transgenics will also reduce. And secondly, these are the sort of technologies that we would require to address the climate change scenario that is staring us. With that, I thank you, Dr. Vishwanathan. There are a couple of small questions which you answered live because people have been shooting questions in between, but they were taken up. So because we are running short of time, I thank you once again.
Thank you, sir.
And
Thank
move
you.
on to the next speaker, Dr. Vijay Paranjipe. Dr. Paranjipe is an alumni of JNU, and he is currently working with the Sadhguru Management Consultancy, and he is advising the Bangladesh on BT Brinjal project, and he has a wide experience in the industry where he has worked in different capacities to devise R&D strategy, technology scouting and assessment, IP strategy, business growth, and whatnot. So thus he brings in a very rich experience from a private sector, which is playing a very crucial role in taking these modern technologies to the farmers. I now request Dr. Paranjipe to deliver his invited talk. Thank you.
Yeah, thank you, Dr. Bhatt, and thank you, the organizers, for inviting me for this presentation. So our earlier speakers have talked about what research goes on in the lab before a product is commercialized. So I think now the story is slightly different. I would be talking about a product which is already commercialized. I feel many in the audience must be feeling that BT eggplant story would have been a good story, would have been a good successful story in India, but for the known reasons, it could not happen. But I think there is an opportunity to learn from our neighbors how they have adopted this technology and make it successful and grow. I'm moving on to the next slide. So I will start with giving some background, maybe to set the context. Then this is an example where a public-private partnership had played a very significant role in making this technology succeed in Bangladesh. And I will touch upon the progress which has been done so far, because the product was launched about 6 years ago. So what the incremental and the strategic steps were taken during the last 6 years, how the adoption increased, the coordination between the different public sector stakeholders to make the project successful, building across the value chain, and finally, how the economic and health benefits which have been derived by BT eggplant for the farmers. And finally, I will be mentioning about the way forward to sustaining this technology in Bangladesh. It's eggplant is a known food. I think it's we make bharta, we make good curry, we have cuts. So I think this is a known food in Asian countries. And if you go to Bangladesh, this is very common. When you go to restaurants or anywhere, you will find this delicacy available there. Eggplant is, this particular slide just conveys the meaning where eggplant is mainly grown in India and China. And apart from that, it is also grown in other countries. But the major production happens in our region. So it is very important that we protect this particular crop in our region by available technologies. As we know, the major destruction is caused by the brinjal shoot and food borer, and it causes around 60 to 70% damage for the crop. And the insects can cause damage to the shoot, stem, then the fruit. As such, and I have experienced this in India also. And whenever you go to market or you get some brinjal and you cut it and you see the insect inside. So this is a very common pest in Bangladesh as well. And if you see, this particular pest is because it's found mainly in our region. So it is very important that we control this pest and help to sustain the growth of eggplants. So brinjal in Bangladesh is the second most widely grown vegetable crop in Bangladesh after potato. And about 1.5 lakh resource poor farmers are growing this eggplant in 50,000 hectares of land. And the most important thing to note here is the farmers are spraying nearly 80 to 100 times of insecticides to control this pest. And this story is similar to what had happened in India, where the farmers were spraying so much amount of pesticides to control the bollworms. Let us see how was the technology access. So Mahiko, the Indian company, developed the eggplant expressing the CRY1 AC gene, which could help to control the fruit and shoot borer. Later, in partnership with Mahiko, USA, Sadhguru Management Consultants, and Cornell, this particular event was transferred to Bari. Bari is the Bangladesh Agriculture Research Institute, which currently owns the technology in Bangladesh. And this EE1 event was backcrossed into about 9 varieties, of which 4 varieties were released for commercial use in 2013. I would like to mention here that the technology was also made accessible to India, including UAE, Dharwad, and also in Philippines, but they are yet to be regulated and commercialized. In Philippines, it is at the regulatory stage, so I'm sure very soon we should see an approval coming out of Philippines. As I said earlier, this is a very good example of a public-private partnership and delivering a GM product to the farmers. So on the left-hand side, you see the players which provided the financial, technical, and the project management support. So Mahiko provided the technology, USA provided the funds for driving this project. Cornell University and Sadhguru Management Consultants were involved in providing the technical support as well as managing the partnership between the private sector and the public sector. On the right hand side, you have the implementing partners in Bangladesh. They are the Bangladesh Agriculture Research Institute, doing the research, doing the biotech research, and basically owning the technology there. The Department of Agriculture Extension is the one which promotes the technology amongst the farmers by distributing the seeds, talking about the technology. That's what their job is. BADC is involved in the actual seed production. It's an equivalent to National Seed Corporation in India, where involved in seed production. And Ministry of Ag is what overseeing the entire activity in Bangladesh. As I mentioned, this project was supported by USAID, and there were 2 projects. There was one project from 2004 to 2015, which was the ABSP2. I think some of you from US, Harvard were associated with this particular project. And from 2015 onwards, it was a new project called the Feed the Future Biotech Partnership Project Improvement Project. And I have been associated with this project since 2015 now. This slide basically summarizes how the technology was made successful with taking some strategic and incremental steps. So after the approval in 2014, just 20 farmers were provided this particular seed in just 4 districts. 2014 onwards until today, Bari has been carrying out demonstration trials. Nearly 2,000 demonstration trials have been carried out in Bangladesh in different eggplant growing regions, basically taking the technology to the farmers, demonstrating the technologies so that they adopt it in a better way. Later on, when the number of farmers started increasing, the demand started increasing, and 2016 onwards, the Department of Agriculture Extension is another government body, started distributing the seeds for wider adoptions. So they have a bigger reach. They went out to more districts in Bangladesh to talk about the technology, distribute the sample seeds so that the farmers get a feel of this particular technology. So 2016 onwards, there was an increase in demand. So there was a need to produce the seeds. So Bari, being a research organization, was incapable of producing a large quantity of seeds. So the production started with BADC, which is the Bangladesh Agriculture Development Corporations. And since 2017 onwards, BADC has been producing seeds. Later on, we realized as a part of project management that three different government organizations don't talk to each other. So we had to bring all these three organizations on the same platform. And then for the last 3 years, we have been all working together, having regular meetings, exchanging thoughts, exchanging ideas, learnings from the previous seasons, planning for the next season. And I think from 2018 onwards, all these three stakeholders, Bari, BADC, and DE, are really working in coordination and in sync to make the technology successful in Bangladesh. Yeah, this slide basically describes the adoption. As you see, in the last 6 years, the technology has been adopted very gradually, with the number of farmers increasing. So for the first 3 years, it was just 250 farmers, and 2017 onwards, when two different organizations started contributing, like the extension division, the seed production division, the number of farmers adopting this technology is increasing. And as a result, the area is also increasing. So currently about 18% or less than 18% farmers are using the eggplant and the BT eggplant, and around 5 to 6% of the acreage is under BT eggplant cultivation. The BT eggplant in Bangladesh is a variety, so the farmers have an option to maintain the variety. And we did a survey for the last 2 years, seeing how many farmers retain the seeds and use it for the next season. So we did find about 15 to 20% farmers do keep the seeds for the next season, so that we estimate, so for the 2019-20 season, around 30,000 farmers may have cultivated the BT eggplant. Going to the next slide. Yeah, and this slide I can just quickly, due to short of time, I won't spend a lot of time on this slide. Basically, this describes the role played by different actors, Bari, BATC, and DAE. And I think it has been because of this coordinated effort that the technology is slowly establishing, and more and more farmers are adopting it. And that has contributed to the success of this technology. And as a result, the supply chain is being maintained. Though the demand of seeds are slowly increasing, so there is a need going forward to increase the production. So one thing to note is that it is completely the public sector which is driving this particular activity in Bangladesh. Stewardship, capacity building, and communication are the three pillars which have made this technology successful there. So civil stewardship includes adopting field stewardship as well as laboratory stewardship, ensuring the genetic purity, the viability, and the protein levels, expression of protein levels, and at all stages of plant growth. Promoting planting of refuge. This has been challenge. It was a challenge in India in cotton. It's a similar challenge in Bangladesh. We have to explain to the farmers why there is a need for refuge. But as of now, we do not see a major challenge, because it's less than 6 to 7% of acreage is under BT eggplant cultivation. But maybe over a period of time, when the acreage increases, we may see some resistance building up. So we are encouraging farmers to plant refuge, so that there is a delay in the buildup of resistance. In-season monitoring and all those things are always going on every year. Capacity building. So being a public sector organizations, people change. As a result, we have to ensure that people who come on board for working on this technology are properly trained. The training is provided for the labs, for the field, for the seed producers, for the dealers who are now selling the seeds, extension professionals. The training has become a part of this project for the last 6 years, and it has to continue. Communication. Communicating of the technology is also important to promote this product in Bangladesh. So as in India, there will be people who do not like this technology, and it's our responsibility to promote a science-based public awareness. So we do it via publishing blogs, scientific publications. We have a dedicated communication team in Bangladesh as well as in the US. There's a website provided to you. You can go to the website. It can tell you more about this project and different communication efforts carried out so far. We've sensitized the farmers via TV, radio, demonstration trials. We go to the fields, talk to the farmers, and farmers are really interested. When you show the product to them, they really jump on it, and they just ask for the seeds. We develop the videos so that they can be shown to the farmers. So these are all like communication related activities to promote the technology in Bangladesh. A digital initiative of developing a BT Vegan app. So as you see, due to COVID, it was very difficult for us to reach out to the farmers. So I think we launched an app which helps to connect with all the stakeholders, right from the seed producers, the trainers, the extension professionals, the scientists, as well as the farmers. So all the information we have made available to the farmers via app, and the farmers also can interact with the technology providers. So this has been launched just this season, and now our job is to see how farmers would be use this particular technology made available to them. Yeah, these are some pictures of some extension activities which we carry out, which could be in terms of farmer training, field days, courtyard meetings. Courtyard meetings are the ones where we gather farmers in a particular small corner of in a village, chowk, and then we try to explain to them about this technology. And we have used the national media platforms like TV, radio, and to promote the technology. Along with the seeds, we also give a small pamphlet which talks how to plant, what agronomic practices to be adopted, what controls should be taken, the planting of the refuge, and we suggest all the guidance we provide to the farmers. So this is some insert which goes along with the seed. We have these posters displayed at the dealer shop, and we try to use this material to promote the technology amongst the farmers. Yeah, this is the app which we had recently launched. It is an app available both in English and Bangla. And it's very simple, user-friendly, which on a click of button, you have everything on the fingertips, both for the all the trainers, the extension professionals. You can know about the technology, the seed source. If the farmer wants to know where the seed is available, you just click there and list the agencies and the dealer shops where the seeds can be purchased. What are the cultivation practices to be adopted, stewardship practices, FAQs and myths to explain any questions which the farmer may have. And then at the bottom, you see we have some options to enter the information. So farmer can enter the sowing data, harvest data, and post a query to the technology. So, and Bari will monitors the data which comes to them and helps them to plan for the next season. Since the product was launched, so now we have conducted several studies. Some independent studies have been carried out to really understand the benefits, economic benefits, as well as the health benefits derived by this particular technology. So as you see, there's a negligible infestations by fruit and shoot borer compared to nearly 50% or 60% field damages caused by the insect. And if you really visit the field, this picture is similar to what Dr. Prakash showed for BT and non-BT cotton. And there's an amazing field that's completely controlled. You don't see any insect, no fruit damage, wonderful fields to see in Bangladesh. There is a nearly 61% savings of pesticides, and this is something when we conducted the survey just last season, farmers pointed out this particular benefit most rather than the economic benefit. They're saying that we are not spraying any pesticides. Our children don't use, they don't touch the pesticides, and this is something which which came out of this survey. They were not worried about the yield which they got because nearly 6 to 7-fold yield. The farmers obtained a 6 to 7-fold yield, but the most important point which impressed them was the avoiding the use of pesticides. So I think that's something good, and really this is the technology is available which really helps the farmers not only from the health point of view but also economically. Yeah, I'll also skip this particular slide. This is just a study which we had carried out to see how the technology is being currently adopted. This we carried out just last season, a survey which was carried out by the third party. So one thing I just wanted to point out here was the farmers said that they are increasing the use of labor. So now they require more labor to harvest, to grade, to pack. This is something is generating not a big employment, but it's a need of labor, and it also indicator that the increase the volumes of they're increasing the volume, so they need more people to harvest. And in case of eggplant, the harvesting goes on for 2 to 3 months. So every week they would like to harvest, and now with the availability of app, they have a way they can upload the harvest data, and then we are trying to see how we can connect them directly to the dealer or the consumer, the wholesaler who buys the eggplant from them. So I think this way we are trying to establish a market connect for the farmers. And also there was I think I can skip this one. Yeah, so this is the way forward. Now available in Bangladesh for 6 years now. We really have to see how do we take it, sustain it, and also grow it in Bangladesh. So it will start with development of new varieties, agronomically superior, and also now hybrids. We need to look into hybrids which are more suitable in different geographies. There are several varieties which are grown in Bangladesh. The currently approved varieties may not be suitable for the entire geography. So I think this is something different in India, where the sub-licensing model worked very well. The technology providers sub-license the technology to different seed companies, and the seed companies were able to take the technology to corners of the country. This particular model worked very well in India. So I feel that when we take plant, if at all, whenever it gets approved, we have a very good business model which has worked well for cotton. So now in Bangladesh, we are trying to see how we can bring in newer varieties. Again, we are taking help from MICO to develop this backcrossing and do the work for them. Implementing laboratory and stewardship practice, I think this is a very important things to really sustain the technology, and we continue to do it and do periodic audits and capacity building amongst the stakeholders. So this is something, an ongoing practice which have to do if you want to sustain a GM technology in any way. Development of a second generation product. So now we have not, it's not started yet, but definitely we have a 2-gene product in our mind to see how it can be developed, like the way we have in India, Bolgard 1, and then we had the BG2 or the Bolgard 2 cotton. Finally, we have to really work towards creating the enabling environment. Like policy level interventions are also required to promote an event-based approval process. Now, currently in India, we have an event-based approval process. So now once the event is approved, it does not have to go to any biosafety studies, but that's not the case in Bangladesh. So now we are working with the regulators and giving an example of India, like how the variety-based approval to event-based approval has really helped the spread of the technology. And the private sector participation for technology, for GM technology development in Bangladesh using the private companies in Bangladesh, and also dissemination is not done yet. And this is something we are trying to see how the government can enable this for the success. And so if the private sector also gets involved, more funds can come into these technologies for doing research, more varieties can be developed. So at the moment, the project is being supported by USAID. Maybe at there will be some time when the technology has to be owned in Bangladesh, and so we feel that the private sector in Bangladesh can play a good role. This is the team, the project management team which we have. We say diverse team with a diverse skills. So Marcelis Acevedo is a project director. She has an experience of implementing international projects, and she is a pathologist by training. I am a molecular biologist, having a training in having worked with industry on on cotton. Anthony Shelton is an entomologist and has been associated with the project for the last 15 years now. Dr. Jahangir was a director at Bari, and he is our to-go person in Bangladesh. Rituparna is a seed system specialist. Syed Hussain is an economist who we have on our team. And then Matt Hayes is our communication lead. And finally, I would like to mention here that this technology was successful in Bangladesh because of the political will. And the former agriculture minister said that we will be guided with science-based information and not by non-scientific whispering of a section of people. So and she was so firm that she said that we have to get this technology. And I think when the instruction came from there, the teams in Bangladesh, they really worked hard and ensured that they were able to develop this product and bring it out to the farmers of Bangladesh. And we feel that the success of BTech plant can pave way for future biotech products in Bangladesh. And this is my last slide. So I just want to draw your attention to our website. You can go to our website and you can get to know about the progress of this project, what we are currently doing on a plant in Bangladesh. And thank you very much for the opportunity. And I can take any questions if you have.
Thank you, Dr. Paranjape, for an excellent talk. In fact, your talk really highlights what it takes to take the technology, scale up as well as deployment on a very large scale. In fact, this is an aspect which is not much appreciated by public sector scientists who are more content doing the research part. I'm really thankful to you for bringing out this aspect. And also, this also illustrates, as you rightly said, how the policy makers' decision making is very critical for technology spread. I'm reminded of Dr. Norman Borlaug, who always acknowledged the role of Dr. C. Subramaniam played at that time for ushering in green revolution. The similar role is being played by Dr. Mathia Chowdhury, the Minister of Bangladesh, who has taken this bold step of bringing in BT brinjal, an edible crop. I think this is where I think our government has slipped in taking the right decision at that time for reasons well known to most of us. I thank you once again because there are a couple of questions like whether it is available in the white brinjal or only the other one. Your slides have revealed. That's why we are running short of time, and there are no more important questions that I can see on the chat box. If there is any, we can privately communicate with Dr. Paranjipe. And thank you very much.
Thank you very much.
With this, we come to the end of the invited talks. We are really running. We have only 15 minutes left, whereas we have 5 speakers. My only request is that all the speakers should restrict it because these presentations are more specific. Please try to finish it in 5 minutes instead of 10. This is also a challenge that I'm throwing to you all because we have to manage time. There is no way. So I request now Dr. Raut, Gyan Ranjan Raut from Orissa Agriculture University to make the first presentation. I would request you to skip the introduction and only highlight the take-home message in each of the slides without going into the explaining any of those. Thank you very much for your cooperation.
Thank you, sir, for kind introduction, and I'm very much grateful. And I will just share my slide. Hello.
Okay,
Hello.
Go ahead. Share your slide.
So just
Sir, already you are in Zoom meeting, sir. Just go to the share option, share screen option. Next one.
it's not coming.
If there is problem, we'll move to the next speaker. By that time, you can fix your technical glitch.
Yes.
Huh? Is the next speaker ready?
Just a minute. Sir, just a minute. Sir, just a minute, sir. Just opening. It's coming. Hello. Hello.
Yes.
Sir,
Please share your
this is
Okay.
yeah.
Now go to slideshow.
Yeah.
Click there.
This is the topic on the development of new movement genotypes resistant to powdery mildew through CRISPR-Cas technology. And all of you, this process and stable source majority of India 3 billion.
Sir, Rawat sir, kindly put on presentation mode, sir.
Okay, presentation. Sorry, you are able to see nice slide?
Yes, sir. Now it's going well, sir. Thank you.
It is the 3 major diseases which occur in case of the process, particularly in the Mubin, and this disease of the YAV, the Mubin yellow mosaic virus, and the CLS and the powdery mildew. And this is the PM is observed, PM is observed in all the growing stage, and particularly in the reproductive stage. And it is this disease is a very under the different climatic conditions, particularly in the season, it occurs in the PMD, and is loss is a huge loss in 40% occur in the region due to the lack of the varieties resistance to multiple diseases, particularly in the moving. And this is our hypothesis: the powdery mildew is effectively controlled by the knockout of the MLO genes in the moving. Occurring the powdery mildew, the white powder in the affected in whole of this plot, and PMD is the one of the we need to the engineers of the broad spectrum resistance to pathogen of the MLO using CRISPR-Cas technology. And many of the MLO gene is widely alleles in the conserved in the monocot and dicot. It convert to the PMD. And with a natural also MLO-based genes as they have been reported in barley, tomato, and peach. So I am not going to details, and this is a CRISPR-Cas technology. It's a very simplest, versatile, and genetic manipulation method. Two is a very important key molecule. One is an enzyme is called Cas9, a piece of the RNA guided RNA. And we use the analogous strategy to enhance resistance to powdery mildew in movement by targeting the MLO. The number of the gene MNO-like protein in movement, and this is the BRA, this is 210, and this is the BRA 30. It's most closely related of the P, and also on the clover. And we target the genes with a high moving variety, which is the single CRISPR-Cas technology. These as the two of the genes we have the incorporated in the high yielding variety, which is a highly YAV resistance IPM 0 to 14. And on this gene, our genotype, which is putting it through the genetic transformation system. And we have a screen to the T0 and T1 generation, and we have done the many events on that. This is the phylogenetic analysis, and which is these are the two genes, which is a MLO genes, which is going to the use for the Vigna radiata. And this is the use in the also reported in tomato MLO genes and in the other crops also. And this is the plasmid we have used on the plasmid, which has used for this for transformation. And this is the around is the 30% of the kilobase of the construct. And we have used verified through the PCR, and we see the different event we that they transform this colony by this using to the MLO genes. So we have a transfer to the plants and to the tissues, and of the transform the genome edited plants in T1, T2, and generation. And the number of the further we analyze to the PT2 because the neomycin is the antibiotic resistant, and then we have seen that all the event we have seen that this is the recognized by this our transport. So also we have seen in the primer of the Cas9 primer, and we have seen that it is a positive remark. And then we have did our experiment on this experiment of the under to the control plant versus genome edited plants, and we have also did the experiment on this the bias experiment. And on this point, and we have found that this is a plant we have having the resistance to powdery mildew. Compared to the control plant on basis of phenotypic traits condition, undergoing, and I am not able to putting all result here, and the optimization of the CRISPR-Cas genome editing and MLO gene knockout lines will provide the invaluable tool for me, not only wood but also the wood in it. This is my conclusion. And the acknowledgement is DBT BBRC, and also collaborator is our Northampton Research International United Kingdom and the BHU Institute of Agriculture Department. Thank you, sir, for this kind chance to speak about this topic on the genome editing and the new genotype production in movement. Thank you, sir.
Thank you, Dr. Raut, for a short and nice presentation. We are not taking any questions. There are no questions, in fact. Now we move on to the next speaker, Dr. S. Bhagyarani. She will be talking on enriching carotenoids in commercial cultivars granulated through transgenic approach. Dr. Bhagyarani, please.
Yes, sir.
Again, try to confine to 5 minutes.
Sir. Yes, sir. Sir, kindly ask them to unshare the slide, previous slides.
Sir, Rawat sir, pause your slide sharing option, sir. Rawat sir.
Yeah.
Pause the slide share option in the Zoom screen, sir.
Can you hear me?
Yes, madam.
Okay. I think you can see my slides also.
Put on the slideshow mode, madam.
Yeah, it is in the slideshow mode only.
Yes, madam. Yes.
Okay. Good morning, good afternoon to everyone. I am going to present the topic entitled on enriching the carotenoids in commercial banana cultivar granulate through transgenic approach. Actually, the carotenoid, which is a natural pigment which is available in plants, animals, even algae and bacteria. It is having the high activities, that is properties like provitamin A, antioxidant, and also it is involved in gene regulations as well as immune response. The main reason is the immune system is it supports the production of the cells, particularly for the white blood cells, and thereby our immune system is increasing. It is also having antioxidant activity, so it indirectly helps our health of heart as well as even skin. And it is having some of the carotenoid are having provitamin A content. It is also increasing our eyesight as well as it is also protecting the disease of age-related macular diseases. So based on the functions, even though more than 700 carotenoids are having identified, 6 carotenoids are having high health benefits, and based on the functions, they have classified into provitamin A carotenoid as well as the non-provitamin A carotenoid. But both are derived from the single carotenoid biosynthesis pathway. Only the branches are the byproduct, which is derived from the alpha branch and the beta branch, are having different functions. For example, alpha branch, which is having the lutein, it is the end product, is having the property of antioxidant, whereas the end product of beta branch, like beta carotene as well as the beta cryptoxanthin, which is producing provitamin A content, rich in provitamin A content. So if you are consuming less carotene, that is irrespective of the PVA as well as the non-PVA carotenoid, we are facing the problem of mortality, blindness, and impaired immunity as well as the brain development system. Even the less consumption of non-PVA carotenoid is resulted in 37% of the people are affected by cardiovascular diseases as well as age-related macular diseases. So our aim is to enrich the food that we are directly consuming, that is like banana. It will provide whether both carotenoid as PVA as well as the non-PVA carotene. So this is our main ambition. Based on that, we have chosen the banana. Since it is available throughout the year and it is a cost-effective, very cheap method, and it is grown in large area in India when compared to the other food crops. And moreover, these carotene are unstable for thermosensitive and as well as the temperature sensitive. So if you are storing it in a long time, there is a chance of losing the carotenoid content. So if you are enriching the fruits like banana, there is a chance of upon ripening the expression of carotenoid is also increasing. So we have chosen this banana as one of the main crops. And while seeing the diversity for beta carotene, it is amazing that Asupina, which is rich in, that is nearly 6,000 microgram per 100 gram of liquid, whereas our commercial cultivar is having only 64, that is nearly 100% fold higher than our normal commercial cultivar. So we have to transfer the gene from Asupina, even though it is a rich, it is not highly that is preferable by the consumers and poor yielder. But through conventional breeding approach, it is not possible to improve our commercial cultivar moving to its female sterility, and there is no seed set at all. So the alternate approach is transgenic approach. So in this, we have isolated the gene, that is phytoin synthase gene, which is the precursor of carotenoid biosynthesis pathway, which can produce by branching the end of both alpha branch as well as the byproduct of alpha branch as well as the beta branch. So we have chosen the precursor gene of the carotenoid biosynthesis pathway, and which was driven by the ubiquitin protein, and we made a construct. This construct has been obtained from the Dr. James Dale, UT, Australia, and that construct has been used in our granule cultivar. That is ECS co-cultivated we have made, and upon stringent selections, we have developed some of the transgenic lines, and they have confirmed through PCR, and the PCR confirmed plants have been planted in the transgenic networks. We are having the very good transgenic networks facility at ICAR NRCB, and in which the plants have been grown. Since it is to know the stable performance of the expression, the main crop as well as ratoon crop yield has been has been taken. So these are some of the transgenic lines which is showing 10-fold higher than the controlled grand line, normal grand line. So since it is a pigment, carotenoids are pigment in nature, so by simply seeing the pulp color itself, we can witness that these are all rich in carotene content. And the different gradation of transgenic lines have been obtained. So based on the high expression of the PVA content, the lines have been selected. So nearly interestingly, all the transgenic lines are having higher PVA content than the control, and it is we can say that from 5 to 77 microgram of that is PVA content has been obtained in transgenic lines. But nearly 6.7%, nearly 150 plants have been evaluated, analyzed for PVA content, and found that 6.7% of the plantlets are having more than 40 microgram of PVA content. And since, as I told, it's to know the stable performance, the same plants have been also analyzed in the ratoon crop also, and found that some of the plants are produced less PVA content and some of them are produced higher PVA content than the main crop. But we are very much interested in stable performing lines. So apart from the that is rich in carotenoid, we are interested in stable performance. So we have identified the stable performing lines, and they have further evaluated compared with the yield performance. So here the nearly 10 plants, that is 12 plants have been identified, which is showing stable performance in both main as well as the ratoon crop, and there is no yield penalty has been obtained for these transgenic lines. So based on these criteria, we have shortlisted to 12 transgenic lines. That is, and interestingly, it is showing that all the transgenic lines are rich in lutein, alpha carotene, and beta carotene. So in altogether, it is rich in carotene content. Suppose if the expression of this that is excess carotene, whether it is affecting the other quality parameters, that we have to confirm. For that, we have done some proximate analysis that other quality parameters such as TSS, acidity, and total sugar and total starch have been analyzed, and there is no difference between the transgenic lines as well as the non-transgenic lines has been identified. And simultaneously, the identified lines have been subjected to sudden analysis to know the copy number of the genes, and found that two plants showed that is double copy number, whereas 3 plants having single copy number. But one plant in one plant we could not be able to find out the banding pattern. And it is interesting that the plants which is having the 2 copy number is showing the higher expression of carotene content. So this information also we have obtained. So to compare the higher transgenic PVA content transgenic lines have been compared with the commercial cultivars and found that it is high fold. Even the Nandran, which is normally that is considered as a high PVA content, it is high carotenoid content than the normal Nandran content, which is rich in PVA content. Apart from that, but Nandran is low in lutein content. Here we can see in this picture, it is depicting that the lutein content is very high when compared to all the commercial cultivar. Even though the Nandran is rich in PVA, but it is low in high that is lutein. So here we got one line which is having rich in beta carotene as well as the lutein content. So both the carotene is enriched in a single transgenic lines. Here, the papaya, it is well known that it is that is fruit crop which is rich in beta carotene, and so for that we have compared with the PVA content of the beta carotene as well as the lutein content, and found that 5.6-fold higher PVA content than papaya and 8.9-fold higher lutein content than the papaya. Similarly, but this is the different lines. This is TGN.
Please conclude in next 1 minute.
Yes, sir. So similarly, we have identified the one line which is having both rich in beta carotene as well as the PVA content. And as a conclusion, based on the conclusion, we derived that nearly 4 to 5 granite fruits will meet the RDA of PVA carotenoid as well as it will meet the demand, that is meet the demand of 35% of the RDA of non-PVA carotene, that is lutein. So the consumption of 4 to 5 granite is sufficient to meet this demand. Then our future plan is that is we have to go for a biosafety analysis to further commercialization of these enriched carotene enriched banana. So a lot of visitors have been visited. That this is the Dr. James Dale who visited our transgenic networks, and he witnessed that the expression of high PVA content in this one. And other delegates have also visited and appreciated our effort. And I am here giving thanks to all the team of the biofortification projects and acknowledge these are all the review committee members who visited the transgenic networks and also witnessed the expression of the PVA content. And I want to acknowledge the DBT Biotech for funding as well as QUT Australia for funding as well as providing the construction. Thank you so much.
Thank you, Dr. Bhagirani. Good work. Keep it up.
Thank you,
I now move to next, Dr. Parameshwara, genetic transformation of sorghum for improved salinity tolerance. Dr. Paramesh, please again request that stick to the take-home message rather than explaining the slides. No explanation of southern or whatever. Please start.
Okay, sir. Okay, sir. Very good morning, sir. Can you see the slides, sir?
Yeah, please go ahead. Share your slides.
Yeah. I have tried. Okay, sir. This is the title of my presentation, sir. This is about the importance of sorghum crop. And I'll move on to the importance of salinity in agriculture. This is the important limiting factor for crop production. And there are several mechanisms for salt tolerance in plants. There are continuous attempts also to improve salt tolerance, which are met with limited success. Therefore, genetic transformation techniques needs to be tried. These are the 3 approaches we can improve the salinity tolerance in. And genetic transformation of sorghum is difficult because of recalcitrance nature of sorghum to tissue culture because of rapid induction of phenolic compounds and low efficiency of regeneration, and also genotypic genotypic specificity for regeneration. However, this challenge is met in our research program in development of salinity tolerant transgenic sorghum of sorghum, and also development of any selectable marker-free transgenic sorghum under confined conditions. This is a summary of genetic transformation of sorghum using different particle bombardment and agrobacterium-mediated transformation methods. Most of them are optimized with reporter gene, and they have got very good percentage of transformation with most of the times 3 explants being used, that is immature embryos and immature inflorescence, only 1 case with shoot apices.
Talk about your work.
Yes,
avoid
sir.
the others.
These are the advantages of the in vitro regeneration system optimized in our research. And these are the steps followed in multiple shoot generation, where we have used 5-day-old shoot apices, and shoot meristem induction was done with using BAP and 2,4-D. Initiation and multiplication of shoot buds was done using BAP and 2,4-D in different concentration. Same way, somatic embryogenesis was done using BAP, 2,4-D, and TDZ. Elongation was done using BAP and IBA different concentrations. Further, it rooted and acclimatization was done in the polyhouse after culture room. This biostick gun method was optimized using particle inflow gun, and these are the optimized parameters using PJS 108 plasmid with Gus and bar genes. In that transgenic Gus expression, there was maximum of 32 blue foci per explant, and all the explants were having at least 10 Gus spots. In agrobacterium mediated transformation of optimization, these are the parameters optimized, that is culture density of 1, optical density, and the time of infection of 20 minutes, and a co-cultivation period of 3 days was optimized. In both the cases, acetylcysteine engine concentration was 200 micromolars. This is a transgenic gene expression in agrobacterium mediated transformation, where you can see the very good infection and spread of Gus expression is there. A combination of antibiotics of carbonicillin and cefotaxime at 100 and 300 milligram per liter was able to control the growth of bacterium effectively in the medium. And this is the gene used in my research study, that is PCSRP gene, which is cloned from the wild grass species, that is Potracia coarctata, which is native to salt marshes of Southeast Asia and West Bengal. It is of 1.2 kb size and encodes for 280 amino acids, of which serine is being.
Go to your results, please.
Yes, sir. Yes, sir.
Avoid all these material methods.
Okay, this is the sensitivity of the explant estimated. That is around 300 millimolar sodium chloride concentration. The explants were unable to grow, and that was considered as most sensitive. And these are the steps followed in agrobacterium mediated transformation. We started with 1,500 explants, and
still you are in the material methods. Please go to result.
No, sir. These are all results. Sir. There are 15 putative transplants were obtained with 1% transformation in about 23 weeks. That is using agrobacterium mediated method. Using bioelastic gene gun method, 20 to 50 explants were used, and 16 putative transform plants were obtained with 0.71% of transformation. And these are the molecular characterization of PCSRP transgenic plants using both PCR and southern analysis. This is the result of the 16 putative transgenic plants. 15 were positive with the gene of interest. This is the southern analysis with 4 events, single site of insertions. And in the inheritance in the T1 transgenic plants, there was a very good perfect goodness of fit ratio with 3:1 Mendelian segregation ratio was seen in the T1 transgenic plants. And T2 transgenic plants were also giving very good southern analysis. And 2 of the events were found positive. All the plants which are there in the 2 events were found positive for the gene of interest. While screening for salinity tolerance in T1 regeneration events, better place with 200 millimolar concentration used, and the germination was tested in that. Out of 15 events, 9 were showing goodness of fit using chi-square analysis, that is being seen here. And T2 generation seeds screened for salinity tolerance were done in hydroponic solution with 250 millimolar sodium chloride stress, and 2 events were showing very good confirmation. Overall, 86% germination was observed in all the events. And this is after transferring them from hydroponic solution to the pots with 150 millimolar sodium chloride concentration, there was very good expression of these genes. And also there was very good correlation between the leaf and root ratio in the progenies of the events which we are obtained. And these are the conclusions we got. Very good regeneration protocol for sorghum genotype M35-1. With both the methods, we are able to get around 15 and 12 independent transgenic events with transformation efficiency of 0.7% and 1%. And molecular characterization helped us to confirm the stable integration of PCSRP gene in sorghum genome. That's all about my work. Thank you for the opportunities.
Thank you, Dr. Paramesh. good work with sorghum, which
sir.
is now also sequenced, opening up many opportunities for transgenic modification. I now move on to Ramakrishna. Dr. Ramakrishna will be talking about co-overexpression of DREB1 and BCZ8012 for drought tolerance in tomato. Again, Dr. Ramakrishna, stick to your results, no introduction, no this one, so that we can concentrate only on what you have achieved.
Yes, sir.
Please go ahead.
Respected chairman and co-chairman of this session, dignitaries, seniors, and my dear friends, myself Ramakrishna from Institute of Environment and Sustainable Development, Banaras Hindu University. I welcome you all on my presentation, co-overexpression of 80 driven BC Z12 increases drought tolerance and fruit production in double transgenic tomato, Solanum lycopersicum plants. Now, this is a key point about the tomato, though it's a botanically fruit, but often treated as a vegetable. Tomato is a rich source of lycopene.
Please avoid that. Everybody knows tomato here.
Okay.
I'm telling you, you go to your results straight away.
Okay, sir. Okay. So these are the basic point about drought. Now coming to the how I develop double transgenic. This is the detail of gene, whatever we have taken for the development of double transgenic tomato plants. Now we have previously developed double transgenic tomato, which expresses Arabidopsis thaliana, DREB1A gene, and Brassica carinata, BCZ12 gene. We just cross these two events, D86 and ZT1, to generate double transgenic gene, and we screen with a primary specific gene, and then selected 720 seedlings, and then we screen on visual basis, that is wilting. Then we advance the generation, and if homozygous F4 double transgenic tomato lines were evaluated for drought tolerance. For drought tolerance study, we expose 0 days, 7 days, 14 days, and 21 days of water stress. That is 80%, 40%, 25%, and 15% of the field capacity of moisture. The condition of the experiment were 50% relative humidity, 25 degrees centigrade day temperature, 15 degrees centigrade night temperature, and 16-hour light and 8-hour dark period, with a light intensity of 350 micromole. Now coming to the result, fresh weight, dry, fresh weight, shoot fresh weight, shoot dry weight, root length, fruit total flower are the major visual parameter which can be impacted by the drought stress. And then we observed that with the increase in days of drought stress, the shoot fresh weight, shoot dry weight, root length decreases with a increase in drought stress intensity. But in case of double transgenic line, these decrease were less in comparison to the wild or single transgenic. Now total fruit number and total fruit weight. The total fruit number and total fruit weight also decreases with increase in drought stress, but the decrease was less in comparison to wild or single transgenic in double transgenic crop. Relative water content is the key indicator of drought stress. Relative water content decreases with increase in drought stress, but in case of double transgenic,
conclude in 1 minute.
But in case of increasing drought stress, electrolyte leakage was increases with increase in drought tolerance, but drought stress, but it it was very low in case of double transgenic. See, in similar way, chlorophyll concentration index and photosynthetic efficiency was also observed. Lipid peroxidation, hydrogen peroxide was also low in double transgenic crop, but proline concentration was high in double transgenic crop. Now the gene expression analysis were also carried out. The expression of ATDRE1 gene at 40 days was observed maximum, while in 21 days it was decline. Similar way, BCZ12, the expression of BCZ12 gene increases with increase in time. It was maximum in 21 days in double transgenic crop. The expression of proline gene also monitor, and it increases with increase in drought stress. The maximum proline gene expression was observed in 21 days. Now, coming to the conclusion, double transgenic tomato plants were double plants overexpressing ATDREB and BCZ12 gene were successfully developed by gene pyramiding approach on the basis of relative expression of ATDREB1, BCZ and P5 gene expression. DZ1 and DZ5 line for drought stress tolerance revealed significant low peroxidation H2O2 content electrolyte leakage. In all drought stress conditions, DZ5 showed the significant high drought.
Okay, we can see. Please stop
okay.
there. Yeah. Okay. You don't have to read the slides. Thank you very much.
Now future.
I take to the next person. Sorry, Dr. Ramakrishna, to interrupt because we are encroaching in the other session. Please respect that.
Thank you, sir.
Mr. Santosh is the last speaker.
Yes.
We will be talking about isolation and molecular characterization of tomato eukaryotic initiation factor for development of 40 virus resistance. Can you
Yes, I
Ramakrishna, stop sharing your slide.
Is audible, sir?
Yeah. Please try to complete in 3 minutes. Take home message.
Can I start, sir?
Please don't go to introduction. Just go to your final results. You could be very effective if you only talk about the result.
Yes, sir. Yes. Yes, sir. Fine, sir. Okay, respected chairman and co-chairman. This is my title of presentation. The isolation and molecular characterization of tomato, that is eukaryotic translation initiation factor 4E genes are involved in the resistance of tomato virus. That is called the group of 40 virus PhD work. Then coming to the brief introduction, tomato as a major vegetable.
Please stop introduction. Go to your results. Otherwise, you lose.
And in India, it is producing about 20 million tons in the year of 2018 and 19. So this tomato is majorly affected by SIP. Yes, these are important diseases of viral disease.
Santosh,
Why we choose the virus? Here, for virus replication, they need a. Yeah, madam. Yeah.
Skip your introduction slides. Skip your introduction slides. Go to result directly.
Yeah. Yeah. Yeah, these are the objectives of my research work.
Santosh, you are not audible, nor this one is working. You are not audible, at least to me.
Hello. Yes.
Okay.
No, sir.
Yeah, please go ahead.
Hello.
Please
Hello.
go ahead.
Yes, sir. These are the objectives of my work. Yeah, for my work, we are taking. Yeah, madam.
Don't have to say anything. Go to your results. We have been telling. You are wasting time.
Just Yes. Yes, sir. No, sir. These are the results we are using for SGRNA construction. We are using a different software for our study. We are using a CC-Top. It is a bioinformatics tools for SGRNA identification and evaluation and characterization. These give an bioinformatics based on the different rate values like high and medium, low range. We have selected only the high rated SGRNA for our trait of interest. And these are the different SGRNA we have taken for our study for each gene, AF4E1 and ISO and AF4E2 for 3 genes for 3 different SGRNAs. These are the different sites. These are different positions of SGRNA. From 4E1 is 382 to 405 base pair, and whereas in 66 to 88 base pair. In case of 4E ISO2 is 293 to 31 base pairs. These are the target sites we have taken for SGRNA construction. This is a vector for construction of binary vector for agrobacterium mediated transformation. Then finally move to the different for isolation of three. This is a conformation of 3 genes with a different length. That is 693 and 666 and 603 base pairs. 4E1, 4E ISO and 4E2 and 4E ISO. This is a precise conformation of plasmid by using normal plasmid isolation protocol. These can be cloned into a plasma cloning vector, that is the PTZ vector, using any DH5 digestion with both the agrobacterium and our SGRNA vector with the Int3 restriction digestion. So this is a cloning site with 1 kb ladder, and this is the SGRNA or digested with the Int3. Then this sequence is cloned by using an GAT and ATC finding cloning sites. And this can be, these 3 genes are cloned and confirmed as we submitted for the NCBI website. And also we check for blast resistance, around 99.71% of homology with the already predicted sequence of EA4E1 genes. We checked for 3 genes also, 4E ISO and 4E2 also. And this is the FISR confirmation of mobilized agrobacterium EA4E gene with our desired vector in a PBI121 vector. This is the comparison of the chiliminal motile virus symptoms between the different indicator plants. That is the in the datura and Nicotiana tobacco glutinosa and as well as tomato orca vicas. This is the 15 days of days of post of inoculation, and this is the observation we see in the 30 days of post virus inoculation. And this is the material used for standardized procedure for agrobacterium material.
Please conclude in 1 minute. 1 minute more.
Yes, sir. These are the overall tissue culture regenerated protocol. We standardized and we maintain in the transgenic lines or maintained in the transgenic biseptic lighthouse. So in case of transformation, we see the efficiency of hypocotyle is more. So further studies we've taken for hypocotyle results. This is the overall summary. We taken around 8 mutants from each gene. From 4E1 is the 4E1 from 4 mutants, whereas in 4E2 is 2 mutants, and whereas 4E ISO is the 2 mutants. Is a different percentage of transgenic line mutated. This is the sequence confirmation. These transgenic lines were mutated with comparison with wild type. This is the PRAM site where we target of SGRNA 4E1 and 4E2 and 4E ISO. So sequence variation we can be seen. So where the phenotypic observation were compared with the wild type, we can observe this can be target site for mutation analysis. So then finally these sequences are submitted in NCBI website. We got plots of different. These are the different NCBI.
Okay, thank you. We could say that.
Clones we deposited in NCBI website. So finally,
thanks, Dr. Santosh. I have to stop you there because we are running completely behind schedule.
Yes. Yes, sir.
Thank you very much. I'm really sorry that we could not give full time to everyone.
Yes,
I take this opportunity to thank the organizers on my personal behalf and on behalf of my co-chair, Dr. Katagiri, and the reporters, Dr. Nishani and Dr. Joyappa. Thank you very much, and I hand over to the organizers for further.
Thank you, sir. Thank you, sir. We take pride in, we are honored, sir, you chaired this session. Such a knowledgeable and quality discussions we had in this brainstorming session. Thank you, sir, for excellent management, and you are encouraging us, and you keep on encouraging us, the young scientists. Thank you very much. I would also like to thank Katagiri sir. Sir, we are like privileged. Yesterday we had a keynote address of yours, and today you are here co-chairing the session. Thank you. Thank you very much. Sir, any suggestions on the session, sir?
Since we are running out of
concluding
time,
remarks, sir. Concluding remarks.
Well, we had a wonderful session, and transgenics and genome editing, the two frontier technologies, I think they are now ripe for application in horticulture. Particularly, as I said in the beginning, the genome editing is going to revolutionize our horticulture crop breeding, particularly in perennial crops. Although we have not seen many examples in the case of banana. And I can see great opportunities, for example, in citrus and various other crops, for example, seedlessness, maize sterility, and citrus greening in particular. The major challenges for which we had no solutions. I think these new technologies are opening up wonderful opportunities. And also, as speakers have shown, there are hurdles to cross in terms of commercialization. These have been brought out by both Dr. Prakash as well as various other speakers, including Dr. Paranjipe. I'm really grateful to everyone for a very illuminating talk and a session. We have overrun our session. This is one aspect I think we all also have to learn, that we should stick to the time. And I'm always worried that we are overstepping our time. That's why I thought I should not really take time to conclude this any further. Thank you very much. And
thank you, sir.
It was my privilege to be associated with this. Thank you for the honor. Thank you.
Thank you.
Thank you, sir. Thank you so much.
Thank you.
Sir, Katagiri, sir, any remarks?
Only one statement.
Yes,
It should be an eye-opening to the government. Government has to open eye for science. That's all. Thank you.
Thank you, sir. Thank you so much, sir. I thank both the chairman and co-chairman of this session, and I would also like to thank the rapporteurs, Dr. Sandhya Rani Nishani and Dr. Jayappa. Thank you all. So we'll move on to the next session, the second session of day 2, that is triad mapping and marker and genomic assisted breeding. Triad mapping and genomic assisted breeding. For this, the chairman of this session is KP Vishwanath sir, former Vice Chancellor, Mahatma Phule Krishi Vidyapith, Rahuri, and co-chairman of this session is Dr. D.L. Savitrama, Dean Agri, US, Bangalore. Because of unavoidable circumstances, Dr. KP Vishwanath sir is not here. I request Dr. D.L. Savitrama, Dean Agri, US, Bangalore, to take on the chairman and chair the session. Madam, I would like to have a brief introduction about Dr. D.L. Savitrama, Madam, presently the Dean Agri, US, Bangalore, as our professor of genetics and plant breeding with rich professional career, guiding so many students with many awards, and she has focused research on marker assisted selection for water use efficiency and leaf spot resistance in groundnut. And she has worked on water use efficiency in tomato and green gram, as well as transgenics for salinity tolerance. Madam has several awards to her credit, publications in peer-reviewed journals, and she has handled 35 projects so far at different levels, national as well as international levels. Madam, we welcome you, madam. Welcome you to the session.
Thank you.
Yeah, madam.
Thank
Please.
you, Lakshmi Devamma, for
Thank
the introduction. Now, this particular session is about trait mapping and marker assisted breeding. So this is supposed to be Dr. KP Vishwanatha is supposed to be chairing this session. Due to unavoidable circumstances, he is not here. Now I will be requesting Dr. Manjunath Gowda, who is a silkworm geneticist, would be co-chairing the session. I request Manjunath Gowda to join the session and co-chair the session. Now, I think he has joined the session. Now, let's start with the keynote address. This is by Dr. B.M. Prasanna. Dr. B.M. Prasanna is online. Sir, good afternoon, sir.
Hi, good afternoon, madam.
So you would be giving talk on its keynote address on genomic assisted breeding for maize improvement. Before you start, I would like to give a brief introduction about us. Dr. B.M. Prasanna leads CIMMYT's global maize program, which focuses primarily on maize improvement in sub-Saharan Africa, that is in Latin America and also in Asia. He provides technical oversight for an array of multi-institutional projects and the development and deployment of improved stress-resilient maize germplasm in the tropics, as well as novel tools and technologies for enhancing genetic gains and breeding efficiency. He also leads maize, a CGIAR alliance of more than 300 research and development institutions worldwide that seeks to mobilize global resources in maize research and development to achieve a greater strategic impact on maize-based farming systems in Africa, Asia, and Latin America. Under Dr. Prasanna's leadership, CIMMYT established a state-of-the-art maize double haploid facility in Kenya. He and his team have developed at a forefront in tackling the maize lethal necrosis epidemic in Eastern Africa, as well as the fall armyworm challenges across Africa. Of course, it's a very brief introduction, sir. Kindly take over and deliver your keynote address.
Thank you very much, Chair, for this very kind introduction, and to the organizers for giving me this opportunity to make a presentation in this conference. Okay, I hope you can see my slides.
Yes, sir.
Okay. Okay, the topic of my presentation will be genomics assisted breeding for maize improvement. I lead the global maize program of CIMMYT and the CGIAR research program on maize, focusing on maize improvement in sub-Saharan Africa, Latin America, and Asia. Although maize is grown in more than 180 million hectares worldwide, we need to recognize the fact that approximately 90 million hectares are there in the tropical regions of sub-Saharan Africa, Latin America, and Asia. Maize is a very, very important food crop for millions of people in Africa as well as in Latin American countries. It's a preferred staple food for approximately 900 people who survive on less than $2 per day, and provides roughly 15 to 56% of the total calorie intake in these three continents. Yet at the same time, tropical maize suffers from multiple stresses, not only the drought, but also poor soil fertility, devastating diseases, insect pests, and there are, of course, institutional challenges, especially in terms of providing access to high quality and affordable inputs. CIMMYT's global maize program therefore focuses on these three geographies, sub-Saharan Africa, especially Eastern and Southern Africa. All these yellow regions are those where maize is grown, but we focus our efforts in the tropical rain-fed environments of Eastern and Southern Africa, Mexico, and South America, especially the Latin American countries, and South and Southeast Asia, with a focus on higher yield coupled with yield stability, improving stress resilience, enhancing nutritional quality, and strengthening seed systems. And these are quite important for more than 80% of the rain-fed regions in these tropical countries, which are quite vulnerable to various abiotic and biotic stresses. The first most important thing to do any breeding work, be it genomics assisted or even conventional breeding, is to have a well-established phenotyping and germplasm testing network. And that's what this slide shows. For the range of traits that are relevant for smallholders, in these geographies. We have well-established hubs in Mexico, Colombia, Central America, Ethiopia, South Africa, Zimbabwe, Kenya, then of course in India, Bangladesh, Nepal, Pakistan, and so on. So these are CIMMYT managed phenotyping and breeding hubs that analyze our germplasm for an array of traits relevant for these markets. Maize genome, what do we know? A lot is known about maize genome, but yet a lot still to know. In terms of maize genomics, B73, of course, was the first inbred line to be completely sequenced almost 20 years ago. And now we have a series of tropical genomes being resequenced thanks to the efforts happening through CIMMYT and partners. What does this tell us? It tells that there are several structural variants replete in the maize genome, including deletions, insertions, inversions, translocations. These are all contributing to maize genomic diversity and play a very important role in phenotypic variation. In fact, remarkable is the phenotypic and genetic diversity between different inbred lines in maize, and those differences are almost as much as that between humans and chimpanzees. So no two breeding lines coming from different genetic backgrounds are similar because of these structural variations. But of course, with these advances in genomics knowledge and tools and resources, there are several applications now possible in breeding. We can dissect the genetic structure of the germplasm used in breeding programs, monitor very effectively the genetic purity and identity through DNA fingerprinting of key lines, provide insights into the allelic content of potential germplasm for the target traits of interest, select segregants with the desired marker trait associations, thereby minimizing significantly the costs of phenotyping in breeding programs. And finally, through the methodology of genomic selection and genomic prediction, we can also significantly improve breeding efficiency. These are not just hypotheses. These are all being practiced in mature breeding programs like those of CIMMYT. So behind all this are enabling resources, tools or technologies. What are those? First of all, we have a very strong knowledge of the genetic and molecular architecture of various traits of interest, established high quality phenotyping platforms and protocols, and developed an area of association mapping panels, plus biparental and multiparental mapping populations for validation of genomic regions of interest. We also have low density, medium density, and high density genotyping platforms available to provide genotyping at affordable costs. Coupled with that is the double haploid technology. And finally, most important, of course, we also need to have, any breeding program needs to have capacity to analyze genotypic and phenotypic data for taking selection decisions. So breeding programs need to focus on increasing genetic gains in the target environments of interest. And that happens through focus on this breeder's equation. And the breeder's equation, therefore, the key elements are selection intensity, selection accuracy, and genetic variation. These come under the numerator, whereas the cycle time, the time taken to complete a breeding cycle, is what we call cycle time. And that cycle time is the denominator. You can increase genetic gains by improving the elements in the numerator significantly, as well as by reducing the time taken to complete a breeding cycle through all these elements. So this, therefore, breeding programs, whether it is international or national breeding programs, need to continuously monitor or assess the genetic gains that you are making through your breeding approaches. In case of CIMMYT, the genetic gains across our breeding pipelines in Africa, Asia, and Latin America, these typically range from 1.5% to 2%, which is not at all a mean task compared to what has been done in the temperate maize environments, because improving the genetic gains in that fashion in stress-prone environments of the tropics is much, much more tougher compared to what we do in temperate environments. Yet at the same time, there are opportunities to accelerate genetic gains in maize breeding programs, again by focusing on various elements depicted in this breeder's equation. Irrespective of the genomics era, we need to recognize that phenotyping is the key for success of breeding strategies. Phenotyping throughput, accuracy, and efficiency are really very important. It is crop agnostic. Therefore, in case of CIMMYT, we focus on not just establishing a phenotyping network, we have developed harmonized phenotyping protocols for key traits of interest and widely disseminated to our partners so that our partners and we follow the same type of protocols. Establish the dedicated phenotyping facilities at key locations like this maize lethal necrosis screening facility at Naivasha, Kenya. We also go for digitized through hand notes. And we are also extensively working on high throughput tools for both proximal and remote sensing tools for reducing the time and cost of phenotyping of target traits. The first most important application of molecular assisted breeding or genomics assisted breeding is undoubtedly DNA fingerprinting of the key inbred lines. The importance of this cannot be underestimated. It has multiple applications, not just diversity analysis or estimating identity by descent, but also quality assessment and quality control in the breeding programs. Undertaking, once you have the genotypic data of the inbred lines, it also facilitates genome-wide association studies, identification of key genomic regions and haplotypes for target traits, imputations for genomic predictions, identifying new crossovers and selection of new crosses. And we can also, through modern DNA fingerprinting methods, monitor varietal adoption in our target environments. I won't go into the details, but these are, if you do this very well, you can have a clear differentiation of your germplasm and the substructure within the germplasm, including, for example, CIMMYT's lowland tropical, subtropical, and highland tropical lines are clearly differentiated. Quality assessment and quality control is really key, whether it is maize, cotton, rice, or any other crop. Periodic checking of genetic purity and identity of the parents of your commercial varieties, as well as the key lines that are being used in your breeding programs through an identified set of mapped SNP markers. That's what we do, and we published a protocol, a manual on QA/QC in maize breeding and seed production. These are all open access materials which you can easily access through net. So this is therefore a simplest at most important application of molecular markers. Why is this so important? Because outcrosses do happen. Mistakes do happen in the field for many reasons. There could be inadvertent selfing in a highly cross-pollinated crop like maize. Seed mixtures can happen. Packaging errors do happen. Harvest errors can happen. So therefore, if you look at the overall cost per return of QA/QC, it is enormous. So this is something that every breeding program, including public and private sector, need to rigorously implement. In terms of trait pipeline, we have a very robust marker discovery, validation, haplotype optimization in breeding parents. Then we go to haplotype validation in our breeding populations. Then we optimize the genotyping assay and finally deploy those markers or haplotypes in our breeding strategies. So typically, all these, if you look at these traits, some traits are under discovery phase, some traits are under validation phase, some traits are under haplotype optimization and validation phase. Some are already being used for genotyping assay development, and as many as 5 major traits we are deploying routinely in our breeding programs. It's no longer something like marker-assisted breeding and conventional breeding. For these traits, marker-assisted breeding is an intrinsic part of your breeding strategy. And these are used not only for, not just for marker-assisted backcrossing. I believe fundamentally these markers should be used more in marker-assisted forward breeding so that you have enrichment of your populations for those traits of interest. So if you look at it, provitamin A, maize streak virus, maize lethal necrosis, haploid induction rate, aflatoxin resistance, Tarsicum leaf blight resistance, these are all under deployment stage, where thousands of samples every year from our breeding pipelines are used for understanding the genetic status of our DH lines or inbred lines, and then going towards product development. So forward breeding is the key here, not just marker assisted backcrossing. Trait marker association, when do we use that? When there is measurable improvement of the trait by selection of that particular genomic region or a haplotype, effective across broader genetic backgrounds. And it has to ultimately have utility in terms of resources, spending your resources for discovery, validation, and deployment. If these criteria are fulfilled, then we should go for marker trait pipeline strategy. DH lines or double haploidy is fundamental now for advanced maize breeding programs for undertaking forward breeding for selected traits. We have state-of-the-art double haploid facilities at Quebeco in Kenya, as well as at Agua Fria in Mexico. And one such facility is being established. It's actually construction is happening at Kunigal in partnership with University of Agricultural Sciences, Bangalore. And it will be functional in 2021, and it will be first amongst the public sector maize double haploid facility that would cater to the demands of breeding programs across Asia. And so this is a very, very important step towards strengthening breeding programs in Asia. We also have developed superior second generation tropically adopted haploid inducers for the first time and distributed it worldwide. So as far as CIMMYT maize program is concerned, we have generated as many as 350,000 maize DH lines in the last 7 years from 2,124 populations at an average of 160 DH lines per population. These are really impressive figures if you look at the number of DH lines we are getting per population, and with good seed set in each of those lines. Again, in terms of molecular breeding for nutritional enrichment, you can look at this out of several countries which have deployed nutritionally enriched maize using CIMMYT maize germplasm. Some countries you see here, the ones which are highlighted in green, India, China, Kenya, Ethiopia, all these countries have already deployed molecular marker assisted products with enhanced nutritional enrichment. Approximately 1.4 million hectares of the tropical maize area are covered by CIMMYT-derived biofortified maize varieties in Africa, Latin America, and Asia. We focus not only on improved protein quality through QPM, but also high rates of provitamin A, high concentration of provitamin A, and high concentrations of zinc now. These are really important traits, especially zinc in the context of COVID-19 is an important mineral to focus on to strengthen the immune response. What are the other success stories? MLN in Eastern Africa, a major disease that broke out in 2011 and then spread to several countries across Africa just within a matter of 4 years, causing devastating effects on farmers' fields. This is a combination of two viruses, maize chlorotic mottle virus and sugarcane mosaic virus. And so it is, and having resistance to this combined infection is an extremely tough task. But that's where we succeeded, not only in developing improved lines and hybrids with MLN resistance, but also simultaneously analyzing the genetic architecture of MLN resistance in maize. Manjagowda, my molecular breeder at Nairobi, contributed a great deal to this important work. And then we detected a major QTL in a line called KS23, a genomic region on chromosome 6, making a very strong impact on improving MLN resistance across multiple genetic backgrounds. And using that major QTL, we transferred that genomic region to as many as 52 elite drought tolerant but MLN susceptible CIMMYT lines, which are parents of dozens of commercial hybrids in sub-Saharan Africa. This is a massive marker-assisted backcrossing effort, leading to, you can see, significant shift in the phenotype from highly susceptible, almost dead lines to highly resistant lines with the transfer of this genomic region. And genomic editing is now in progress in collaboration with Corteva. Fine mapping of this chromosome 6 QTL has happened already, and we have identified those target genomic regions. And very, very soon, we will have, this will be the first instance in tropical maize with gene-edited project for a major disease. And through that, we are also establishing a pipeline so that we can reduce the time required for developing MLN-resistant versions. Genomic selection or prediction in breeding is really important now, especially for improving complex traits, which are not controlled by one or two haplotypes, but there are multiple genomic regions from multiple pathways contributing to trait like drought tolerance or heat tolerance or waterlogging tolerance. In those cases, it is better to go through a random set of markers that cover the entire genome and have a good training population and have a tester population to estimate the genomic estimated breeding values or GEBVs and use them to make selections or prediction of performance. So genomic selection is now mainstreamed in CIMMYT's maize breeding programs in Africa, Asia, and Latin America. And we use this for prediction and selection of untested lines, untested hybrids, as well as for population improvement through rapid cycle recurrent selection. You can see here in this graph, stage 1 trials, where the number of lines genotyped are really 3,000 that are going into stage 1 test crosses. Of this, the number of lines phenotyped are only 853. So not even half of them have been phenotyped. And using this phenotypic data and the complete genotypic data, evaluation of those test crosses in 3 optimum and managed drought stress, we predicted what happens in terms of the performance under drought stress and under optimum. You can see an excellent correspondence here. That means the observed versus predicted the correlation is very, very high. That means you don't need to necessarily phenotype all the lines. You can test half and predict half, provided you have good genotypic data on all the parental lines and a sound statistical model to estimate genomic estimated breeding values and make those predictions. A series of publications have come through CIMMYT's program in the last 4 to 5 years. I won't go into the details, but this is one such publication on empirical comparison of tropical maize hybrids selected through genomic selection versus phenotypic selection alone. And you can see here, genomic selection can significantly improve breeding efficiency in tropical maize programs. The top 15% of hybrids advanced through genomic selection and phenotypic selection gave 21 to 23% higher grain yield under well-watered and 51 to 52% more grain yield under water stress conditions than the mean of the checks. But the most important thing here is genomic selection reduced the cost by 32% when you compare with only phenotypic selection with the similar selection gains. That means GS can reduce both the cost of the breeding program and improve the breeding efficiency in terms of genetic gains. This is an excellent proof of concept in tropical maize improvement programs. Not many breeding programs are there in the world that are mainstreamed genomic selection in breeding programs, not just publications, but this is mainstreaming. And we also did rapid cycle genomic selections in multi-parental tropical populations. So you typically go through genomic selection from cycle 0 to cycle 1 to cycle 2 to cycle 3 and estimate what kind of genetic gains you have made from cycle 0 to cycle 3. And you can see here, this paper published again by Zhuaxai Zhang, my molecular breeder at Mexico, shows that rapid cycle genomic selection can be a very effective breeding strategy for simultaneously conserving the genetic diversity and at the same time achieving high genetic gains in a very short period of time. Compared to what it takes 7 years, this strategy could be completed just in 4 years. That means you save 3 years of precious breeding time to achieve higher levels of genetic gains. My final word, it's important to have a mature breeding program, to have tools, breeding management tools, as well as biometric capacity and bioinformatic capacity to integrate phenotypic and genotypic data, to implement strategies like genomic selection. So we use tools such as BMS or Maize Field Book or Gobi Data Ladder, and DNA sample tracking systems. First of all, for trial preparation to field data collection, then going into genotypic data storage, linking the phenotypes with the genotypes, and ultimately moving into genomic prediction and using selection and visualization tools on what are selected and what are unselected, coming out of this whole strategy. So therefore, breeding programs in India, this is really, really key. In order to strengthen the breeding programs, we need to also have strong biometrics and bioinformatics capacity to very well couple pedigree, phenotypic, and genotypic data. Ultimately, all these need to be translated into farmers' fields, and that's what has been achieved. For example, this is adoption of stress-tolerant maize in Africa from a stage when we have hardly 500,000 hectares of drought-tolerant maize grown in Africa in 2010, when I just started at CIMIT. Today, in 9 years, 2019 data this is, we reached 111,700 tons of certified seed of stress-tolerant or drought-tolerant maize, covering as much as 5 million hectares in Africa, and adopted by 8.67 million households, and benefiting as many as 52 million people in Africa. So these are all achieved through very strong public-private partnerships. Our seed is disseminated by more than 100 seed companies in Africa. And these seed companies invest in scaling and adoption, in dissemination of these improved varieties. I sincerely thank the partners, the maize CRP partners in Africa, Asia, Latin America, my colleagues at CIMIT for their excellent work and commitment to the mission. The funding agencies, a number of them support our work, Bill and Melinda Gates Foundation. In fact, Bill Gates released a short video with a blog in July 2019 highlighting CIMIT Global Maize Program's work on drought tolerance and disease resistance. So it's a big shot in the arm for programs like ours, USAID, BMZ, Syngenta Foundation, and Maize CRP. Once again, sincere thanks to colleagues in organizing this conference, and many thanks particularly to Fakhruddin for inviting me to share my thoughts on this important topic. Thanks a lot, Chair, for giving me this chance. You are not audible, madam. You are muted.
Thank you, sir. So for giving such an informative talk, you started from markers and you gave till now whatever the work which has been done. And you also have given how important phenotyping is important, even though we are using markers. And you have given for how many thousands you have done for inbred lines, DNA fingerprinting for multiple traits. And also your work on nutritional traits like you have provitamin A, zinc, and all that is such a great work. And the identification of QTLs and how we can introgress them so that we can use them in different, incorporating different traits, especially on MLN resistance. And most important these days, what all the scientists are working are on genomic selection and prediction in breeding. This is one of the mainstreams here. And you also gave an insight into rapid cycle of genomic selection, sir. It's a great talk, wonderful talk, sir. Thank you very much.
Thank you,
on
madam.
behalf of the organizing team and also on my behalf. Thank you, sir.
Thanks a lot.
Thank you. Now we shall move on to the next talks. This is an invited talks. The first person I would be inviting is Dr. Derek Batchinger. I hope he is there on this in the thing. Is he there online?
It's Madam, yes, Madam.
Dr. Derek, please.
Yes.
Sir, I invite you. You are a vegetable breeder on pepper. You're working at Taiwan World Vegetable Center. He holds a PhD from New Mexico State University, where he studies specially on disease resistance and RF genes in capsicum. He has extensive experience in international agriculture and food security. He has got his major research area on development of pepper varieties in diverse genetic backgrounds, combining resistance to multiple diseases and stresses for diverse markets. And he is working on major diseases, especially viral, bacterial, and fungal diseases, tolerance to high temperature, flood, and drought. Means he is working both on biotic and abiotic stresses, also on quality and nutritional value of pepper, and is studying on diverse cytoplasmic male sterile and genic male sterile lines to enable hybrid cultivar development in underdeveloped countries. You are working on all the fields of breeding in pepper, and I invite you for this talk on necessity of, I'm sorry, I have not given the talk's name. It's a necessity of accurate phenotyping for the identification of loci conditioning traits of interest. Sir, please, floor is yours and you have 25 minutes. I request you to complete your talk in 25 minutes, sir, please.
Am I audible?
Yes.
Okay, thank you so much for the kind introduction. And also I would like to thank the organizers for inviting me to present this talk today. So yes, I work at the World Vegetable Center. I'm the pepper breeder here, working in both chili and sweet pepper as introduced. And I thought it would be interesting to discuss, I assume that a lot of the presenters in this section will talk a lot about molecular aspects of plant breeding, but I think it's important to consider phenotyping. And so that's why I picked this topic to discuss. So at the first slide here, this is the effects of climate change under a +3 degrees average increase. So you can see here in South and Southeast Asia, where we live and work for the most part. I'm sorry, I can't hear you. It's okay?
Yes, sir, you go ahead, sir.
Okay, sorry. Okay. So you can see here in South and Southeast Asia, where we all live and work, that there's expected to be a significant reduction in yield as a result of climate change. And of course, there are three main sources of agricultural productivity. So the first one is farming inputs, cultivation of more land, and genetic improvement. But of course, there are clear issues with farming inputs, cultivation of more land. So we have dwindling resources of fresh water. Also, petroleum is finite. And we have a big problem with pollution. So those really limit the availability or the sustainability of increasing farmer inputs, right? Because petroleum is used to make fertilizers and pesticides. And of course, a lot of existing farmland is lost due to urban expansion. So we cannot expect that more arable land will be under cultivation or able to be cultivated under climate change. So this really leaves genetic improvement as the most viable option for increasing agricultural productivity. So how do we increase yields or agricultural productivity? So plant genomics and phenomics represent very promising tools for accelerating plant breeding, resulting in increased yields. And I think, as you will hear today and probably have already heard yesterday and earlier this morning, many plant breeders have adopted genomics and various genetic breeding techniques. However, the use of phenomics is extremely limited in crop production and crop breeding systems. So you can see I took this picture from Wageningen University, and it kind of is a big group of images that represent all the different types of phenomic technologies that are available. And we'll talk about some of these later. So molecular markers, I think everyone is quite familiar with molecular markers and how they can be a powerful tool to improve selection accuracy and make breeding more efficient. Though especially if your trait is simply inherited, is very costly to phenotype, and has a low environmental effect, or the environment plays a low role in the variability. And of course, we have the requirements to make a marker, which is that the variability exists for the trait of interest and that you can accurately capture the variability, what you're looking for. And so in plant breedings, however, molecular markers often fail to meet our expectations. The selection accuracy is often not as high as we would like. And several reasons for this. One is that there's a large focus on biparental populations, meaning populations derived from two parents. But even when we use GWAS, genome-wide association studies, we often do not find really widely applicable molecular markers. And this is because we have to worry about two separate associations. The first is the trait of interest and the QTL, and then also the association between the QTL and the marker. So it's doubly difficult to get good markers. So how do we improve our markers? The first is to increase genome coverage. So this means increasing the read depth for our sequencing, things like this. However, there are two studies here that show that higher genotype density does not always improve marker accuracy. Of course, there's possibility to move away from biparental populations and focus more on GWAS. However, this is limited based on the presence of traits in diverse lines. So, for example, if you're looking for resistant to begomovirus and you want to use GWAS, it may be very difficult because sources of resistance might be quite limited. We can increase population size, which is highly correlated with accurate QTL detection, especially for small effect QTLs. However, this is extremely costly because every generation that you have to phenotype and increase seed of through single seed descent is costing money. And then the last one is to improve accuracy of phenotypic data collection. So this can be extremely complex, extremely expensive, but automated phenotyping offers a reasonable solution. So here's an example of the cost of sequencing, and you can see that it has drastically decreased over the past 30 years or 20 years or so. Yeah, and there are several reasons for this. One is because, in the interest of time, I'm not going to get into details, but basically the advent of reference genomes. So once you have a reference genome available, it's much easier for you to do your sequencing. And of course, we have to remember that genotyping is just A, T, G, and C in different orders. So it's not that complex as compared to phenotyping, which is heavily influenced by the environment and many other factors. So methods for collecting phenotypic data for many traits have not changed in more than 100 years. And this is a bottleneck in progress, especially for traits with complex inheritance patterns, which, to be honest, most traits of agricultural importance have complex inheritance patterns. So I show this picture here of measuring SPAD on maize. You can see the SPAD meter is quite small, but the maize leaf, and in fact the maize plant itself, is quite large. So it's difficult to say that you're getting an accurate reading of the true SPAD of that maize plant. And so I'm going to give an example and discuss quite in depth about biomass. So for many crops, we know that biomass is highly correlated with yield, and accumulation of biomass is central to agricultural productivity. But we also know that biomass is unquestionably a quantitative trait controlled by many genes. It also changes with plant development. So depending on the plant stage, whether the plant is vegetative or reproductive, and also changes over time. However, when we measure biomass, we typically do it in a destructive manner at a single time point towards the end of the season. So this really limits our ability to accurately measure biomass and to determine the change in biomass over time. So then we have the advent of automated phenotyping, which uses sensors to capture data. So the sensor I'm going to talk about is the plant eye. However, there are many to use. I am familiar with the plant eye. That's the one we use at the World Vegetable Center. So it captures 3D imaging. So that's more easy to understand. It's just pictures of plants, and then also multispectral imaging. And I can talk a little bit more in detail about what is multispectral imaging. So yeah, automated phenotyping allows us to capture data in a non-destructive manner and delivers precise and objective plant parameters in real time. Yeah, so non-invasive plant screening. In our situation in World Veg, our system runs across the field 3 times every day. So I can see every plant in the field 3 times a day. Across all environments, so whether it's rainy or hot or at night, it's also measuring very precise and objective. So you lose the role of human bias in this type of analysis. It's also very high throughput. It can reduce costs. So the setting up of the automated phenotyping system is quite expensive. However, over time, when you don't have to pay so many laborers to measure traits, the cost of measuring the phenotype gets a lot cheaper. It integrates in many systems, so in the field, in the greenhouse, in the growth chamber, yeah, indoor and outdoor. So here, this is a picture of a sugar beet. So this is the type of 3D imaging we can capture from the PhenoSpecs, or the automated phenotyping. So this is an example of how multispectral imaging works. It's what it does is it measures the reflectance wavelength of the light coming back from the plant. So it shines a light on the plant and then measures the light that is coming back. So you can see here a dead leaf, a stressed leaf, and a healthy leaf, and then the different quantities or percentages of wavelengths that are coming back. So here's images of those same sugar beets using near infrared imaging and also NDVI. So you can see how we can see certain leaves are more stressed, certain parts of the plant are healthier than others. So this is just an example of the types of things we can do. So now back to biomass. So this paper that I mentioned back in 2013, they use this breed vision automated phenotyping facility to measure biomass 3 times during the experiment at BM1, BM2, and BM3, you can see here. And then they did QTL analysis for each of the biomass measurements. And what we can see is clearly there's a huge effect of 5R no matter what stage you measure the biomass. However, at stage BM2 and BM3, there are many loci contributing to increased biomass, in addition to 5R. So if you were to do this analysis at BM1, likely you would only find a major QTL on 5R and 2 minor QTLs, one on 1R and one on 6R. However, doing this automated phenotyping system, we're able to more precisely and more accurately figure out which genomic loci are contributing to the trait. So in 2019, World Veg established a field-based automated phenotyping facility, which was funded by the Council of Agriculture in Taiwan. So this is a really beautiful image of our system. It's a flooded trial of okra or bindi. So, but I'm not going to talk about that. I'm going to talk about pepper. So we recently established a core collection of capsicum across several gene banks in Taiwan, Europe, as well as Israel. And the core collection consists of 400 accessions, sorry, which is quite a lot if we want to phenotype. So what we did is we set up an automated phenotyping experiment, and we did manually measured traits and automated measured traits. And we were interested to find, the trial was conducted during the heat stress time of the year. So we were looking for, first of all, variability for heat stress, and also be able to identify loci contributing to heat stress. So the manually measured traits were heat stress related traits, and then the automated traits varied. So biomass, NDVI, lots and lots of things. So this is just an image of the system running at night. So we have no workers in the field at night, but the system is still running. Here it is in a rainy day. Of course, no worker wants to be in the rain, but the system is still measuring on the pepper. So this is that NDVI that I've been talking about, that normalized difference vegetative index. And there's a little bit of a how to say, a legend here, where you can see that as NDVI increases, it gets closer to 1, those plants are healthier. So you can see this is the distribution of beautiful, normally distributed panel here of NDVI for the core collection. So this indicates to me that there's a lot of diversity, and it's also, there's a lot of diversity for heat stress, because NDVI is it's a measure of stress. So this is growth rate. So this is the daily rate of change in biomass over time. So of course, not so normally distributed, but also quite interesting. We have several plants that grow very, very quickly and grow a lot during the experimental period here, and then several that aren't growing much at all on a daily basis. So we might think that these over here are more tolerant to heat stress. So this is leaf area index, which is again very similarly related to biomass. You can see again quite normally distributed, with a section over here of outliers that may be tolerant to heat. But one example I'm going to talk about in quite detail is leaf angle. So we know that leaf angle is significantly influences plant performance. This has been shown over 50 years ago with maize, where there a 40% increase in yield was resulting from erect leaf maize. Also, changes in leaf angle occur because of circadian rhythms, so night and day. This is quite common in legume crops or legume plants. Also in response to stress, so low light levels causes a shortened leaf angle. Also high light levels, increased solar radiation, and flooding. They all result in reduced leaf angles. The plant is responding to these stresses by reducing their leaf angle. So using this automated phenotyping system, and since data were collected throughout the entire production system every day, 3 times a day, we were able to identify, leaf angle at different time points. So we isolated leaf angle under high light and low light days, without precipitation, and during the hottest days and during the coolest days, also at midnight and at noon, to get some idea of how leaf angle is changing in response to stress, and also just normally between night and day. So these are the results for the low light day and the highlight day on the left of the screen, and the difference between the leaf angle at noon and midnight. So low light is pink, highlight, sorry, I have it backwards. Low light is blue, highlight is pink, noon is pink, midnight is blue. So you can see there's a much greater difference between highlight and low light than there is under normal circadian rhythm. So this means that leaf angle could be affected by the environment. And so this better demonstrates that. So here the pink is the highlight versus the low light, and then the blue are there's normal circadian rhythm leaf angles. So the pink, this very large histogram here, this is the leaf angle on the hottest day of the season, and this blue is the coolest day. So we can see that there's a lot of variability for leaf angle in response to stress. So as far as we know, leaf angle has never really been studied in capsicum, and we think that it has a lot of potential to be used for GWAS and to identify stress tolerant lines. However, we haven't done this work yet. So all we've done so far is phenotyped. We have not conducted GWAS yet. So in conclusion, the use of molecular markers has not been as useful as a tool in plant breeding as we had hoped, although it is still useful. It's not the silver bullet that we had all hoped for. Several factors contribute to reduce the efficacy of molecular markers, as we laid out. And phenotyping is likely the most expensive, but also one of the most important factors in developing molecular markers. And automated phenotyping provides a less expensive and more accurate way to obtain plant phenomes. So at this point, I would like to acknowledge the team here in Taiwan who helped collect a lot of these data or that I showed today. And I would also like to acknowledge the funders, the donors to the World Vegetable Center. So the automated phenotyping project was of course funded by the Taiwan government in addition to EU Horizon 2020 and G2P Seoul, as well as the Government of Korea Rural Development Agency. And so with that, I would like to thank you all very much for your attention, I think probably during your lunch break. So I really appreciate how many people are here at this time. So at this point, I'd like to turn it over, Madam. Thank you.
Thank you. Thank you. Thank you very much, Dr. Derek, for a wonderful speech on phenotyping, especially because when we talk about marker-assisted selection and markers, many times we ignore very good phenotyping, whatever phenotyping, especially about automated phenotyping, what is and how accurate the results are going to be. It's such a wonderful talk what you have given, and the facility, the phenomic facility which we need to have for doing a good phenotyping is such an important thing. Thank you very much. On behalf of the organizers and myself, I thank you.
Thank you.
Thank you. Now let's move on to the next speaker. Before I move on to the next speaker, I have done a small mistake by not inviting the rapporteurs, Dr. Ratnakar M. Sheth and Dr. Raghavendra G. I'm sorry. I hope the rapporteurs are here. They're
Yes,
online.
madam. Yes,
Okay.
madam.
I'm sorry for inviting you late. Now let's move on to the second speaker. That is invited talks in this, Dr. K.V. Ravishankar. Dr. K.V. Ravishankar, who is also my friend, who has specialization in genomics and marker-assisted breeding. And he has many years of experience, over 22 years. And he has developed microsatellite markers for mango, banana, okra, pomegranate, Garcinia species, and sapota. He is involved in gene pyramiding and development of tomato hybrids, assessment of genetic diversity and characterization of Indian mango cultivars. He has many projects who is involved especially on functional genomics for fusarium wilt, drought, and also on plant types and many other traits. And he has many more projects to his credit. Now I invite Ravishankar to give his talk. The time is 25 minutes. Ravishankar, please.
Thank you. Yeah.
Not able
Thank
to
you
hear.
very much, madam, for nice introduction. One minute. Share the screen. Can you see my slides?
Yes, sir. We can see, sir. Put on slide.
Thank you. Thank you. Thank you so much for invitation. Next, another 20 minutes, I'm planning to talk about application of genomic tools in improvement of tomato. Here, basically, I'm looking at the tools which we can employ for improvement and also understanding the many drought tolerant and abiotic stress in tomato, biotic and abiotic stresses in tomato. Coming to the history of tomato, it is now many of you know that it is originated in some Central America and the Andean region, and it gets domesticated from the Andean region, and then it moved to the Central America, and then it further domestication occurred in the Europe and also in North America. So initially, the domestication and the selection was only for the fruit size, number of fruits per plant, and shelf life. So these or most of these characters are fixed in the present day tomato lines, but some genes like fruit quality traits and stress tolerance traits were lost during the domestication, because more emphasis was given to only for the fruit size and number of fruits and number of fruits per yield per plant. So gene involved in the defense response also lost during the domestication.
So coming to the history of the breeding in tomato, so our history of breeding in plant, you can see the domestication might have occurred more than 10,000 years back, but the first resistant cultivar came in 1940 in Pan American by Bourne and Tucker, which is resistant to fusarium wilt. Then commercial hybrid came in 1946. That is using single cross. And in 1960, the first mechanical harvest tomato cultivar is developed. So, and 2012, we have whole genome sequence of tomato, and you know that it was published in the Nature. So, but in post-genomic area, we have so many things, like we have marker-assisted selection, genome-wide association studies, and EQTLs, MQTL, genome-wide SNP, marker, now CRISPR also is a part of the breeding program. Coming to what are the different omic approaches one can apply in tomato, I think you might have heard this, many of these talks which they cover one of these things. Basically genomics, which deals with the DNA, the genes, QTL markers, and structural genomics. And transcriptome, you basically look studies RNA, mRNA, microRNA, non-coding RNA, non-coding RNA, which are involved in the gene expression. And coming to the proteomics, we have protein and the localization and their functional aspects that deals with the proteomics. Coming to the metabolomics, you have many metabolomics, like in especially when you consider the tomato fruit, they have like a lot of the sugars, acids, and flavor, volatiles. All these are involved in metabolomics. And the phenomics, actually, last speaker spoke about the phenomics in very detail. So where here we can identify natural variation and mutant lines which are there, which is also part of the genomics, which is required for marker-assisted breeding and other things. Okay. So coming to the next, how we can apply genomics to the breeding. So there are three aspects in hello, can you hear? Three aspects in the genomics. One is functional genomics, structural genomics, and comparative, like the comparative genomics. So in functional genomics, usually sequence-based approach, where we can see the different transcriptomics, or we can also look at the genome editing and also like overexpression, basically single gene based overexpression or mutation. Structural genomics, we do next generation sequencing and also identify single nucleotide polymorphism, SNPs, and look for the quantity trait lie where the trait is governed by multiple genes, and also marker assisted selection. And also we have comparative genomes where we can, the knowledge from other crops, like for example, rice, maize, or from the Arabidosis, we can employ that in the breeding. To taking the one example, genome-wide association, this is a study where more than 300 lines of the tomato were studied for the fruit site. So they could, and there are some species also they have studied. They could identify clearly there is three regions for where the selection has occurred for the fruit size. One is the PIM region, where the initial fruit sizes were around 2 to 20 gram. Next, it came to slightly bigger to 20 gram. That these are the region which contributes in South America. From the Central America, the South America, these are the genotypes which contributed for the fruit size. Later, due to the selection, you can have big, that is this particular genomic region which contributes for the, now the present size is 100 gram per fruit. So, and also there are some region which contributed for processing, that is where we have acidity and TSS. And there's another study which they have taken some 192 genotypes, and genome-wide association study has been fruit shape, pericarp thickness, fruit weight, fruit height, and fruit width. So there are 41 SNPs were associated with these seven fruit characters, and out of that 9 SNPs associated with more than 2 traits. That means there's a great amount of selection has gone for the fruit color, fruit shape, pericarp thickness, fruit weight, and fruit height and fruit width. Coming to the another very important character is this flavor. So we all like the highly sweet and flavored tomato. So but during the domestication, flavor has not been the priority. So only, as I told you, the yield was the top priority, and post-harvest shelf life was the bit. But the recent meta-analysis of 775 tomato accessions identified around 2 lakh SNPs in the 3 genome association panels, and they discovered 305 SNPs which are significantly associated with sugar, acids, amino acid, and flavor related volatiles. So these SNPs can be one of the markers which we can employ in breeding new or improved tomato genotypes. So, and also they identified the fruit citrate and malate contents. They are most impacted by that. They are neglected during the selection. Coming to the constraints in tomato production, as you see the pathogens and the insects, and also the abiotic stresses like heat and salinity. These are all the constraints which are reduce the tomato production. Coming to the first, I'll take for the biotic stress where we are all working. So here we did we developed pre-breeding lines using, it takes like 8 to 10 years to develop conventionally, to develop through the backrust breeding and to pyramid triple resistance. For example, we took the TLC resistance, early blight resistance, and then Phytophthora resistance. But so these markers, usually these characters are contributed by different wild relatives like MI gene, that in the root knot nematode gene comes from the Peruvianum, and a TY gene that is TOLC resistant gene. They come from Habrochytes, TY2 come from Habrochytes, TY1 and TY3 comes from Chilensis, and TY4 and 5, 6 comes from Peruvianum, and Phytophthora resistant genes which are coming from the Pimpinellum phylum. Using marker assisted selection, we could do this development of new hybrids within 2-3 years. We have Arca Rakshas which are resistant to early blight and also TLCB. Samrat also is resistant to early blight and TLCB. And we have a new hybrid which is resistant to tolerant to bacterial wilt, TLCB, and early blight and late blight. So this using marker assist selection, we could backrust breeding, we could easily develop a new hybrids within 2-3 years. It saves a lot of time and cost also. And coming to the identification of QTLs. So QTL is a, we know that there is a QTLs, we all know that what is the quantitative trait loci, which is governed by multiple traits which are governed by multiple genes. And there is a new technique called QTL sec. Here what we can do is in any segregating population, you bulk the lowest bulk and highest bulk, and also sequence this bulk and also sequence the parent. Here we can identify some major genes which are contributing to the traits. So what they did in recent study in tomato, they are looking for the heat tolerance. So they identified the contrasting genotype. 1698 is the susceptible, and Elite 2093 is a tolerant gene. They did like QTL-seq, and they identified, and they also did the gene expression. Here they did eQTL and also QTL, RNA-seq and also QTL-seq. They could identify the regions which can contributing to this temperature tolerance. They have, okay. So similarly, there are many studies which are contributing many genomic regions which are identified in the tomato, like in the 3 QTLs for the fold, stress, and 8 QTLs for, they are coming from the different wild relatives like Pimpinellum polium and Penelli. So these are different QTLs which are identified in the tomato. This can be used for the tomato improvement program. And also their genes also they have identified by fine mapping. And you can see that some of the transcription factor and some of the miRNA, like some of the genes like MIB and some kinase genes and the alleele genes, and these are all the genes which are giving the abiotic stress tolerance. This is a big list. I'm not going to do it because of the lack of time. And similarly, the disease resistance is also very important characters, and people have tried and identified the for example, late plate resistant gene Phytophthora, the PH2, PH2, PH3. These are coming from Pimpinellum polium and many different wild relatives. And we have pyramided in ARCA with all these genes. And early like genes coming from habrachites and Pimpinellum polium, these QTLs have been identified because it's a polygenic character. And here, like there are different physioramilt resistant genes. These markers are already being used by the breeders to pyramid these characters, and verticillium V genes and Buckrait at B. And most important is tomato leaf curl virus, TY2 TY3. They are allelic, and TY2, TY4, and TY6. These are all TY1, TY3 comes from the chilensis, and TY2 comes from the habrachites. And other TY5 and TY6, they are coming from the Peruvianum. These have been regularly used by the breeders for pyramiding to the improved varieties or improved hybrids. Coming to the CRISPR, I'm not going into the details because already people have spoken about it. But here, what the people are doing is like in vitro breeding. So you can change the genotype or phenotype of the plant inside the lab without doing the actual breeding. You know the gene, and you can identify the target genes, and then you can change the gene expression or gene itself. Okay, so these are the different steps involved in the pyramiding. CRISPR-Cas, that is sequence selection, construction, transformation, development of edited plants, and validation of mutants. These are different steps. And here, I would like to you to concentrate on two things, like you edit the gene or edit the epigenome, like promoter regions, to enhance the expression of the genes, so that you get either drought tolerant or disease resistant gene, or you can knock out the drought sensitive genes for a year, so that you will get the tolerant or the resistant genes. Okay, these are the different methods one can try in CRISPR-Cas method. And there are many people use like for abiotic stress tolerance, maize, tomato. They are using AB-dependent kinase signaling has been altered to get drought tolerance. And tomato and another SYN-PR1 also, another gene has been altered to give drought tolerance in tomato. And coming to the most important, the recent study which they have done, this is called de novo domestication of wild relatives. Solanum pimpinellifolium. They have taken one wild relative called Solanum pimpinellifolium. It has very small flower and less number of flowers and less number of branches and less number of fruit, and fruit weight is very small. So they have identified 6 loci like self-pruning, fruit shape, size, fruit weight, fruit number, that is locus, and nutritional quality. They'll also look for the lycopene beta cyclase to increase the beta carotene. These are 6 loci. The information comes from the different crops like Arabidopsis, rice, and other places. So what they have done, they altered these 6 loci in the tomato. So what has happened to them? So there is an increase in the number of flowers. There is a 25% increase in the number of flowers, and there is a increase in the number of locules. The wild has, you can see, only 2 locules. The locule number has increased to 8 to 9 in the transformed or edited tomato lines. And fruit weight also increased. Here the fruit weight is increased, and the lycopene content, which is very low in the wild, it has increased to 500 times in the genome edited crop plant. So that means you can do the breeding inside the lab. Now it is inside like or in vitro breeding. That is, you can do the breeding inside the lab nowadays if you know the genes and their phenotypes. So similar way, there are many attempts have been made in in tomato to increase the abiotic stress tolerance. For example, TME, downy mildew, powdery mildew, and fusarium wilt. And abiotic stresses like heat stress, chilling stress, and drought stress. These are going on. So another study is a metabolomics. Like as I told you, in the wild species, the fruit weight is very low, and then it increased. Now presently it is 100 gram. Over the years, we have lost many metabolites which confer for the flavor and also the sugar. So what they have done, they have taken 600 lines and they have done both transcriptome sequencing, and also they have done whole genome sequencing. They could identify some of the genes, for example, MIB12, which is the transcription factor, which can change the color of the fruit, and which is pink color, which can impart pink color. And also they identified some of the compounds like anti-nutritive compound which are lost during domestication. So general strategy now for the new trait of interest, so you need to have the first trait in mind, like root architecture, disease resistance, or other things. And then you need to look for those traits in the wild relatives, like genetic resources. And you also look for SNP. By using genomic tools, we can identify SNP, or we can do genotyping by sequencing and resequence genome, identify QTL mapping and genome-wide association. And using comparing the both trait and marker association, you can identify this gene or allele. So then you can manipulate those candidate genes using marker-assisted selection or genomic selection or genomic editing. So these are the 4 quadrant which we have here. So this is important for any crop improvement. So this is going on in tomato or rice or maize, because it's the most well-studied crops in this world. So coming to finally, the conclusion is tomato genome. So we have genetics and breeding QTLs, and structural genomics. We have markers and structural variations which you need to identify using GBS, and also functional genomics like transcriptome, proteome, and metabolomics. This helps in identifying the genes and which are impart those particular traits, and also chemicals which impart particular traits. And omics and system biology, it helps to study the network of genes and their interplay. And application you can have in the varietal improvement, you can study evolution and domestication of these crops, and also look at the functional aspects of the genes and gene regulatory networks. So these all aspects one can do using the omics platform. Thank you.
Thank you, Ravi Shankar. You're wonderful. Your talk was so informative. You have talked about tomatoes so much, starting from genomic approaches in breeding to the CRISPR-Cas, QTLs, metabolomics, everything you have covered. And tomato is one crop where many species are there, and you could use all these species into our plant breeding to increase our yield, disease resistance, insect, all biotic and abiotic stresses, and also into nutritional, flavor, color, so many other things. It's a highly informative talk. Thank you very much for giving such a wonderful talk. Thank you on behalf of the organizers and also on behalf of me. myself. Thank you. Now we'll move on to the third speaker, Dr. Somshekar Punnuri, who is from Fort Valley State University. He is a research professional who has done PhD in plant sciences, Oklahoma State University, and his research interests are physiological improvement through the genetics and genomics of cereal crops and biofuel crops, leaf spot disease resistance in palm millet using marker-assisted selection, development of insect and disease resistance varieties in crops with emphasis on the use of host plant resistance through recently evolved genomics assisted breeding and genomic selection. And he'll be giving a talk on using genomics and high throughput phenotyping to unlock the economic, agronomic, and nutritional potential of sorghum and palm millet. I welcome you, Dr. Somshekar. Please, you can start here.
Thank you so much. Sure. Thank you so much, ma'am. What I did actually is I already had a video, so I'll share that video because
It's not opening.
okay, just a minute. I thought you were able to see it.
No, I cannot see.
Okay, just a minute.
Yes, I can see the slides now, not the video.
Can you see now?
It's the slide actually. Yes, yes, you can start.
First and foremost, I'd like to thank
You can increase the volume.
Sure.
We are not able to hear. We can only see the slides.
Okay.
Maybe you can speak on that because the slides we can see.
Right. Let me see. Can you see now? Can you hear?
Okay, maybe you can just.
Are you able to hear it?
No, we cannot hear. But it has started, but we are not able to hear that video sound. Maybe you can show the slides and maybe you can speak.
Yes, probably. That's what I would do now, actually. But it will be like overlapping for me, so I thought.
take time for the next this one?
Sure, sure. I will take time actually.
You will take time means you will be the next speaker, or if you want to make it all right and get back?
Yes, I'll make it all right and get back to you.
Okay, I'll call you for the next speaker. I'll call you as next speaker then.
Okay.
Is it fine?
Yeah, sure.
Okay, thank you. I'll call you as next speaker. Now, I will call, sorry for the interruption. There is a small problem in video playing. Now I will call upon Dr. Chinnababu Muva. He is a head R&D, Enza Zaden, C, Malaysia, will be speaking on commercial application of technology for accelerated breeding with precision. And a brief, he has got experience in this company for 5 years as head R&D, Southeast Asia and senior breeder on pepper. He has worked in Syngenta for 10 years as an NSPP lead vegetables and diversified field crops and senior breeder on hot pepper, and also has worked on Zuari Seeds Limited. So now Dr. Chinnababu Muva can start. Please start your talk.
Thank you very much, madam. Thanks for the introduction. Can you hear my voice?
Yes,
can
yes,
you see the
I
slides?
can hear you. Yes.
Yeah. Thank you for the organizing committee for giving me the opportunity to present on the commercial application of technology for accelerated breeding with precision. My presentation goes in a different direction. I mean to say that the most of the support we are getting from the biotechnology team, and then how can we connect these biotechnology tools and methods in the plant breeding aspects. And now what will be the empirical plant breeding methods, and then now how we are progressing with the new methods of plant breeding, incorporating all these technologies together. And it is a bit complex. And then when you are working with multiple traits, and I will take you towards that one, how Enza Zaden is working towards development of vegetables. First of all, I would like to tell it's a value chain, how the demand comes from the value chain. It is from the consumer, and then it goes to the retailer, and then it goes to the trader or exporter, and then finally the demand goes to the grower in different kinds of interests, and also trade interests, and also quality interests, and also the transportation point of view. And finally, it goes to the commercial companies to deliver the products. And then as a company, there is a market pull to do the research. And then we push with technology into the market in the commercial breeding aspects. The concept is commercial breeders focus on very different set of traits together to come up with a plant idiotype which is suitable for the market. The basic is first, as you put the rubbish in, it comes out only rubbish out. That's what, when you listen to the Dr. Derek, what he said is it should be a nice phenotypes linked with the markers, then only you can give a nice things. It means to say that if you put it quality in, then the quality output will be coming. And then I will touch the basic breeding because I want to connect you with the acceleration and also with the accuracy. So there is some of the points which will touch related to the art, where the breeder will have all sorts of things like paints as a genes, and then he has a study, and then greenhouse to do all the things, and then molecular tools he have, then finally he will get a beautiful products. And then you have a lot of variability to use in vegetables, and then which we can exploit it by your creativity. And then as a science, we see by the support of biotechnology teams, we have tissue culture, and we have different techniques, and also molecular tools, and also the software tools. Everything we have to incorporate together in the breeding, actually. And then finally, breeding as a man-made evolution. Finally, it's a man-made evolution using all the creativity, and finally you will come up with the products which are driven the chain, the value chain. It is from customer to the breeder, I can say grower. Then what's the concept actually? Here the concept, as Prasanna stated, I am again taking this same thing. For any genetic gain is most important. For the genetic gain, heritability is most important, and then selection intensity is most important. When you have very high number of selection intensity, then you will work with very less number of populations. And then at the same time, length of the cycles. The earlier days, it's 1 or 2 cycles per year, but now it is not sufficient to have more genetic gain. So in this presentation, I try to more put my emphasis on heritability of the traits, and also how can we increase the selection intensity, and also the life cycle management. So the evolution of technology, if you see, the first is beginning with empirical plant breeding, and then no use of markers once upon a time, and then now it evolution towards the simple traits, and then use of 1 marker or 2 markers related to traits, and then now you have many number of random markers, and also the foreground selection, background selection, and also the phenotyping different models. And now the present way of doing the breeding is completely predictive phenotyping or predictive genotyping. Before you sow the seeds in the soil, you have to predict what plant is coming, what traits are coming, and your expected plant idiotype is coming or not. That is all in your hand with whole data availability and also the genomic tools actually. That's a predictive approach. Then comes to the accelerated breeding. How can we accelerate? Is it a shortening of a generation time? I mean, is it 3 months per generation or 6 months per generation? Is it a shortening of time, or is it reduction in number of generations? I mean, I need 5 generations or I need 6 generations. What is most important? For me, it is both is important. Then only we can increase the genetic gain. How can we do that one actually? So we have different kind of breeding populations when you cross between parent A and parent B, and we have so many methods like pedigree methods or any other things are there, or you can go through the double haploids, or we can go the backcross populations. But this is a classical empirical breeding. Can we do something different? The technology is available. Why we every time we want to go parent A and parent B, can we go from back? I mean to say, take a F1 and then go back to identify your parents, and then modify something and incorporate some more resistances and come up with the same hybrid with edited traits. So this is the new way of breeding. Breeding methods is coming up. It's called reverse breeding. Methods are very easy with the use of biotechnology tools. So now, how can we shorten the life cycles? Earlier we used to do 1 life cycle a year. Can you make it to 2 generations per year? Yes, it is possible because of we have all greenhouses and also controlled conditions and all these things, and also different environmental models we have. Or can we still increase 3.5 cycles per year? So when you reduce the cycles, number of cycles, and also reducing each generation, you can definitely achieve the acceleration in the breeding cycles. You can also manage the temperatures of different environmental conditions to reduce the vegetative phase and to get the more reproductive phase immediately, so that you can forward the generations F2 to F5 very fast. So for something, the people like in tropical situations, then we have, we have good temperatures and the stress, and then we can apply hormones and all these things, and to achieve all this cycle reduction. I mean, a single cycle, we can reduce it actually. If you can see here, this is one of my experiment earlier, that is 45 days old crop. It is a pepper crop. It's already at the flowering stage and fruiting is happening. I need only 1 fruit.
one fruit contains 100 seeds. So my generation can go ahead very fast. So 3.5 cycles of hot pepper, 4.5 cycles of tomato, these kind of cycles in a year, we can come up with a finished lines actually. So use SSD, but it is a modified method of SSD as a breeding method, a strong method, but you have to incorporate molecular markers as a pre-selection or post-selection so that you can reduce the number of populations still, and also your accuracy will increase. And something like this, the same as double haploids. In vegetables, we are more using the double haploids like in brinjal and also in hot pepper, and cauliflower, cabbage, cucumber. Almost all the crops we can use double haploids. Here you can see that you can select the parental lines in the segregating populations using marker-assisted selection. The desired plants can be subjected to the double haploidy, and then within one generation you can get a fixed lines. So this is the way we can accelerate our breeding programs actually. And then I will go to the precision in selection. So I will just touch the basics actually in the precision, how we can do it. It is a dominant simple trait of backcross programs. You can see that it's a simple one, but when it comes to the recessive, it is more robust, but the molecular marker assisted selection can be useful more in reducing the life cycles actually. If you morning onwards, people when they are talking the advanced technologies, what they are using, the mutation breedings and all, finally they are coming up with recessive traits, not the dominant traits. Also, there are a lot of QTLs are coming up, and you cannot do a normal dominant backcrossing program directly in a short cycles actually without the support of biotechnology tools to speed up your programs. So earlier, it is a marker-assisted selection has changed completely the scenario of plant breeding programs. Earlier, you select the gene by way of phenotyping and then by observations. Now, you link up with the marker and you do the indirect selection, and you can do a pre-selection. I mean, before sowing the seed itself, you will be selecting. Even some of the large seeded vegetables, I can say in cucurbits, you can cut the seed and then subject that seed for the marker assisted selection. Only selected seeds, not by doing the leaf sampling actually. So the technology is mostly accelerating the generation and also with accuracy. And then you can see here a simple example that we have tomatoes, both yellow type and also we have red type, and even another 2-3 types are available. White type like cherry types are there, and pink tomatoes are there. They are all differentiating at only one locus, and then you can develop a single product which is similar but only differentiating for the colors using the marker assisted selection and also the background selections. When you select the background selection, then you will have a same kind of hybrids with the different colors. What is SNP? This is a single largest, I can say, the tool today being used by all the private companies. I can say, then you have a non-red gene at one single SNP gives the color actually, and also the red types are there. When you make the combinations, then you will get the hybrid. So this is a very simple way of doing the breeding and developing the parental lines by using simple SNPs. But other than this SNP, the beauty is the rest of the genome is similar. Both the parents are same, except one locus. And then you can see different kind of products. But suppose you have a best tomato variety which is suitable for a heated greenhouse segment. Then you want to bring it to the open field situations and also with different level of resistances because from greenhouses to open field always resistances are differing. How can you make it? It is all possible by marker assisted backcross breedings. It means that you select your resistant genes by way of forward, front selections, and also the background selections with the genomic selections you can use by way of developing different hybrids actually, but all looks the same. So the technology, how it is came accuracy is GMAS. Earlier, we used to sample many plates, and also you do a lot of run a lot of gels and then do the things. Now the technology has changed. You have a one single sequencing, and then you can almost 96 by 96 plates we have, and then we can sequence at a time. We can run 9,216 markers. And then we can do a foreground and background selections very easily. So the revolution is coming from because of GMAS. And then backcross versus MABC. Just to touch the base, it is 99% after backcross 4. But now with backcross 3, 100% we can get the by using marker-assisted background selections actually. So here, the why I am stressing more here, the basics, this is again, this is basics. This is not a breeding program of current breeding programs. This is again, I can say 5 years back, we were doing all these things. But now, again, going forward, the breeding methodology has completely changed in private companies. You can see here, the recurrent parent versus donor parent. How similar they are before we transfer the resistance or any trait. That makes the decision how fast we can reach to the final stage. Here at F2 BC1 stage itself, we can get a new material by using a background selections for the steep trait on the sweet peppers. Actually, this is one of the example I can say. When it comes to the integration, then you had seen the acceleration of breeding by way of SSDs or double haploids and all these things. And then you have to incorporate or integrate your biotechnology tools to have more accuracy so that you can do your breeding program so fast. While you are going ahead to F4 or F5, it means that you should already have a fixed parental lines, and also you have already known that your hybrid is going to perform better. How can you predict that one? This is the thing. The questions before you start the breeding program, any person will think like this. Parent 1 and parent 2, when you cross, you will get F1 and next is F2. We study like this. F1 is heterozygous and homogeneous. I have my own question mark. Is it really heterozygous? And then F2s, again, heterozygous and heterogeneous. Again, I have a question mark. Is it really heterozygous completely? So I mean to say that the questions coming to me is F1 always 100% heterozygous? For me, no. Is segregating F2 population 100% heterozygous? Again, no, because there is a level of similarity between the parental lines which always go forward from F2 to F2 because of this. The level of heterozygosity is less actually. So you need not to work for 100% to make it pure. You have a gap of some heterozygosity to work in your breeding program. And then use that similarity as a tool, as a background selection to go your breeding cycles very fast. Then what is the percentage of homozygosity in F1? Then it depends on the parents' similarity that we should know. Then should we have a balance between homozygosity and heterozygosity for the success of the F1? This is a biggest question mark at the moment. People will think that always, if I have more heterozygosity, I have more heterosis, and my hybrid will be superior. But that is not the right answer, because you should always maintain percentage of homozygosity and heterozygosity as a balance, and then only your hybrid is going to be successful. And then why only some varieties are successful and adaptable? Yeah, a lot of companies are coming up with a lot of hybrids, but not all hybrids are successful in the market. But at the same time, universities and all institutes are coming up with a lot of varieties. But they are all not successful. Only some are successful. Even in your trialing, you have a lot of hybrids, but finally you come up with only 2 or 3 hybrids. What is happening to the unselected hybrids? Yeah, don't throw it. There is a lot of knowledge you will get from the unselected hybrids. Why they are not getting selected? So that question has to be answered. Maybe in the future slides you can see that one. Can we define SCA and GCA, specific combining ability and general combining ability? It means that yes, we need to correlate between the hybrids, what not selected hybrid, what is selected hybrids, and also different universities adaptability, and then look back into the genome. And then there is always a unique sequences which are giving specific combining. Also, these unique sequences are most useful for adaptability. I can touch this one later. And then how much? I mean similarity. Then these are the questions will be coming as a breeder when you start your program. And then what determines your program and how to use this in recurrent selections. This is most important when you use all these biotechnology selections. The recurrent selections are most important. The genetic maps from different organizations you will get. You take all the materials of IIHR, you take all the materials of IABR, you take all the materials of US Bangalore, you take all the materials of LAM and then map them. There will be some sort of similarity, there will be some sort of dissimilarity will be there. So what it gives you the success, the dissimilarities are their adoption levels. The similarities are the basics of the material. So you differentiate the things and then you will get unique patterns for the success of each organization. Here I wanted to tell, if every breeder thinks in the same direction, there will not be this much variability. So in the world, each breeder is having different kind of creativity, different kind of art, and finally they are coming up with a different kind of varieties. And then when you create this variability and this automatically comes in your dendrograms, and where you have to see the successful breeders, why they have selected a unique sequences unknowingly in the past, and then you capture that unique sequences by a way of comparing the genome, genetic maps between varieties to varieties. The GMAS data can be more useful. Then you can identify really the unique sequences. You can link up with the adaptability, or you can link up with the heat setting, or you can link up with the special quality traits, or you can link up with any other traits which are more useful in the value chain. That's what they are successful in the market. So these maps will be very much useful for us at the moment in Enza-Zidane, that we have all the data of each and who are having the strongness and where they are strong and all these things. Now, what I will do is these unique regions will be connected with the different environmental models. I mean to say South Asia, Southeast Asia, or tropical humid and different temperature environments, and these unique sequences where they are expressing on all these things. So if you know these unique sequences, yes, we know, and then we can do a recurrent selection, F1, F2, F3, F4. Whenever you are going continuously, you keep selecting that lines by way of selecting only unique regions by background selection. They are not at all related to any traits. They are just a genome selection, genomic selection. And then finally, this man-made evolution is going towards unique sequences, and then it will be more useful in varietal success. That's what, if you take out the different companies, IHR, LAM, IAVR, all these materials, when you subject it to genomic mapping, and then you will have really unique sequences you will be getting actually. Can we identify these regions? Yeah, yes, because we had seen right now, and all the biotechnology team till now, what they are talking is the same thing. So it's only a programming that which can link up all these things. And so pre-selection by recurrent selections of the material is possible, and then only we can use these recurrent selections in our plant breeding program. This indirectly gives you accuracy while you are doing the developing the populations. So suppose if you have a F2 populations, and within that F2 populations you have more homozygous like a parent 2 genome. In the downside you have more homozygous P1 genome like a parent 1 genome, and you have very similar genotypes will have, and some you have more heterozygous genome, quite similar genotypes are.
Not able to hear? Not able to hear to you? Dr. Chinnababu? Cannot hear? Hello? Hello? Lakshmi Devamma, can you just call up and find out? His internet connection maybe might have disconnected.
Yes, madam, we are calling.
Yeah.
I'm sorry, I got disconnected, and then I'm connecting back with my presentation. Sorry for the inconvenience. I'm finishing my presentation shortly. I hope you are able to see my screen now.
Yes, yes, we can see.
I will go to the presentation mode. So this is the biparental population where you have small fruit size to the big fruit size, and then the beauty here is variation optimizer. Either you can select big fruit or small fruit, also different kinds of fruits. With the way you can do the selection here, you can see when you apply the variation optimizer, you have everywhere the possibility of selecting different fruit sizes, and then you use the similarity of the background or dissimilarity of the background, and then subject it to them into SSD or a double haploid, and then you can reduce the generations. It's the best example what you have. It means that you can have any flavor when you have the two different parents actually. Then the final thing is purity markers. What we do is we start selecting the parental lines for their purity from F4 onwards, so that while the line is developing with the different traits and also with idiotypes, background selections, also we apply the purity markers at F4 stage itself, and within 1 or 2 generations, maximum by F5, F6. By prediction, we know what hybrid you are getting. By prediction, what traits you are getting. By prediction, what at what level you are there in the parental purity, and then come up with your hybrid which is suitable in different environmental models, because you already selected your parental lines suitable for different environmental models. And then the cost. Now people will think about the cost. Particularly the private companies always think about the cost. If you see here, I had shown only till 2015. It is to run a complete genome. It is less than $1,000 in 2015. Way back in 2001, it is more than $100 million. And then still, I can say in 2019 and 20, the value has come down very far less, and then it is now very much economical in utilizing this technology actually. So finally, what I wanted to say is from molecule to the successful variety, it is impossible that a breeder alone cannot work, a phytopathologist cannot work, a biotechnology person cannot work. So we together has to make up the team and then have a good communication. What we have to do together has to make the planning and then project, and then you can have a nice tomato in your hand. Thank you very much. I'm open for any questions if you have.
Thank you, Dr. Chinnababu. What an excellent presentation. You have started from the basics. So only when we have good basics, we'll be able to be a good breeder. And you also told in your last slide, last sentence, that unless and until biotechnologies and breeders go hand in hand, they can be one person. We need to understand both of them, both the subjects thoroughly, so that we can accelerate our plant breeding program. And of course, cost is one thing which is very important, but still when we have to go for hasten the technology by using marker assisted backcrossing and using markers and other things, it's a wonderful talk, and we need to understand the most basic things, like starting from the genotype, crossing, and to forward them till the final selection of variety and hybrid comes. One thing what I liked is why so many hybrids and varieties have been released. Many of them do not stand in the field. That is what is so important. It's very nice, excellent presentation. Thank you.
Thank you very much, ma'am.
Thank you. Now I'll call upon Dr. Somshekhar.
Yes, ma'am. For all the disturbance, actually, I'll try one more time playing the video. If it doesn't work, I'll just use my slides.
Okay, please. He'll be speaking on using genomics and high throughput phenotyping to unlock the economic, agronomic, and nutritional potential of sorghum and pearl millet. Please.
Okay. Just let me know within next few seconds if it doesn't play. I will just use my regular slide.
Okay.
Are you able to see?
No, it's not playing.
No, not playing.
Yes.
Okay, all right. I'll just use my regular slides here.
Okay.
Please. All right. Sorry for all the disturbances. Today, I'm giving a talk more on sorghum and pearl millet using genomic assisted breeding, what I'm doing here at Fort Valley. My name is Som Punuri, and I'm working here at Fort Valley, Georgia. So today, outline of my talk is more on sorghum and pearl millet, how they are important in the context of climate resilience, and how we can use genomic assisted breeding for these two crops, for improvement of these two crops. I'll quickly go over pearl millet GBS project. I will just give a small talk on that. Then I'll give more talk on my sorghum projects that are currently funded. If time permits, I'll give more on wax mutant projects and soil microbiome projects that I have. And I would like to conclude my talk today by giving an importance of how these two crops are getting inundated with all the genomic resources and how we can improve these two crops. So you all know how global warming has been affecting us. It's very drastic effect that we are seeing over the last 50 years, that there's a huge change in temperature with around 0.5 to 1 to 1.5 degrees Celsius change across the globe, as well as here in the United States. We see a lot of frequent drought and less water availability in states like California and surrounding states. So definitely it becomes a collective and global responsibility for all of us to address some of the food insecurity issues, as well as less food production in the future. And also with the increased temperature, we are going to see a lot of increased pest outbreak in the future. So definitely we need to grow crops that can withstand all these pests as well as changes in the climate in the future. So that's why these two crops have been very important for me. Pearl millet and sorghum, as you all know, they are very climate resilient crops, and they are known for their drought hardiness as well as their ability to survive on poor soil fertility. They're also a good source of nutrition, as well as recently there is a buzzword here in the urban market on more gluten-free products. So these two crops are very well known, and they're getting more attention for gluten-free food crop products. So speaking on pearl millet related projects, what I want to say is that it was in 2012, I wanted to develop a GBS marker, and we wanted to develop a linkage map for this population. It had around 184 recombinant inbred lines from these two parents, and we did develop a GBS marker in collaboration with Cornell people. So we used APEK1 enzyme at the time, and we developed a lot of GBS markers. The way we developed the GBS marker was we got all the Illumina sequencing reads, and we called all the SNPs through the Tassel GBS pipeline. So this resulted in around 500,000 raw SNPs. So we used these raw SNPs. We called these raw SNPs by aligning our reads against around 20,000 scaffolds of Pearl millet genome sequencing consortium. At that time, still the pearl millet genome was not available, so we could only use few scaffolds provided by them, and we called our SNPs. So later on, we filtered for high quality SNPs from these. The way we did was, if you want to read more about these filters and parameters, you can read our paper. But I wanted to go quick here. The filters were more on removing all the heterozygosity, as well as removing all those paralog misalignments due to sequencing errors. Also with all the minor allele frequency ratios between 0.25 to 0.75. And then we also had identified potential outcrosses with the minor allele frequencies. So we really got very high quality SNPs. Then we clustered all these SNPs into different linkage groups based on hierarchical clustering in our software using Perl scripting. Actually, one of my bioinformatician helped me doing all these things. So if you see here, we did group or cluster these based on the linkage association mapping here, based on their linkage disequilibrium. When we merged all these, we were expecting 7 linkage groups, because pearl millet has 7 linkage groups. So if you see in this picture, we could clearly see 6 different groups except one group. So the one group had very least number of markers, and I will explain why we had that least number of markers. So finally, we ordered, cleaned up certain markers. Some of the markers, unwanted markers or redundant markers, were removed, which had, which were due to sequencing errors. So finally, we had a very good linkage map. This was finally compared with the consensus map. So if you want to know more about this, you can go to our plant genome paper. If you see this, we mapped around 16,650 SNPs in this map. Probably this was high density genetic map at that time. Around 1,191 are main core markers here, and we had around 300,000 sequence tags. So these 300,000 sequence tags are the ones which can be used by researchers in calling their SNPs. So I was telling, we had a least number of markers on this chromosome 4. This was because the recombinant inbred line population we used had 2 parents, and those 2 parents had a common ancestor, most recent common ancestor. So therefore, we did not find more recombination in the on that chromosome, and we could not find more polymorphic markers there. So then we try to compare our map, how good it is with the consensus map. We wanted to see those collinear markers. So it was probably agreeing in most of the part. So probably it covered almost same as the consensus map. but probably all these mapping experiments will tell that we really cannot get accurate same measurements of these markers. So that's very common here. So we could not agree for some of the parts. So what we did was, after we used all these markers, we wanted to plan them in the field, all these recombinant inbred lines, and wanted to do a QTL mapping studies. So we did QTL mapping by collecting a lot of phenotypic traits. So like panicle length and panicle diameter. So we got really a very good number of QTLs when we used in our QTL. So to highlight just one or two traits here, panicle length and panicle diameter, they were on chromosome 3. They had a pretty good significant QTLs with a good large score. So the outcomes from Pearl Millet project for genomic assisted breeding is that these results were used by Pearl Millet Genome Sequencing Consortium for assembling their genome assembly. So that was one of the best used from our linkage map. Then it also helps other researchers in calling their SNPs using the sequence tags. So then it also helps in comparative mapping and synteny mapping. Finally, hope is that we are going to use all these SNPs for QTL identification. And from there on, these markers will be used in marker assisted selection as well as genomic selection. So just I want to highlight that at that time, I did not have a lot of next generation phenotyping methodologies at that time. So, but recently in my sorghum process, I could use those technologies. So to wind up about Pearl Millet project, there is still a lot of hope to improve for drought resistant and several other traits like insect and disease resistant traits using all the genomic resources that it has generated. Now Pearl Millet genome has a complete genome sequence. There are a lot of other genomic resources available. Definitely these will accelerate genomic assisted breeding for Pearl Millet. Most of the nutritional studies can be done. You can improve the quality of forage crops. Just to highlight that Pearl Millet and sorghum in the US, they are mainly used as a forage crop, even though we use some of the grain type for feeding birds and animals, but not for human consumption. As a breeder, I would definitely focus more on developing breeding population using all these genomic resources, characterizing certain traits in the field, as well as developing certain mutant lines. So I'll switch gears here. I will take you to my sorghum related projects, which are currently funded by USDA and NIFA. So there was a severe pest called sugarcane aphid in the year 2013. So this became all of a sudden an invasive and most destructive pest on sorghum. So if you see here, this sugarcane aphid is very distinct from other aphids that are seen on sorghum and other cereal crops. People started giving more attention to this pest because it basically does suck the sap from phloem and xylem and interrupts a lot of water and nutrients in the plants. So it really causes a lot of loss, heavy loss. If there is a huge sugarcane aphid, you see that there is no head at all. The panicle doesn't emerge from the sorghum and you don't see those things. And they are seen almost in every stage. Right from soft dough stage, right from flowering, then soft dough stage until maturity. We also see a lot of honeydew and sooty mold comes because of the honeydew, which hinders photosynthesis. It also clogs a lot of harvesting machines. So it's a huge problem. So we are using 1 to 9 scale about this 1 to 9 scale by Sharma et al. To assess the plant damage by these aphids, how much damage they cause. We also quantify number of aphids using 0 to F scale, which is basically like just quantifying how many aphids are there. So if you look at this scale, it's very daunting, very tedious to do in the field. And these aphids are present on the lower surface of the leaf. So you have to really flip the leaf and see how many aphids are there. So it was very daunting, and that's when I started thinking if we can develop some high throughput phenotyping method, we can definitely enhance our experiments. So At the same time, I was fortunate enough to attend a workshop at Arizona. This workshop was on high throughput phenotyping, and this project is called TerraRef by Todd Mockler. They have a huge gantry in Arizona, and they're trying to evaluate a lot of biofuel accession lines here using a big gantry, which is all automated. So this is when I started thinking about using some high throughput phenotyping method for my proposal. At the same time, there were some robots also developed, by Steve Long from University of Illinois. So I approached this guy from Iowa State University. His name is Josh Peschel. So he already had a robot, but this robot did not suit our needs. This robot was according to their biofuel or other related traits. So I said, we really need a robot which can take a lot of pictures, capture images from the bottom of the leaf. So that's when we tried to put this proposal, and we were lucky in getting funded from USDA and NIFA. So we got it funded, and we started evaluating our lines in the year 2019. So I'll quickly go material and methods here. So we used around 286 sorghum association panel lines in RCBD design in Tifton, Georgia. So we evaluated for 2 seasons, that is 2019 and 2020, both manually and using robot and drone. So using next generation sequencing, we developed GBS markers. Okay, I forgot to tell that out of these 286 lines, we added a few more 10 extra lines. We designed those and we included them to this sorghum association panel. The sorghum association panel itself is very huge, around 377, which I will tell later. So we took around 277 of that and added few more lines, and we wanted to do this genome-wide association studies. So this is Josh. He came all the way from Iowa to show us how to drive the robot and phenotype and collect a lot of data. That's when we started using this robot in the field. So, but we had a lot of problems in the first year. The robot had a lot of camera related problems and sensor related problems, even battery related problems. So we started to address all those problems in the year 2020, and we approached another new company called EarthSense. So EarthSense, actually, it's by Chinmay Soman and Girish Chaudhary here in US, in Illinois. So this robot has at least 4 different cameras all around, and these cameras can do a lot of things. They can automate and take a lot of pictures, and you can use this data to automatically analyze later. So that's how we use this robot in year 2020. We have collected huge set of data here. So this I'm trying to show here, we did both manual phenotyping for the sugarcane aphid as well as robot data for all the entire sorghum association panel here. So this is a glimpse of pictures from the robot, which shows how a robot goes and travels through this field and takes the pictures, and it actually takes a lot of videos. So I just edited that video and took these pictures. So just to highlight on genome-wide association, I know a lot of speakers have already spoken about genome-wide association studies. Basically, they take into consideration a lot of historical recombination events that lead to this genome-wide association mapping, means that lead to rapid decay of linkage disequilibrium. So that leads to finding genome-wide association markers. So we find a lot of significant SNPs with the highest p-values. So sorghum community is blessed to have a lot of resources such as sorghum association panel, which is available for public to use. Like Casa et al. In 2008, they developed, they made around 377 photoperiod insensitive breeding lines, which were racially, geographically, and phenotypically diverse. So Morris et al. Actually developed a lot of GBS data for all these 377 lines. And recently he has even developed around he has developed GBS for 40,000 PI lines. So we used both GBS and phenotypic traits to do this GWAS study here. higher the p-value means higher your ability to find these significant associations. So we used a mixed linear model because mixed linear model addresses both population substructure and kinship matrices because this is a very robust model which can address these issues. But I'll also tell we use both MLM and GLM later, and I will tell you why we did that. So these are the results from our GBS data for those new 10 lines. When they were added to the existing GBS data, we could still see that a lot of agreement, and they were forming the same set of substructures. So we did not have any problem with respect to population substructure when we merged all this data. So this is a PCA analysis of all these lines, which showed at least 3 to 4 clustering of all these lines. So again, the parameters remain same as before in calling these SNPs. So population structure of the panel were determined using the principal coordinate analysis. So here are the results from field phenotypic data. So the data I'm presenting today, it's all about manual data. We don't have any data from the robot yet. They're still under processing stage. We're still trying to do a lot of processing. So if you see here, this is how much variation we could see in these traits. So most of these traits, they were able to fit into normal distribution curve except for F-hit count and F-hit damage. So for the what we did was we took every trait and log transformed them, and we used them in GWAS analysis. So these are the results from GWAS analysis for year 2019. So first year we just tried to use MLM model, mixed linear model, where we just used one trait, that was flowering trait, as a covariate. So when we use one of these traits as a covariate, we thought it will definitely increase our probability of finding these associations. So if you see QQ plots here, for all the plant height and log transform plant height, we really found very significant tutorials, and these results were very much consistent with the previous results of other researchers. Whereas we could not really find much signals for F-hit damage and F-hit count. Probably these traits were skewed, that's what we thought. But what we did recently in the year 2020, we wanted to actually compare this GLM model. We wanted to do a GLM model with 1,000 permutations by using at least 3 covariates. So those 3 covariates were 2 principal component as well as 1 flowering trait as a covariate. So we used those 3 covariates and we did GWAS. So definitely it improved our results. It enhanced our results. We were able to find a lot of genome-wide association markers with a higher p-values. And if you see here, these 2 traits are more on plant height. So those were taken as reference because these traits were identified even in MLM model. So again, this is a slide which shows how when you log transform these traits, it really enhances your capacity to, your ability actually to identify these genome-wide association markers. So these results are not yet published. We are about to publish them soon. Again, this is an example when we average 2 years data and try to find how much increase we can really see, whether it's a GLM model or MLM model. We wanted to see these GWAS results. So this is an example where we took pictures of each plot. We have almost pictures of all 286 lines for each rep. So we have for 2 reps. So we have a lot of pictures. So this is just a representation of those pictures. So hope is that in the future, we want to study more on panicle morphology and diversity of this panicle in sorghum diverse panel that we have. So we had major challenges in this capacity building project. There are a lot of pest and plant related problems. There are a lot of robotic and technical problems. All these were related to environment as well as terabytes or 3 terabytes, it's really problematic. So that's why. So the outcomes from this is that we will definitely develop some good phenotyping methods, as well as we'll identify some good SNP markers. Again, I just want to highlight how important is phenotypic accuracy is needed to increase response to selection. So unless we improve on phenotyping methods, we really can't do anything much. These are some of my wax mutant projects where we were trying to identify how these waxes affected. Actually, we find mapped these regions, and these regions are now found that we found that there's a homolog of bodyguard gene here. Again, I did some soil microbiome projects. Really, I don't want to go much deep into this due to time constraint. My student, she did this. So she found sorghum has a very good soil microbiome enrichment for particular microbiomes like proteobacteria and actinobacteria. So in conclusion, I would say that genomic assisted breeding has seen a phenomenal increase with all the genomics data. Definitely it can be increased if we can increase, if we can enhance this phenotypic accuracy. And sorghum per millet have a huge genomic resources, as well as their huge germplasm resources, and all this genomic assisted breeding can enhance a lot of traits such as drought, as well as economically important traits. Again, most important is handling of data storage and management. So big data science, it needs to be explored more. So I'd like to acknowledge my postdoc, Adisiu, who joined recently. He has been helping me a lot. And I had 2 graduate students who helped me in this project, then undergraduate students. So these are the members of my capacity building project and area-wide project. So these are the references that I used. So I would like to thank organizing committee, and I would like to thank Dr. B. Fakhruddin. He has been a great mentor to me since my master's degree at IIBT US Dharwad. He is a great source of inspiration. So I would like to thank him personally. I'd like to thank my other mentors at US Dharwad. I'd also like to thank the entire UHS Padalkot team who conducted this conference. So it's really a tough job in this COVID time to conduct this. So you guys have done a great job. Thank you so much. Have a nice day. If you have any questions, please do email me at this following email IDs.
Thank you, Dr. Somshekhar, for an highly informative talk. You have given both on pearl millet and also on sorghum. And also you have given about capacity building project, which was really very nice about using robots for phenotyping. And it has really given you good results. But you also mentioned about its problems and also how to take care of them so that you can do good phenotyping, and a lot of other projects which you mentioned on phenotyping, on phenotypes of BMCs and soil microbiome projects and several other things. And it was a highly informative talk. I'm so glad that you are from our Dharwad Agriculture University. It's very nice. And there are few questions which I think there are in chat boxes. I think you can reply on to that, because paucity of time, I'll not be able to repeat those questions. Thank you. Thank you very much.
I'll do that. Thank you so much.
Thank you very much. Now we shall move on to the oral presentations, where there are 5 speakers in this. The time given is 10 minutes. They will have to stick on to the timings. If they don't stick on to the timings, you will have to stop your presentation at that time. All the lead speakers and also keynote speakers have really maintained the timings, and wonderful presentations were made. Now these 5 speakers, I'll first call upon Dr. Manish Srivatsa, who's a principal scientist from Division of Fruits and Horticulture Technology, ICR, IRA in New Delhi, who will be giving presentation on construction of genetic linkage map in mango. Dr. Manish. Okay. Dr. Manish, please. Are you online? Dr. Manish? Okay. Are you there, Dr. Manish?
Madam, I think he is not joined yet. Kindly move
Okay.
to the second speaker.
Yeah. We will then we shall move on to the second one. Dr. Girish Channappa, Senior Scientist, ICR, Indian Institute of Rice Research, Hyderabad, will be giving a presentation on development of an advanced backcross nested association mapping population derived from Oryza sativa cultivar IR64 with Oryza glaberrima. Girish, please.
Yeah. Yes, madam. Good afternoon, madam.
Yeah.
Yes, sir. Good afternoon, madam.
Yes.
So I'll be sharing my slides.
ಕೇಳಿಸ್ತಾ ಇಲ್ವಾ ಅಲ್ಲಿ
ಅಲ್ಲಿ ಬಹಳ ಡಿಸ್ಟರ್ಬೆನ್ಸ್
ಆ ಕಡೆ ಈ ಕಡೆ ಮಾತಾಡ್ತಾರೆ
ಇದು ಸಿಸ್ಟಮ್ ಕೆಳಗಡೆ ಇದೆ.
I know whether slides are visible.
Yes, sir. Yes, sir. Yes, sir. It's visible.
Thank you. Good afternoon to madam, Sakrama madam. I'm happy. I'm proud to say I'm a student of madam, and also thank you for the organization. So I invite all the delegates and audience for this presentation, oral presentation on development of advanced backcross nested association mapping population derived from Oryza sativa, cultivar IS64 and Oryza glaberrima. So myself, Girish, Senior Scientist in Indian Institute of Rice Research, Hyderabad. So to begin with, so we know that Oryza has two cultivated species, so Oryza sativa and Oryza glaberrima. Oryza sativa is mostly cultivated in Asian and South Asian regions, and Oryza glaberrima is another cultivated species which is widely cultivated in African regions. Although both they have the same genome, 2n is equal to 24, and AA genome, but they have the cross incompatibility problem. So because of that reasons, Oryza glaberrima and Oryza sativa not much been intercrossed. But we know the concept Nerica, where new rice for African regions. Oryza sativa was extensively utilized in crossing program for genetic improvement of Oryza glaberrima, because Oryza sativa has yield advantage when you compare to glaberrima. So they wanted to transfer several yield related traits to improve the genetic potential of the Oryza glaberrima in African regions, and several varieties have been released in Africa. But when we tested those Nerica lines in Indian, most of the lines did not perform up to the mark. So probably because of the adaptability issue. So in this program, what we thought actually that can we utilize Oryza glaberrima for genetic improvement of Oryza sativa cultivar to broaden the genetic base of the Oryza sativa, because we know that since the release of IR8 and other varieties 1960, there is any very less yield gain has been achieved in this one, although we have self-sustained in rice production. But genetic gain per year is very comparatively less. So that could be because of the narrow genetic base that exists in the parental lines, which have been utilized in the breeding program. So this program idea, we started that we want to utilize the Oryza glaberrima for genetic improvement of the Oryza sativa. So we know that Oryza glaberrima is endemic to African regions, and it has potential traits like early seedling vigor. When you compare to our sativa cultivar, it has a high early seedling vigor, one of the very important traits in direct seeded condition, and also wheat ability of the rice of Oryza glaberrima is much higher when you compare to our indica varieties. And also they have the several biotic and abiotic stress tolerance like blast, bacterial leaf blight, nematodes, insects, and pest resistance, and also like drought. Most of the African cultivars have potential for drought, and also salinity tolerance, and also low phosphorus stress tolerance. So with this idea we started, and the approach we wanted to begin with the approach what we wanted to use. There are two mapping strategies which have been commonly utilized in breeding program. One is linkage analysis, and another one is genome-wide association mapping. So both the methods have been extensively utilized for mapping and identification of QTLs and even oligogenes, and they have identified the functional markers. But both the methods have their own limitation, because power of mapping, if I talk on power of mapping, the power of mapping is very high in linkage mapping as compared to GWAS. And the resolution of mapping, the resolution mapping is very high in GWAS as compared to linkage mapping. And the identification of multiple alleles is quite possible in GWAS, whereas it is very difficult because there are only two alleles will be segregated in a given biparental crosses. So we can only detect 2 QTLs in linkage analysis. And linkage mapping population requires hybridization, which otherwise not required in GCAS, because you will be using naturally existing. And GWAS is mainly depends on LD, historical recombination. I think many speakers have spoken about this. So this is the idea was to that can we combine the principles of both linkage mapping and GWAS and have a mapping population which you can utilize for mapping higher power of to increase the power of QTL detection and also with higher resolution. That was the idea. So that is what is known as nested association mapping. It is basically a multi-parent strategy wherein the multiple diverse donor lines which have been selected from the wide array of germplasm will be crossed with a single donor parent, single recipient parent or common founder parent, and will develop the individual real population which together constitute the nested association mapping population. That means we will be having a set of a donor parent, diverse set, and we will be selecting one founder parent. We can also call them otherwise as a recipient parent. Will be crossing with a set of lines and developing independent real population together constitute real population. It is basically an integrative mapping strategy which combines the principles of linkage analysis and association mapping to achieve or realize higher resolution and also higher power of mapping complex types. That was the basic idea. So principally, it exploit historical LD like similar to GWAS, which are exist among the diverse donor parents which are utilized for the development of non-population. Some disturbance I am getting. Hello, yeah, hello. 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 Can you hear me?
Yes,
Hello,
sir.
madam? Yeah.
Yes, sir. Yes, sir.
Yeah, there was some disturbance. Sorry. Yeah. So it basically depends on 2 principles. It exploits the historicality that exists among the diverse parents which are utilized for development of non-population, and also there will be a recombination developed. Recombinants are developed during the development of non-population, similar to linkage analysis, linkage mapping. So both the principles are exploited to achieve high resolution, high power of mapping. What we are doing, like as I explained, there are different methods of developing of non-population. Like we can cross the elite rice varieties with germplasm, set of germplasm. We can go for development of real population, and also we can go for backcross population. This is called a near-resident non-population, or you can also known as AB non-population, advanced backcross non-population, and through double applied non-population, where if there is a feasibility to go for double applied, if standardized protocols are available, we can also go for the development of DH non-population, which you can see a lot of times. So these non-population which are immortal in nature, so that can be you can go for high throughput genotyping, preferable SNPs, and also we can go for trait evaluation, high throughput phenotyping facility. If both the data you can generate, then you can go for joint QTL analysis, and also you can go for NAM-GWAS approach for identification of QTLs. So this is basically the real population which are developed from NAM will be mosaic of both the parents which are used for the development of. This is just a diagram how the lines have been segregated. So this concept was first proposed by you on co-workers in 2008, and particularly in maize, the conceptualization, and they developed 5,000 lines derived from 25 real population. They used extensively for identification for several traits. They have extensively utilized. This is an example. So in your case, what we did, we selected the glabrima lines of around 33 lines we have selected, and we went for analyzing the morphological diversity, and then we went for the D-square analysis to identify the diverse lines, because that is one of the most important parameter for developing non-population. We have to identify the diverse donor lines. That is the basic step for developing population. So based on morphological diversity, we have shortlisted some of the lines, and also we went for molecular diversity. We have used genomide SSR markers which are present in the rice. We have genotyped the germplasm and went for cluster analysis, and we got around 5 clusters. From each cluster, we have selected some lines. So based on both morphological and molecular diversity. In addition to that, we also characterize the germplasm for early seedling traits bacterial blight and blast resistant, and low phosphorus tolerance, and also salinity, and like biofortification trait like protein, iron, zinc, and also yield parameters. We are characterized based on morphological traits and also based on molecular diversity analysis and based on these donor traits. So we have shortlisted around 10 lines that we should use for the donor parent for development number population. These 10 lines we made a cross with IR64. We got the F1s, and the F1s was completely sterile. Although both are cultivated species, both are having 2n is equal to 24, both are having AA genome, but there is a 100% F1s were 100% sterile in most of the cross. To some extent, we can see 5 to less than 5% seed set. That could be because there are several sterility genes have been reported in glabruma. They express when they come in contact, when they come together with sativa genome, the sterility gene will express, and also there is a embryo sterility genes have been reported. And despite they are having fertile genes for both pollen and female traits, the compatibility of pollen germination, lot of issues there. So this is the reason why glabruma is not extensively utilized for genetic improvement of whereas sativa. That is, so we decided that we should go for the backcross breeding. So we went with the backcross, and even in the backcross breeding, so CCS was significantly low. You hardly used to get less than 5% even in backcross. So through backcross breeding, we went and we got minimum amount of seed. Then we started selfing. Once we got a seed set, then we started selfing. Now we are released up to PUC1 F4 population, which are having more than 1,600 lines we have developed. And these characters, I am not presenting all the details, but the fact is that we are phenotyping these populations individually. We are phenotyping this population for early seedling vigor and also low P tolerance, and this is blast. And also we have screened for bacterial blight. We are individually using this mapping population, individual mapping population for mapping this bacterial blight resistance. And these lines, so it is a very important. And this is about our detail of the cross combination, and this is about the population size, what is presently we are having, and these are the targeted traits which are known. So this population need not to be necessarily used for this one. You can use a number of traits because there will be transcriptive segregation. So those transcriptive segregation also we can exploit using this 10 population. So the utility of ABNOM population, which have developed has a lot of potentiality that to understand the crossing barrier between the two cultivated species. As I explained, although both have belongs to a genome, but still there is a cross barriers. Despite in addition to the sterility gene, there are several unknown factors which are affecting the fertilization program. Because of that, there is an hindrance in the development of F1s and further backcrossing. And this population also will be useful, similar to the maize, how they have used it. We can use this population for understanding the genetic several complex traits which are segregating between the populations, so that we can study using this population. And also we can identify the novel alleles, QTLs or genes which are derived from varroa. Yeah, this is my. Yes, madam. And also this population, because this population is very large, there will be 1,800, 1,600 population. So this population we can use for differentiating the pleiotrophy, whether it is linkage, this kind of phenomena. And there is recombination events and segregation distortion. Of course, there is a high amount of segregation distortion have been reported. So this will be an ideal population for understanding the identification of genomic regions associated with segregation distortion and the rare allele and minor allele. And now people are using norm population for using genomic selection, prediction of GEB, genomic breeding value. So this population is highly potential. This is what about my presentation. Thank you.
Okay. Thank you, Girish. You have developed a very good population where you can use it for many other things. Many traits also you can use it. Good presentation. Thank you. Thank you. So next is Dr. Sarvamangala Colin. She is an assistant professor of genetics and plant breeding at College of Horticulture, Bagalkot.
Please unshare, Dr. Girish, please unshare that.
Yeah, I'm doing, madam. I'm just doing.
And she'll be speaking on exploring the genetic resources of carrot for functional genomics and marker-assisted breeding. Dr. Sarvamangala?
Yes, ma'am. I'm unable to share the slides.
Girish, you please unshare your slides.
Yes. I unshared it.
Slides are visible, ma'am?
No, not yet. Yeah,
It is visible.
you can start.
Yes, ma'am.
Please, before starting, please give less introduction.
Yes,
And
ma'am.
briefly tell your material and methods and go on to your. Okay.
Okay, this is Dr.
Visible,
Sarvamangala.
madam.
Today I am presenting on the exploration of the Indian carrot germplasm for marker-assisted breeding and functional genomics. So we all know vitamin A deficiency is the major reason for childhood blindness and is also a public health concern in most of the developing countries. It is associated with many other disorders and however, it is a preventable by consuming the foods rich in beta carotene. Carrot is one of the most potential food crop and a rich source of different types of carotenoids due to a wide range of carrot colors in its edible taproot. Being a temperate crop, availability of the carrot to the consumers residing in a tropical region is necessary. Botanically, carrot belongs to the family Apiaceae with a diploid chromosome number 18. Although it is a model system for carotenoid accumulation studies, however, breeders face number of challenges like it is highly outcrossing. Availability of inbreed lines are limited due to the high inbreeding depression and being biennial nature make it a long durated crop. The temperate adaptability is the reason for limited studies on tropical carrot improvement. These are some of the primary breeding objectives emphasizing on the root and seed yield and quality parameters. There are a number of breeding methods. However, marker assisted breeding is the top priority due to the availability of the high quality genome sequence. In order to characterize and utilize the Indian germplasm accessions, we initiated the tropical carrot improvement at UHS Bagalkot that represent the tropical climate of Karnataka with the initial funding from DBT Government of India. Like onion, carrot has to pass vegetative and reproductive phases to complete their life cycle. Here, root is the edible part and seed is the propagating material. Hence, a farmer can get benefit from both of these economic tissues. After root production, the top portion of the superior individuals will be sown for seed production. And germplasm need to be maintained by sip pollination under control condition. So this is the germplasm block at our campus. This is just a brief overview of the research work what we have done. Around 150 germplasm lines were collected from different parts of India representing the diverse geographical climate comprising of multicolored carrot. And they were subjected to phenotyping and genotyping in order to understand the population structure, genetic diversity. And the market rate association was done by association mapping for important root traits. So with this basic information, we could able to get a few superior lines adaptive to tropical climate. Among them, 4 elite lines were selected as founder parents for combining the desirable characters into a single population through marker-assisted recurrent selection program. We ended up with a nutrient-rich tropical biennial carrot successfully, and it is expected to be released shortly. Simultaneously, we selected 2 different genotypes belonging to the Western and Asiatic climate. Am I audible, ma'am?
Yes, madam. You go ahead, madam.
Yeah. These temperate and tropical genotypes were subjected
But PPT is not visible, madam. PPT we are not
power.
able.
There is no power here. Just 1 second.
Nothing can be seen.
Now is it visible, ma'am?
No,
Not,
yeah. The first slide, you please move on to the slide.
Is it visible, ma'am?
Can give, yeah, it is visible, slideshow, yeah.
Yeah. So simultaneously, we selected 2 genotypes belonging to temperate and tropical climate and subjected them to RNA-seq analysis in order to identify the genetic composition of these 2 genotypes and also to identify the candidate differentially expressed genes for important root adaptability and flowering related genes. So they were the genetic variations identified in this study are
Network issues. Lakshmi Devamma.
It's from Madam side, Madam.
Yeah, that's what. We'll just wait for a while.
I think Manish.
No,
is
we'll wait
waiting.
for another 30 seconds. If it doesn't come, then we can go ahead with it. We'll just give them some 30
Yes,
seconds.
madam.
Hello?
Yes, madam. Now you are audible.
No, Sarvamangala, I think her network is gone.
Madam. She tried to come from some other network.
Yeah. Maybe start the next one.
Madam.
Okay, we'll start the next one. Dr. Manish is here?
Yeah, ma'am.
Okay. Dr. Manish Srivatsav, Principal Scientist from Division of Fruits and Horticulture Technology, ICR, New Delhi.
Yeah.
He'll be speaking on construction of genetic linkage map in mango. Please try to reduce your introductory slides and go on to materials and results so that you can make up. Only 10 minutes is given.
Okay, ma'am. Okay. I'm sharing my screen. My slides are visible to everyone?
Yes, you can go ahead.
Thank you very much, respected chairman, co-chairman, and participants. Very good afternoon. I'm Manish Srivastava, Principal Scientist at the Division of Fruits and Horticulture Technology, IRI, New Delhi. And I'm here to present our work on construction of genetic linkage map in mango, which was carried out in our lab by our team. So as you all know that, okay, madam told that introduction is not required, but I want to emphasize a few of the points that if you consider the breeding in case of mango, it is quite difficult. And that is mainly because of the highly heterozygous nature of the crop and long juvenile phase. Excessive fruit drop and larger area is required for the meaningful assessment of the habits. And above all, the poor understanding of the genetics is making the biggest problem for the, and it is a major challenge for the mango breeders. So deciphering the genetics of important horticulture trait is very much required in case of mango to fast track the mango improvement efforts. And for this purpose, we utilized here F1 biparental prosony population of Amrapali and Sensation as mapping population for phenotyping and genotyping. And this particular mapping population was genotyped by 80,000 SNP chip. So as far as the material and methods are concerned, here we use 92 biparental prosony population of mango, and they derived from a cross of a single cross of Amrapali and Sensation. Here we utilize this Amrapali and Sensation because they are contrasting for 7-8 fruit quality traits like peel color, pulp color, fiber in the pulp, then TSS, beta carotene content. So there are so many fruit quality traits of importance for which these parents are contrasting. So we developed this biparental prosony population, and before going for the genotyping using SNP chip, we confirm the hybridity of all the prosonies, and then we perform the genotyping using 80,000 genic SNP chip. And whatever genotyping SNP data we got that was subjected to filtration, and finally we found that 71,000 SNPs were there, which can be utilized for further processing. And they were again subjected for the polymorphism studies, and we found that around 35,000 SNPs were polymorphic. And these polymorphic SNPs were further categorized in 3 groups based on their allelic pattern in the parents and their expected segregation in the prosony population. For example, if it is heterozygous in case of female and homozygous in case of male, they they were grouped together. And second group was where a particular locus was heterozygous for both of the parents. And third group was where it was heterozygous for the male parent and homozygous for the female parent. So we segregated all the polymorphic SNPs, and then we tried to construct the linkage map for female, male, and combined using joint map version 4. At the same time, we were also doing phenotyping of the mango population for more than 30 traits for 2 years, and we will be utilizing this phenotyping and genotyping data for identification of QTLs in case of mango, especially for the fruit quality traits. Here I want to tell that whatever mango gene chip we used on that particular chip, more than 80,000 these SNP probes were kept, and these 80,000 SNPs were belonging to they are representing more than 19,000 genes of horticultural or economic importance. They are categorized in 4 categories. One is cloned horticulturally important genes, second is disease-resistant different gene, and then third category is single copy genes and conjugates single copy genes. So this is a very useful mango gene chip for high throughput genotyping of mango. So as far as the conformation hybridity, we screened more than 500 SSRs, and we found some of the SSRs very useful for hybridity conformation in case of this cross Amrapali and sensation, and that has the combination of SSRs have been used for ascertaining the hybridity of the Prosney population derived from Amrapali and sensation cross. So these are the results about the genotyping using 80,000 SNP chip. We were lucky enough that 100% pass were there, no sample failed, and this quality check was 0.97. Call rate was also quite good. And total, we found that all 80,816 SNPs amplified in all parents as well as Prosneys, and they were subjected to filtration. And we discarded 9,139 SNPs, and we retained 71,000 SNPs for further processing. And out of the 71,000 SNPs, 34,860 were polymorphic. And these polymorphic were, as I told in material method, that they were further segregated based on their segregation pattern. And we finally 15,489 for female map and 11,295 for male map, and for combined map of female and male, 8,042 SNPs were taken. And these SNPs were then again tested for goodness of fit using chi-square test. And finally, we used 13,816, 8,000, around 9,000 in case of male, 5,000 in case of combined. And lastly, we could map only 12,975 SNPs in case of female map, 7,035 in case of male map, and 3,795 in case of combined map of male and female. So these are the statistics of a linkage map of Amrapali, means female parent. Here total 12,975 markers were mapped, and they were spanning over 20 linkage group, and they spanned over 3,650 centimorgan region. And the maximum number of markers were located on linkage group 1, which is more than 2,000, and minimum was in case of linkage group 17, 239 were there. Likewise, as far as the male parent map is there, total 7,000 markers we could map, and the maximum number of markers were again in linkage group 1, and minimum was in case of linkage group 6. And here I want to emphasize that this particular combination is very important, because a particular locus was heterozygous for male and female both. So here for these locus, our Prosney population was serving as a F2 population. So we could map 3,795 markers on different 20 different linkage groups, and maximum markers were located on linkage group 1, and minimum was on 12. If we compare, there are so many genetic linkage maps are published in case of mango. If I take the reference of recently published map by American researchers, Kuhn et al., they have only used 726 SNPs, and they utilized 7 different mapping population, and they could locate these SNPs on 20 linkage group. Another is from China, that is by Lu et al. They used 6,596 SLF markers, and they used a biparental ProSNP population. So in comparison to whatever genetic linkage maps are published there, in comparison to that, in our case, we were able to identify, to map more than 20,000 SNPs on 20 linkage groups. And this is something which is different from rest of the genetic linkage maps published in case of mango. And we are trying to utilize all three maps for getting a consensus map, and that information will be utilized for with the phenotyping data for identification of QTLs in case of mango, especially for the fruit quality traits. So here I want to mention that some of the phenotyping work was also carried out by our team, and we phenotyped the same population for more than 25 traits, from fruit shape to pulp organic acid profiling, beta carotene content, carbohydrate profiling, and all those traits we have taken. And here you can see easily that in terms of fruit shape and fruit peel color, there is significant variation is occurring, means phenotypic polymorphism we have observed in case of ProSNP population, and that is also true for the pulp color. And we got very good variation in terms of pulp color. We observed in terms of beta carotene content in the pulp also. Likewise, we also measured the traits related with the stone, fiber, adherence on the stone, and significant variation we have observed. So we are in the process of getting a consensus map based on male, female, and combined map, and that information will be utilized in combination with the phenotyping data for identification of QTLs in case of mango. This is the work which I tried to explain here before you, and we are working. We are in process of identification of QTLs in case of mango. So I acknowledge the financial support given by Department of Biotechnology, Government of India, and under DBT grant, and as well as ICR New Delhi for all sort of logistic support, Director IRI for providing all sort of facilities, Head Division of Fruits and Horticulture Technology, and more specifically, I want to thank you, Professor Nagendra Kumar Singh, who has extended all sort of facilities available in his lab for genotyping data and genotyping and processing of SNP data. Thank you very much. I hope I have not taken. I'm sorry, due to some network problem, I could not present.
Okay, no problem. Thank you. Thank you, Dr. Manish. It was a very nice presentation. Thank you. Now let's move on to Dr. Sarvamangala had stopped in between because of network issues. Sarvamangala, please unshare your slides, Dr. Manish.
Yes, ma'am, I'm doing.
Sarvamangala, please start. And you have 5 minutes, and you start from where you had stopped your presentation.
Yes.
You have only 5 minutes.
So these are the Indian germplasm collections collected and morphologically characterized for vegetative and reproductive phases for various root and quality flower characters. So this was the population showing the genetic diversity for the umbel and root related traits, and this is one unique root shape of umbel observed in our population, which is not there in the descriptor. So for molecular characterization, we have used different kinds of markers publicly available, and they were characterized for the population. A panel consisting of around 144 accessions with 80 polymorphic markers were subjected to population structure analysis, and 3 clear subpopulations were observed based on the delta K value. And these are the marker diversity measures of the population showing the diversity of the population. Further, neighbor joining tree and principal coordinate analysis of the population confirmed 3 subpopulations. So the pairwise FST and the niche genetic distance among the subpopulations indicated that the subpopulations are showing moderate genetic differentiation that is expected in the highly cross-pollinated crops like carrot. So this was published recently. And further marker trait association analysis was carried out in order to identify the significantly associated marker for important root related traits, and a few of them are being utilized in our MARS program. this is the Mars scheme developed for carrot for tropical improvement. This is the first of its kind in carrot. Here we have done the evaluation at vegetative and reproductive phases at different recurrent cycles after development of base population. And the phenotypic and genotypic selection was carried out. Genotypic selection was done for the target traits for which the marker is available, and genotypic selection was done for the traits for which the markers were not available, but they were important traits. Finally, genetic gain was improved for the target as well as non-target traits, and we have improved the biennial tropical carrot, which is superior for root as well as higher seed yield. So these are the list of gene-specific markers and their details. They were selected for the Mars program after communication with Dr. Philip Simon, who sequenced the genome of carrot and developed these markers. And this is the genetic constitution of the improved population, showing the marker diversity, marker parameters across different desirable alleles. So this is the generation advancement made by random mating using honeybees. So the genetic gain and selection response showed the improvement for the target traits, and this is the genetic constitution showing the population across the molecular markers for their desirable favorable alleles. So with this improved population of C2, we can see that the population is not only enriched with the desirable alleles across the marker loci, but it is also enriched, it is also fixed under homozygous condition without compromising for the yield and other related traits. So this was because of the combination of phenotypic and genotypic selections practiced in this program. So this is the Mars program, Mars population at C2 cycle. Now we have the C4 cycle population. Fine, and the two genotypes, Western and Asiatic carrot genotypes, were subjected to RNA-seq analysis for both root and flower tissues. So, the objective was to identify the differentially expressed genes for important root adaptability and flowering traits, and also to know the genetic composition of these two contrasting genotypes. So the funding was made by VGST, and the computational analysis is in collaboration with the IASRI scientists. So enormous amount of genetic variation was identified across the temperate and tropical root transcriptome in our root tissue. These are differentially expressed genes identified in the population in the study. And these are the SNPs distributed genome-wide in carrot, and the further related experiment and the publication is under progress. The genetic variations identified in this study are being utilized as genic marker-based QTL mapping. So for that, we have selected two F2 populations obtained from two heterotic hybrids, and the mapping is continued for important root-related traits. One more interesting study was we have explored the genomic resources of carrot for the genomically deprived crops belonging to Apiaceae family. So here we have used carrot markers for different seed spices and vegetables for which markers were not available. After studying them, we got good homology between these different crops. Around 10 to 12 crops are being utilized now, and the respective crops for which the genetic diversity is being undertaken using different germplasm lines for the respective crops. So this was published in 2019. Finally, I would like to conclude that we have explored the basic translational and applied research in our carrot improvement project in these 6 years, and we could able to end up with novel variety using the novel Mars scheme, and we have selected some superior multicolored carrot lines which are also under trial. So this is the study, and these are some of the publications, and I would like to thank my research scholars for their dedicated work. I also would like to acknowledge funding agency DBT and BioCare and VGST for their financial support, and UHS Bagalkot for constant support. I would like to thank the Collaborative Institute IASRI as well as Dr. Philip Simon for his expert suggestions during the critical time. Thank you. Thank you, ma'am. Hello.
Kindly unshare your slide, madam.
Yes.
Thank you, Sarvamangala. Now I invite Dr. Irdasappa, scientist plant breeding, who is from Directorate of Cashew Research, Puttur, to present on identification of putative molecular markers associated with nut weight and kernel weight and 4 growth traits in cashew. Dr. Irdasappa.
Very good afternoon, madam.
Yeah, good afternoon. Start sharing your slide.
Hello.
Please reduce your introductory slides,
Yeah.
move on to materials and then results.
Are my slides are visible, madam?
Yes, yes. Start.
Yeah. They are in slide mode.
Yeah, you go on to, presentation. Slideshow mode.
Yeah, yeah.
Yes.
Yeah. Thank you, madam. Yeah. Very good afternoon, madam. I will be presenting my topic, identification of putative molecular markers associated with nut weight and kernel weight and 4 growth sites in cashew. So I would like to brief in short why this study was taken. To highlight here, the cashew breeding is in very slow pace, and so far since 1971, we have released throughout India from the setup of DCR and ASLP and SAUs around 54 varieties. My friend Manish Srivastava has already pointed out the Indian breeding problems, like it applies to cashew too. Unlike in mango or in some other like apples, where the genomic resources are very much available, cashew is having very few genomic resources. So with the limited with the limited availability of those genomic resources, some workers have employed the markers for germplasm characterization, diversity analysis, DNA fingerprinting, and also to develop leakage maps and to detect QTLs. And my friend has already indicated in mango, and mango is being very well applied marker selection. And the major constraint in perennials is development of these F2s or reels and reels, but there is a ray of hope. We can use F1 as F1s which are heterogeneous and heterogeneous and behave similar to F2. So we used the mapping population developed in cashew for linking traits to linking markers to some economic traits. Yeah, this is about material and methods. Yeah, the experimental site is ICR Directorate of Cashew Research. We call it as Keminja campus here in Dakshin Kannada. The mapping population consists of 83 F1 plants, which have been derived from a tall and vigorous variety Ullal 3 and a dwarf succession NRC 492, which is being maintained in National Cashew Field Gene Bank of Directorate of Cashew Research. And these are the salient features of the parents. To highlight here, this is a dwarf parent. It is having very less weight, like nut weight of 5.4 gram, and Ullal 3 it is 7.3 gram, and nut yield 5 kg per tree. This is after a 6 cumulative orbits, and Ullal 3 gives 14.7 on an average kg per tree. And these F1 plants were actually F1s were developed during 2009, and they were field planted at a spacing of 6 meter by 6 meter in the field. And this is the view of the how the Ullal 3 variety looks like a tall and vigorous nature. And NRC 492, which is a collection from Brazil. Even after 10 years, its height is around 3 meter only. And this is the view of the mapping population plot in the field. Yeah, regarding phenotyping, we carried out phenotypic analysis, and the methodology we followed as per the procedure described in the experimental manual on cashew, erstwhile NRCC, which has developed this manual in 2005. And these are the traits we phenotyped, like internodal length, tree height, stem girth, mid-tree spread, panicle length, panicle breadth, apple weight, nut weight, kernel weight, shell thickness, shelling percent, and apple to nut ratio. Yeah, we extracted this genomic DNA from the F1s and parents using a CTAB method with a slight modification. which has been developed from the previous scientists who has worked here. And it has been quantified up to 0.8% of original electrophoresis. And overall, we used 138 SSR primers comprising from 21 from cashew, 28 from pistachio, almond, 24 from almond, and 65 from
Sir, we are not able to hear you, sir.
Internet problem, I think. You can call up. Can you call and find out? Hello?
Yes, madam, we are trying to call.
Yeah. That's why time gets. Oh, you can give it. ಇಲ್ಲ ಸ್ಕ್ರೀನು
ಮಿನಿಮೈಸ್ ಮಾಡಿದ್ರೆ ಸ್ಕ್ರೀನ್
ಇಲ್ಲ ಇದೇನು ಚಾಟ್
ಆ ಕಡೆ ಇದೆ ನೋಡಿ ಮೇಡಮ್ ಅಲ್ಲಿ ಬೇಡ ಮೇಡಮ್ ಅಲ್ಲಿ ಚಾಟ್ ಪಕ್ಕ ಆ ಕಡೆ ಸೈಡ್ ಒಂದು ಆರೋ ಮಾರ್ಕ್ ಇದೆ ಅಲ್ಲಿ either you can pop out or close.
Lakshmi Devamma, what's happening?
Madam.
Calling him, madam. I'm calling him. He is connecting, madam. PowerPoint.
Yeah, he's connecting.
Last
okay.
presentation.
Yes, madam. Madam, we can call the next person. By the time he can be ready.
Yeah. Okay, we will stop this presentation now and continue later.
Yes,
now I'll call Mr. Navneeta Krishnan, J, PhD scholar from School of Agriculture Biotechnology, Punjab Agricultural University, Ludhiana, to speak on mapping QTLs for heat tolerance component traits in a set of triticum, durum, and Agelot speltoides backcross introgression lines. Mr. Navneet, please.
Yes, ma'am.
Share your slides.
Hello, am I audible, ma'am?
Yes, you are audible.
Good afternoon, everyone. I would like to thank the organizers for providing me the opportunity for the oral presentation. So I'm going to present a part of my PhD research work on mapping QTLs for heat tolerance component traits in triticum durum, Agelop speltoides backcross introgression lines. So wheat is one of the most widely grown cereal crops, and around 35% of the global population depends on the wheat crop as the primary staple. So it is predicted that for every degree Celsius increase in the temperature, there will be a 6% decrease in the global wheat yields. So heat stress. Ma'am?
Sir, continue, sir.
So heat stress is one of the major production constraints in wheat production, and especially during the reproductive and grain filling stages, it significantly reduces the grain weight and grain number. So to tackle the heat stress problem in the near future, breeding and deployment of heat tolerant cultivars will be a sustainable approach for maintaining the yield of the wheat crop in order to feed the global population.
Have you shared your slides, Mr. Navneet?
Yes, ma'am.
Have you shared your slides?
Yes, madam. We are able to see here, madam.
Okay, you continue, no problem. I'll check. Continue.
Okay, ma'am. So coming to the background of the study, so this project was started way back in 2012 by we had screened around thousands of accessions of wheat wild species at our School of Agricultural Biotechnology, which led to the identification of Agelop speltoides, which is the probable beet genome donor of wheat, as a heat tolerant accession. And it was able to set seeds even at temperatures as high as 45 degrees Celsius. So we used this accession for initiating the mapping population. So we used the tetraploid cultivar Triticum durum PDW 274 as a recurrent parent, and we crossed it with a wild species Aegilops peltidus accession PAU 3809. So we used the tetraploid wheat instead of the normal hexaploid wheat because of the lower ploidy level, so that we can get the minimum chromosomal abnormalities. So the F1s, they were backcrossed 2 times with Triticum durum, and the population was allowed to sell for the further generations. And we genotyped the BC2 F4 population around 91 backcross inbred lines for using SSR markers, and we mapped several QTLs on different chromosomes. And this work was published in 2016 by a senior PhD student, Mr. Zudu. And we further enlarged this population to 310 lines, and we used the genotyping by sequencing approach at BC to F9 generations at Kansas State University under the lab of Dr. Jesse Poland. And further, we phenotyped the BC2 F10 and BC2 F11 homozygous backcross integration lines at replicated field trials, and we carried out the final QTL mapping. So coming to phenotypic screening, so we used the 310 bills, and they were sown under 2 environments: optimum sown environment and heat stressed environment. So in case of optimum environment, the wheat crop was sown during the 3rd week of November, which is the normal sowing time for the wheat crop in the northwestern plain zones. And in the case of heat stressed environment, the sowing was carried out in 1 month delayed around 3rd week of December, so that the crop will be exposed to high temperatures at the flowering time. And we carried out the phenotyping for 2 field seasons, 2017-18 and 18-19, at Borlaug Institute for South Asia, which is an institute under CIMMYT, Ludhiana. And the trial was carried out in alpha lattice design with 2 replications. So here we can see the field view of the optimum zone and heat stressed field trials, and there is a difference in growth due to the delaying of sowing of 1 month interval. And these are the different phenotypic traits which were recorded in the mapping population, like normalized difference vegetative index, which is a proxy for biomass accumulation at heading stage, peduncle length, spikelets per spike, grains per spike, spike length, days to heading, days to maturity, grain filling duration, plot yield, 1000 grain weight, and plant height. So here we can see the temperature data during the grain filling period for the 2 trials, 2017-18 and 18-19. And here we can see the clear-cut differences in the optimum zone and heat stressed crops. Like we can see to around 2 to 5 degrees difference in the grain filling, difference in temperatures in the minimum and maximum temperatures during the grain filling period for the optimum zone and heat stressed crop. Similar trends were observed in both the seasons. And here we can see the distribution of the various phenotypic traits under the optimum zone and heat stressed environments for the 2 years. So for all the traits, there is a significant decrease in the performance under the heat stressed environment. And here I am displaying some correlation matrix for the different heat tolerance traits with the plot yield, and those traits in the green blue circle, they represent the significant positive correlation, and those in the brown circle, they represent the significant negative correlation. So the traits like grain, 1000 grain weight, grains per spike, peduncle length, and NDVI, they had significant positive correlation in the optimum environment. And similarly, in the case of heat stressed environment, we can see the traits like 1000 grain weight, grains per spike, peduncle length. So these were having a significant positive correlation, whereas the days to heading and days to maturity, they had significant negative correlation. So we are done with the phenotyping. So coming to genotyping, the genotyping by sequencing was done using a 2-enzyme GBS method developed by Poland and co-workers in 2012 using the restriction enzymes PST1 and MSP1. So the genomic DNA was isolated, and it was normalized to 100 nanogram per microliter and sent for the analysis to KSU. And we got the raw reads, and they were processed using the Tassel GBS version 2 pipeline. And the SNPs were called against the A and B genome of the bread wheat Chinese Spring ref sequence version 1.0. And the VCF file generated was filtered further at depth level of 3 and converted into half map file. And from this, we identified the homozygous SNPs for each parental line, that is the Triticum durum and the wild species Aegilops peltidus. And only those polymorphic SNPs between the two parental lines were selected at 10% of missing data. And finally, we ended up with 3,222 high quality SNPs. So these SNPs were utilized for the further QTL mapping. So the integration profiling of the bills, they were carried out using the graphical genotyping 2 software. And here in this picture, we can see the different chromosomes arranged, 14 chromosomes arranged from 1A, 1B to up to 7B. And here the blue bars, they represent the integrations from the wild species, whereas the red regions, they represent the genome of the recurrent parent, that is the tetraploid cultivar. And the gray regions, they represent the missing data. So we can see that in each chromosomes, there are multiple integrations from the wild species. So the number of integrations ranged from around 20 to up to 400 integrations were identified per chromosome. And so the QTL mapping, it was carried out using the software QTL ICA mapping version 4.1 using the CSL functionality, which is mapping in chromosome segment substitution lines. So as these were not the conventional biparental mapping population, we have to use the special functionality for mapping in the integration lines. So the permutations were fixed at 1000 and p-value cutoff was 0.05. And the log value was fixed, the log value cutoff of greater than 3 was fixed to identify the significant QTLs. So overall, we were able to detect 21 QTLs for the various heat tolerance traits using composite interval mapping. So here we can see the screenshot of the software we used for the mapping. So I am showing some outline of the QTLs which were shortlisted having PV greater than 4%. So we were getting some QTLs, minor QTLs having PV like 2 to 3%, starting from 2%. So we just, I am showing the important QTLs having a higher PVE. And we were able to detect QTLs on the chromosomes 1A, 1B, 2A, 2B, 4B, 5B, and 7A. So the QTLs were detected for the following traits, namely NDVI at heading, like 3 QTLs were detected. In case of spikelet number per spike, 3 QTLs were detected. In the case of 1000 grain weight, 3 QTLs were detected, and 1 QTL each for peduncle length and spikelet number. And we also postulated the candidate genes in the marker region using the JBrowse tool in the IWGAC website. And we were able to identify from around in each QTL region, we were able to identify from 1 to 7 genes, and we had scanned the 50 kb upstream and downstream region of the SNP positions to look out for the candidate genes, and only the high confidence genes which were annotated were shortlisted for the postulating the candidate genes. And here we can see all the QTLs were having a high load score, and the PV varied between 4.5 to 8.7%. So, so the future plan of our work will be, we are currently developing a set of competitive allele specific PCR markers like the Cas markers, which are based on the SNPs linked to the QTLs. So these Cas markers will be developed from the SNP tags of the GBS data, and these Cas markers can be utilized for pyramiding multiple heat tolerant QTLs using marker assisted selection into elite wheat cultivars. So also we will be developing the near isogenic lines for each QTLs, and we will be validating those QTLs in the NILs. So we had already shortlisted a few backcross integration lines carrying the above mapped QTLs, and we are in the process of initiating crosses with the popular bread wheat cultivars, as well as developing the Cas markers for aiding in the marker assisted selection. So, thank you everyone for your patience.
Thank you, Mr. Navneet. Good presentation you made. Thank you. Now we shall move on to Dr. Irdasappa, where he had stopped. Are you online, Irdasappa?
Yes, sir, I am. Yes,
Kindly,
ma'am.
Navneet, please unshare your slides.
Yes, ma'am. Yes, ma'am. Done.
Dr. Irdasappa?
Hello, madam.
Yeah, we can hear. Kindly continue where you had stopped.
Yeah.
And I think you have less than 5 minutes.
Yes, madam. Okay, within 5 minutes I'll find out.
Yeah.
So sorry for interruption. I was in this slide. Yeah, we used 138 SSR primers, and there's a PCR application was carried out, and the PCR products were separated on horizontal 3.5% superfine agarose, and the bands were acquired through UV light using alpha imager delta system. And the size of the amplicon was determined with 50 and 100 kb ladder DNA as reference markers. And this, for parental polymorphism, we screened with 138 SSR markers, and the scoring was scoring pattern like this. The female, I need not emphasize all this.
Yeah, you can give the results. All these things are known to everybody.
Results. These are the results of phenotyping of the mapping populations. You can see here I have given minimum, maximum, mean, and standard deviation for each tribe. Here the tree height is 3.10, and it is averaging 5.74. And nut weight, we have got 2.37, 8.49 maximum. The another important trait, kernel weight without testa, it ranges from 0.88 to 2.73 with an average of 1.65. And another important trait, apple to nut ratio, which is generally 1:10. Here it is averaging 7.5. Here 2.812, 18.63 we have got, and it is averaging 7.95. And these are the pictorial representation of what the phenotypic variation we have seen. I have presented only 5 traits here for tree height, mean tree spread, weight of apple, nut weight, and kernel weight without testa. Most of them are following a normal distribution pattern representing the quantitative nature of these traits. And this is the image of the polymorphism in parents using almond SSR primers. And with regard to analysis, we used single marker analysis and chi-square test with that SPSS software version 16. And out of the 138, we got 32 polymorphic markers, and the total number of markers linked to traits are 20, and number of unlinked markers are 12, and we got marker loci not following expected ratio of 17, and with three marker loci followed the expected ratio of 3:1. And these are the significant markers which are linked to traits from the almond AL29, from the mango IM31, and another from mango IM28. See, this indicates the critical value significance at 5% and also at 1%. This is R square. It is nothing but phenotypic variation S-plant. It ranges from 4.5 to 17.6. Here we got more PVE percentage in IM31 for stem girth, which is an important trait, which is an important phenotypic trait in cashew. And here the chi-square value also we have given table. This is a calculated value, and we are given the table value 3.8. And these markers linked have shown the overlapped association with one trait or more than marker with the trait, or trait with more than one marker. So here, for example, all these 3 markers are linked to trait stem girth, and the 2 markers are linked to nut weight, and 2 markers are from almond and mango are linked to kernel weight, and 1 marker from mango linked to tree height and mean tree spread. This is a overlapped association. The conclusions from this study, there has been a considerable variability observed in the population, confirming the quantitative nature of these traits. The polymorphism obtained in this study is not more than 23%. Hence, we need to have a more number of markers for obtaining more polymorphism and then going into screening F1 population or mapping population. And the SMM, single marker analysis, indicated significant linkage of 6 traits with 3 markers, which I have seen in the table in the previous slide. And this I have already explained. And due to low formal markers, the linkage map was not constructed in our study. And however, from the literature, it has been cited that even the absence of with the absence of linkage maps also, this kind of studies can be conducted. So we did this kind of study. It is worth to note here that whatever the linked markers are there here, they are also linked to significantly linked to they have shown significant correlation. Let's say, for example, tree height, tree speed, and stem girth, they have the significant positive correlation with the yield. So these linked markers will be helpful in marker set selection of desirable genotype in the seedling stage, and thus they may reduce time and space for time required for cashew variety development. What is the way forward? The way forward is, to develop more SR primers in cashew or SSRs or SNPs, we need to develop to facilitate linkage map development and to identify QTLs. And whatever we have identified in this present study, 3 markers, we need to validate them in another population to get its conformity. So thank you. I thank the organizers for selecting for the oral presentation. I thank the chairperson, Dr. D.L. Savitrama Madam, who was my guide in my doctoral studies. Thank you very much.
Thank you, Dr. Irdasappa, for a nice presentation in cashew, which is a perennial crop. Thank you.
Thank you, madam.
I think we have completed all the presentations. To start with, Dr. B.M. Prasanna, who has given a very good keynote address about CIMMYT's work on maize, followed by 4 invited talks and 5 oral presentations. All the presentations were so good. I thank the organizers for giving me an opportunity for chairing this session. Thank you, Dr. Fakhruddin and his team.
Yeah.
And
thank you, madam. Thank you. Thank you very much.
Not online, I think.
He is here, madam.
He has made very good effort in organizing such a wonderful event, and he has had so many speakers across the globe. It is such a wonderful experience for me also to speak to, to listen to so many talks which are from varied crops. Thank you very much.
Thank you, madam. Thank you for successfully chairing such a lengthy session with your valuable inputs and suggestions. Thank you very much. On behalf of the organizing committee, I thank you, madam.
Thank you.
I would also like to thank the co-chairman of this session, Dr. Manjunath Gowda sir. And I would also like to thank the rapporteurs of...
He's here, Dr. Manjunath.
Thank you, sir. Thank you so much. Any inputs, sir? Remarks?
Oh, okay. I would like to thank the rapporteurs. I'm very sorry because I'm not able to see them. I thank the rapporteurs, Dr. Ratnakar Sheth and Dr. Raghavendra, who will be really making the good, who would have taken good points from each one of the presentations and who would be submitting to you. Next, Dr. Manjunath Gowda, co-chairperson. He's here. Yeah, the same screen.
Thank you, sir. Thank you. Thank you, rapporteurs, Dr. Raghavendra sir and Dr. Ratnakar sir. I would also like to thank all the speakers for such a valuable talks as well as oral presenters with diverse aspects. Thank you. Thank you, one and all. So now we'll move on to the next theme, next session. Another important brainstorming session, that is advances in molecular biology of biotech stress resistance. And the chairman of this session is our Honorable Vice Chancellor of University of Agricultural and Horticultural Sciences, Shimoga, Dr. M.K. Naik sir. Sir is here, Honorable Vice Chancellor of UAHS, Shimoga. And co-chairman of this session is Dr. M.S. Kulkarni sir, Dean PGS, UHS, Pagalkot. Sir, I request you, sir, to chair and co-chair the sessions respectively. Dr. M.K. Naik sir, our Honorable Vice Chancellor, a distinguished scientist with very productive academic career, has contributed extensively in plant pathology group, in identification of potential fluorescent Pseudomonas strains possessing antibodies. He is instrumental in identification of potential Azotobacter species for biodegradation of pesticides, and he has identified 3 new races of Fusarium udum, and he has also developed species-specific scar marker associated with the release of many varieties in chili, tomato, and rice. Sir has so far guided more than 34 PG students with several research publications in peer-reviewed journals with about 11 books to his credit as well as 2 patents. Sir has several national as well as internationally funded projects with several recognitions and awards. To name a few are ICR Bharat Ratna Subramaniam Outstanding Teacher Award, National Young Scientist Award, Fulbright Visiting Fellowship, University Best Teacher, and many other awards. So thank you, sir. Thank you. We are honored that you are here. Then our co-chairman of today's session is Dr. M.S. Kulkarni, sir, our Dean PGS of University of Horticultural Sciences, Bagalkot. Sir, as a professor of plant pathology with a distinguished career in the university at various capacities, sir has guided so many students, 3 PhDs and many MSc students with several publications, and he has several awards to his credit. To name a few are ICR Fellowship for his Masters, CSIR Fellowship, and recognized as Fellow of Indian Phytopathological Society. Sir, I also welcome you, sir, for this international e-conference for the session Advances in Molecular Biology of Biotech Stress Resistance. And rapporteurs of this session are Dr. Shankarappa KS and Dr. Shivayogi Ravalad. I welcome you also, sir. Sir?
Yeah.
Request MK Naik, sir.
Yes, madam.
Thank you, sir.
So very good afternoon to all of you. And I welcome all the participants and the dignitaries for this particular session on advances in molecular biology of biotic stress resistance. So needless to say, this is an important session, particularly when we talk about biotic stress, as much as nearly 25 to 30% loss is caused by these biotic factors. And many times, what change that the host and the pathogen or the pest brings in, we consider it as an irreversible change. But then our strategy will be generally just to see that we prevent the disease or try to arrest the disease. But then with the advancement in molecular biology, particularly omic era, we have got a wonderful opportunity to whether it is possible to bring a reversible change in the plant because of several tools being explored. As far as genomics, proteomics, they have given their own inputs, particularly in giving a very new insight into the biochemical pathways, gene network, chromosomal location, and also in giving an evolutionary history of plants. So in other words, there has been a very unparalleled perception of these particular new era. That's why I believe that this particular session will throw a much light upon the genes involved in pathogenesis, probably the most complex interaction of host pathogen, the interplay between the host and the pathogen. So that will help us in the selection of an appropriate promoter or transcriptome factor for transformation. These are some of the hopes. So keeping that in view, I would like to thank the organizers for giving me an opportunity, and I also welcome my co-chairman, Professor M.S. Kulkarni, my friend at the time of study at Harvard. We have, I think, 3 or 4 invited speakers, rather 4 invited speakers. First is Dr. R.L. Ravi Kumar. Is he there?
No, sir. No, sir.
He would join that.
He will be.
He is not there. Okay. Then I will call upon Dr. Ramesh Bhatt. Dr. Ramesh Bhatt, who will be speaking on peanut pan genomics, a futuristic resources for molecular breeding. Dr. Ramesh Bhatt is there?
Yes, I am there, sir.
Yeah. Just to introduce Dr. Ramesh Bhatt, very distinguished scientist with over 22 years of teaching and research experience at US Dharwad. He worked on specialty of land races and released 3 blast-resistant varieties, very important disease of rice. He has also cloned 2 novel lectin genes for developing nematode-resistant tomato transgenic, and also involved or engaged in genomic assisted breeding for foliar disease resistance, particularly for groundnut. For the first time, he has also proposed and demonstrated transposon-based marker system. He is also a recipient of Boy Scout Fellowship. He has been awarded Best Research Scientist Award by the University of Agricultural Sciences, Dharwad. The government of Karnataka has awarded him with C.V. Raman Award. So with so much of credentials, it is my pleasure to invite Dr. R.S. Bhatt to kindly make his presentation on this topic. Thank you, Dr. Bhatt.
Thank you, sir, for your kind words and a nice introduction. Okay, I'll be sharing my slides. Is it visible, madam?
Yes, yes.
Okay. Hope I am audible also.
Sure.
Yes, sir. Yes, sir.
Yeah. Thank you, Chairman sir, as well as Co-Chairman sir, and thanks to the organizers, especially Dr. Fakhruddin and Dr. Jagdish, who have invited me to deliver this talk. And the topic is peanut pan genomics, a futuristic resource for molecular breeding. And in this talk, I'll be covering mainly the peanut genomics, pan genomics, as well as the population genomics. These are the new terms and say concepts in any crop right now to understand a crop in a better way so that we can generate a lot of resources for better breeding. And also we'll take up some examples from our own study where we have used these informations to breed for disease-resistant peanut varieties, especially lately spotted rust-resistant genotypes by backcross breeding. So groundnut, as you know, we call it as Arachis hypogaea, which is a tetraploid crop, and it's allotetraploid. 2n equals 4x into equals 40 chromosomes, and important oilseed crop and legume, food, and fodder crop. Globally, it is grown on an area of 28.5 million hectares with a production of 46 million tons and with a productivity of 1,600 kgs per hectare. And in India, it's grown on an area of about 4 million hectares with almost 5 million tons. And in Karnataka, it's about 7 lakh hectares, and areas are coming down year by year. And if you look at globally, the half of the produce goes for oil extraction, while remaining half goes for raw as well as processed food. And peanut is commonly known as poor man's almond because of its rich nutrient contents in terms of proteins, fibers, polyphenols, antioxidants, vitamins, and minerals. Especially, they also contain resveratrol, and which is very important antioxidant, and phenolic acids, flavonoids, phytosterols, and as well as several amino acids, all amino acids, and highest with respect to arginine. So considering all these nutritional aspects, nowadays peanut is considered as a functional food. And when we look at the genomics and pangenomics, it's very important to study or look at the taxonomy of the crop. It belongs to the family Fabaceae, genus Arachis, which contains 9 sections and 81 species, out of which 72 are diploids and 5 are tetraploids and 4 aneuploids. And if you look at the origin of groundnut, then it was derived from a diploid or 2 diploids. This is A genome contributor, Arachis dunanensis, which was crossed to Arachis ipiensis, and the F1 was subjected for chromosomal doubling, and which resulted into the primitive wild type, which is known as Arachis monticola, which is AABB. And from this, actually, the cultivated groundnut was originated. And this all happened almost 10,000 years back. And later, the actual domestication started from about 6,000 years back. And these are the 9 sections, and various tetrapods and aneuploids are indicated in this say figure. And the Arachis section contains about 31 species, out of which there are 4 or 3 species which are forage in nature. And also one is ornamental species, there is Arachis repens. And if you look at the groundnut, then it is divided into 2 subspecies, which we call it as hypogaea and fastigiata. They differ for certain characters like branching pattern. Here it is alternate branching, whereas this is sequential branching. The flowers are born on the main stem here, and sorry, absence of main stem, flowers on the main stem, whereas the flowers are present on the main stem. And these are usually long durated, and these are short durated, and presence of dormancy and lack of dormancy. And again, you have botanical types like hypogaea and hirsuta, which we call it as VR and VB types, and Peruvian. These are not so common. Of course, these are very common hypogaea types. And fastigiata, most of the cultivated types belong to this one, vulgaris types, which we call it a Spanish bunch. And also we have Ecuadorian and Peruviana. So these are the botanical types. And if you look at the overall genetic diversity of groundnut crop, then there are various germplasm collections. In India, about 15,000 accessions. USA has got about 9,000 accessions. China has got 7,000. And using these accessions, various agencies have developed several core collections. And these 3 core collections in India had 1,700 accessions, whereas US had 831 and China had 576. And from these, again, the mini core collections have been developed, considering the overall variability and retaining overall variability, just to reduce the number. So the Indian mini-core collection has got about 184 collections, accessions, whereas US has got 112 and China has got 298 genotypes. And these mini-core collections have played a key role in terms of characterizing the important agronomic traits, as well as serving or supplying the new sources of novel alleles. And they have been observed to be better heritable and with very high ability, combining ability, and showing various high levels of trait associations or correlations. And therefore, these accessions have been used for breeding new genotypes. And peanut breeding suffers from, or there are several challenges. One is to improve the productivity and improving the tolerance to biotic and abiotic stresses. And in case of biotic stress, the foliar fungal diseases are very challenging and cause loss of damage, lot of damage. Then sclerotium, aspergillus, tomato spotted wilt virus, drought and high temperature, of course, abiotic stress, iron toxicity, and also nowadays the quality improvement is a major objective in any groundnut breeding. And the success rate of breeding and bottlenecks. Say, though there are several improved varieties have been developed, because of several drawbacks, genetic and other factors like undesirable linkages, lack of genetic diversity, and difficulty in selection has demanded for the peanut genomics. And when it comes to the peanut genomics, we were all delighted when the first paper came in 2016, reporting the genome sequence of both the AA and BB genomes, progenitors, by Bertiel and his associates in 2016. And also later, Chen and associates in 2016, the same year, they published the genome sequence of Arachis dunanensis, as well as Liu and his associates in 2018 sequenced the Apiensis genome. So now we have the complete genome sequence of both the progenitors, there is a wild diploids, A genome and B genome, and also the genomes of the primitive tetraploid, which I told as Arachis monticola. It is also sequenced completely, and this is done by Yin and his associates in 2018 as well as 2020. And later, the International Peanut Genome Initiative led effort published the Arachis hypogenome, the cultivated groundnut genome, and it was published by Bertioli and his associates in 2019. And this mainly published the Arachis hypogea, subspecies hypogea, a variety called Tifrunner that was completely sequenced. And then the Arachis hypogea subspecies fastigiata, two varieties of these subspecies were also sequenced, and both these studies were published in the same volume and same issue of Nature Genetics in 2019. And this is say several features of the genomes are available. And coming to the peanut pangenome, why pangenome is required? Actually, it's a forward step from genomics. Genome sequencing projects have revealed high degree of genome variation between the individuals. So considering the difference between the individuals, now it is essential to look at each individual with respect to its genome. So therefore, nowadays the pangenomes are very important, and they are being studied in various crops, including rice and other crops. And also nowadays, people have introduced another term called as super pangenome, where even we look at all the species belonging to the same genome or belonging to the same genre. Say Arachis has got several species. If you consider the genome sequence of all the species, probably that would make the super pangenome. And peanut pangenome, we made several efforts to study the features of pangenome, for which we took the 231 genotypes for which the genome sequence is available in the public database. And then we compared to the Tifrunner and observed about 4 million SNPs. And these SNPs were the A subgenome had more number of SNPs compared to the B subgenome, and chromosome number 18 had the lowest number of SNPs when compared to chromosome number 3. And similarly, the copy number variations, they were also varying between the individuals, and also you found some of the presence and absence type of variations. This is very important when we look at especially the genes. Say in certain cases, a gene can be completely absent in one genotype and it can be present in another genotype. So such variations are to be said, we have observed such variations, and which are called as presence absence variations. And this is a copy number variations. A lot of variations only you can see here. This is a genome donor. When compared to Tifrunner, a lot of variation is seen for B genome, but less variation for A genome. And this is with respect to the comparison between B genome donor and the Tifrunner. You can find more variation for A genome as well as less genome variation for the B genome. So like this, you can find a lot of variation between the individuals. This is the comparison for GPBD4 versus Tifrunner. You can find a lot of copy number variations. And we also worked out the structure, population structures, clusters, all those things using 231 genotypes. And what we found is when we consider different individual chromosomes, the number of groups varied. You can see here, when you looked at most of the chromosomes, the number of groups were only 2. Whereas when considered the chromosome number 17 and 18, the subgroups or the clusters were 6. It means the chromosome of 17 and 18 have got better resolving power with respect to clustering. And we also did some kind of population structure analysis. The same thing happened here. The clusters varied from chromosome to chromosome, and this is considering all the chromosomes at a time. And we found about 8 to 9 different population structures, and these are some of the features when compared the 3 different species were compared: the polyploid diploid progenitors, Arachis ponticola and Arachis hypogea. We found a lot of important features, which of course will be coming in the form of book chapter. And also we looked at the epigenome by sequencing about 11 genotypes by DNA methylome sequencing as well as expression pattern. And we found a lot of important features with respect to the epigenetics. And we found out the number of sites. See, the number of sites, CHG sites showed very high number of sites across 11 genotypes. And we found exactly which sites, whether it's intergenic regions, genic regions, and within the genic regions, whether the introns, exons, or the regulatory regions have got very high number of methylation sites, then all those features have been, were studied across these 11 genotypes, and important features have been documented. And also we studied the genes. The number of genes showing DNA methylation varied from genotype to genotype. Say, for example, ICGV 86855 genotype showed 51,000 genes, which showed at least one methylation, whereas this genotype showed 55,000 genes, which showed at least one DNA methylation. And this gene, which is located on chromosome number 11, showed highest number of methylated sites. And also the number varied between the genotypes. And also we found negative association between the methylation sites as well as the expression pattern or the level of expression. And also like this, we made several studies with respect to epigenome. And coming to the transcriptome, which is the study of the RNA. Unlike the DNA, here we study the RNA. And the Arachis hypogaea gene expression atlas has been published for Fastigata type. And also several transcriptomic analyses have been published for various traits. It could be seed dormancy, plant height, pod development, nodulation, like that. So for so many traits, the transcript analysis in terms of RNA sequencing has been done. So these are the important, say, resources for further molecular breeding. And what are the applications of these, whether it's pan-genome sequencing or populations genomics like that? One is the understanding the genome basis of fitness, then local adaptations, phenotypes, and unraveling the local specific effects rather than genome-wide effects, and genome differentiation, how the genome differs from one, say, structure to another substructure, that can be studied, and identifying the domestication related genes, how the groundnut has domesticated and which genes have contributed more in terms of, say, domestication. So such things can be studied. And also for marker trait associations in terms of GWAS or genomic selections, trait mapping, as well as even marker development like that. Say for genomic selection, which is a unique effort in case of peanut, we have constituted about 340 genotypes for mapping purpose. And this is our first study. And also recently we have published another study where we have standardized several models for genomic selection. And these are our mapping efforts. We have different mapping populations. And also we used these informations for developing markers, new marker systems. Because though there are several different marker systems available, including RFLPs, DARTs, AFLPs, SSRs, SNPs, like that, the overall polymorphism is very, very less. It is about, say, 5 to 6%. So we wanted to have a marker system which can lead to very high polymorphic rate. So we consider the transposable elements. And transposable elements, because of their high copy number as well as variation in the site of integration or insertion, they morphism because of their variation in the insertion site. So we made use of that information for developing a new marker system. And in this study, we demonstrated that the transposable elements can be used for developing the marker systems. And later, Shirasawa and his associates from Japan actually developed about 500 odd markers. And later, using 33 genotypes, the resequencing data of 33 genotypes, we developed about 3,000 HT markers. So these are the 3,000 markers across different chromosomes. And also we determined the positions of these transposable element insertion sites, whether it is the intergenic region, exonic region, intronic region, UTRs, upstream region, downstream region, all those things. So this helps even when we identify a marker which is associated with the trait, then it becomes easy even to identify the gene. So this is the beauty of this marker. And we found that out of 3,000 markers, about 82% of the markers were validated and which could be amplified in the lab. And another beauty was the polymorphism rate, as high as 35% in a mapping population which is derived from VL1 into 110. We found polymorphism rate of 35%. This is against 5 to 6% polymorphism rate of other markers. So this is the advantage of this marker system. And also it is very easy to look at the marker on the normal agarose gel in the routine or in a small labs like us or small life like ours. And then we used these markers for extensive mapping. And this was the first map in 2010. And later the same marker was enriched to identify more and more markers in the same region. And then we narrowed down the two important regions on A02 chromosome and A03 chromosome. So this is with respect to A03 chromosome. Earlier it was about 8.8 MB, and with our effort, we could reduce the genomic region to about 2.7 MB with various efforts of fine mapping. And similarly, we did the fine mapping on A02 chromosome, where the area was, or the region was reduced about 1.44 MB. And this also led to the identification of key genes in those regions. For LLS, on A02 chromosome, we identified 4 genes, and on A03, for rust resistance, we identified 6 genes. And similarly, we went for QTL-SIG, and so those genes were validated. And also now various transcript analysis is also done, and those studies also indicate that these genes are very important for late leaf spot and rust, which are very, very important diseases of peanut. And together they can cause about 70% yield loss. And breeding is relatively challenging for these diseases because they co-occur, as well as their undesirable linkage with the pod characters in terms of reticulation and constriction, as well as low yield. So therefore, we thought of using these molecular resources or genomic resources for molecular breeding, for which we took various sorts of recurrent parents, the JL24 and TME2, which are very, very popular varieties even now, even after about 40 or 50 years of release, they are still popular among the farmers. And we used GPBD4 and other donors, which are highly resistant for these diseases. And then we did a lot of backcross breeding, and we developed a lot of backcross lines, and we published, characterized those lines and published in these publications. And then these lines, which we call it as DBG3. DBG3 is nothing but it's an improved line of JL24. JL24, of course, it has got each and every desirable feature, except it is susceptible for late leaf spot and rust. Similarly, we developed another variety called as DBG4. It is almost equal to or similar to TME2, and it has got resistance for late leaf spot and rust. Apart from this disease resistance, these two varieties or new genotypes backcross lines have got yield advantage as well. About 15 to 20% yield advantage is also seen in DBG3 and DBG4. And they have completed station trials, multi-location trials, and now we even the last Kharif season, we have completed the farm trial also. So this is the picture from the farmers field where our director of research and seed unit people visited. And you can see here, this is DBG3 and this is JL24. Similarly, this is DBG4 and this is TME2. You can clearly see the disease resistance in DBG4 as well as DBG3. And this is the seed production. We are hoping to produce about 2-3 quintals of seeds so that they can be distributed for various other trials as well as to the farmers. And the couple of years back, the agriculture minister as well as Dr. Ashok Dalwai, so many dignitaries from ICRISAT also visited. And all these resources in terms of genomics or population genomics have been recently published in a review paper called Translational Genomics for Achieving Higher Genetic Gains in Groundnut. Because after generating so much of genomic resources, it is very important to translate these genomic resources for actual breeding purpose. So this, in fact, similar kind of molecular breeding is also done at ICRISAT as well as TNAU and DGR Junagadh. So in conclusion, what I would say is the phylogenomics and population genomics platforms in plants greatly depend on the reduced cost of high throughput sequencing, because the pangenomics and population genomics, they greatly depend on sequencing and resequencing. And recently there is a paper by Hall and Associates where they have talked of reducing the cost in terms of constructing the library and target amplification, enrichment and sequencing like that. And also it requires the robust bioinformatics tools to compare the genomes to find out the common features as well as differing genetic contents. And then of course, these things can be used for trait mapping, QTL identification, candidate gene discovery like that, so that it can be either used for molecular breeding or even for transgenic purpose or development of the transgenics. And identifying the epistatic interactions, epigenomics can also be useful. Of course, these will be useful very much for genomic selections and genome-wide association studies, and put together, all these resources can be used for molecular breeding. And nowadays, people also use these informations for gene or genome editing purpose as well, as it has been done for FAD2 gene, which contributes for oleic acid content. And nowadays, a lot of efforts are going on to enhance the oleic acid content, because by enhancing oleic acid content, you can enhance the health benefit as well as keeping quality of the oil of groundnut. So thanks to the various funding agencies and my advisor, Dr. M.V.C. Gowda, various collaborators, especially Kenta Shirasa from KDR Japan, people from ICRISAT, my co-PIs from US Dharwad and other universities, and students, PhD students and MSc students, and all the field and lab students, lab assistants, and finally, thank you. And thank you all the participants. Thank you, sir. Hello?
Hello?
Yeah.
Sir? Kulkarni, sir?
No, I don't want to interact because I think
MK Naik, sir?
Hello?
Hello?
Sir, Kulkarni, sir, you can continue, sir.
Madam, Naik sir mute ಮಾಡಿದ್ದಾರೆ.
Yeah, sir. Yes, sir, we are unmuting, sir.
Okay. Yeah, if there are any questions, I can answering through chat, right?
Yeah.
Okay.
I think he has made a wonderful presentation highlighting what is pan-genome and super pan-genome and its features. So also what variations exist. And he has also given transcript analysis of for many traits, isn't it? Regarding fitness, its local adaptation, and genome-wide its effects. And also he has validated the markers. Good validation he has done. And transcriptome analysis. And he has finally concluded that these genomic resources could be very well used for molecular breeding, and has come out with DBG3 and DBG4 and has checked against JL24. I'm very glad to say that since we are missing the TMV2 variety, that's what we see that the groundnut breeding, that is the one deficiency that we are always highlighting, that farmers are not ready to give up TMV2, a very long old variety. But the thing is now that he is telling that the DBG4 is similar to that of TMV2, in addition having resistance to late leaf spot and also for rust. So I think it will go a long way in exploiting this particular phylogenomic and population genomic platform, isn't it? If there are no observations, I think we will thank Dr. Bhatt for his presentation. Thank you very much. Are there any comments from Dr. M.S. Kulkarni? Dr. Kulkarni.
Yeah, please continue, sir. I have already told my views.
Okay.
It's a very good contribution by Ramesh Bhatt and a nice presentation. Thank you.
Okay, then thank you, Dr. Bhatt. Thank you very much. Yeah, now I will invite Dr. Basavaraj Bagewadi. Is he there? He will be presenting on microarray and next generation sequencing for detection and identification of plant viruses. Just to briefly introduce the speaker, Dr. Basavaraj Bagewadi is a postdoctoral research associate with Dr. Naidu. Dr. Naidu Ayurpati at the Department of Plant Pathology, Irrigated Agriculture Research and Extension Center, PROSER. He has also had his experience at Washington State University and conducted research on diagnosis and molecular analysis of viruses infecting grapevines and vegetables. So with this rich experience, I would now invite Dr. Bhagewadi to make his presentation, please. Dr. Bhagewadi, please. So you have been given, I think, 25 minutes.
Bachora sir, kindly unmute your speaker, sir.
Thank you, sir. Am I audible now?
Yes, sir. Yes.
Okay, thank you. So I am going to present today the work I did during my postdoctoral research in USA. So that is on the development and validation of universal plant virus microarray, and compare this microarray for plant diagnostics with the NGS method. So we can classify plant diseases as old, new, emerging, and re-emerging. And all over the world, viruses are one of the major causes of plant diseases. Of about more than 40% diseases are plant diseases are caused by viruses, and the reasons for these viral diseases are including the cultural practices, climatic changes, and trade and commerce. So now I'm going to okay. So we all know, we have discussed since morning the impact of plant diseases, and in some cases the losses is we can get complete loss of crop yield, and also these diseases cause barriers to international trade and commerce, and also adverse effect on our health. And how can we control the plant diseases? We all know that we cannot eliminate the plant diseases once we get the disease in the field. Only way is to prevent them. Prophylactic measures we can take, and among them is the use of resistant varieties which have like long duration resistance. And among management practices is prevention is the best method. For prevention, we have to identify the source of viruses in the seeds as well as in the planting materials, especially in the horticultural crops. If the planting materials are infected, especially in like potato, grapevine, and other vegetatively propagated plants, it is very difficult to manage those diseases in the field. Okay, so there are several methods of plant virus detection and identification, starting from morphological features and electron microscopy. In case of morphological features, it is very difficult to distinguish diseases caused by various viruses. And even in electron microscopy, we can only distinguish the viruses up to species and genera level, but within genera we cannot distinguish viruses to the species as well as different strains. And then came the molecular approaches during 1970s, starting with ELISA. For ELISA, we don't need any nucleotide sequence information. We can use the whole virus to develop antibodies and use for the serological detection. And then later came the gold standard, that is PCR. PCR is highly sensitive, but it has a limited range. Using PCR, we can detect at the most a group of viruses belonging to a particular family or particular genera. And next came during 1995 and in the 2000s, DNA microarray. So DNA microarray, there are various kinds of DNA microarray like cDNA based microarray, oligonucleotide based microarray, bead microarrays, and so on. So with the microarray, on a single glass slide, we can have spot the cDNAs or oligos of the several viruses, and we can, in one test, we can screen for the multiple viruses. Otherwise, we have to do PCR test one by one. That takes a very long time to screen valuable germ plasma or nuclear seeds. After microarray in 2006 came NGS method. Initially, it was very, very expensive. And 2012 and 13 onwards, it became affordable. So as we can see, except DNA microarray and NGS method, all methods detect only few viruses at a time. And there have been several attempts previously to develop microarrays for the particular genera or family or group of plants. For example, viruses affecting potato, viruses affecting grapevine. But there had been no effort to develop a universal microarray which can detect all the known plant viruses like RNA viruses and DNA viruses. So our work started based on the previous work done at University of California, San Francisco. So they first developed in 2002 chip viral chip, and which had which could screen 1,000 viruses, including plant viruses. The chip was common for plant as well as animal viruses, and in that microarray there were probes for around 350 plant viruses. So we thought, can we develop similar DNA microarray for the plant viruses? So initially, to see the feasibility of production, so we took probes available on this DNA microarray to make a small demo chip. So we made a prototype DNA microarray with 750 oligonucleotides of 70 MERS, and that could target 54 virus species in various families. Based on the success of this demo chip, we ventured into full-scale DNA microarray, which can screen all the known more than 1,300 plant virus species. In this slide, we can see the variation in the plant viruses. As we can see, the Clostridioideae, the genome is, they have a very longest genome, but more or less all the plant viruses are about 20 kb in length. And also they are, some are RNA virus, some are DNA virus, single-stranded, double-stranded, etc. So taking into all this account, we contacted a bioinformatician at Utah University, Cal Fisher. So he designed probes in such a way that probes are specific to each species as well as family and also to the order. So we can see here that unlike bacteria and fungi, we don't have common sequences across all the viruses like 16S rRNA, 18S rRNA. We don't have those sequences like those sequences in case of viruses. So we designed the probes called hierarchical probes. Here, family level, genus level, species level. And we obtained by bioinformatics analysis several thousands of probes to reduce the cost. We limited the number of probes for each order. We limited 5 probes, for family 10 probes, genus 10 probes, and for species only 2 probes. With this all, so we designed a total of 9,600 probes, and these probes are in sense orientation as well as anti-sense orientation. So these oligonucleotide probes were spotted. They were initially evaluated for whether they will be effective or not effective based on binding energy profile. And these were synthesized. The probes were synthesized and spotted onto the, using a robot, spotted onto the glass slides. And this is the DNA microarray with 9,600 spots. The red spots are the spot markers. Red spot is wherever we see the green spot, they are the either positive control or the presence of virus. So we can see here, this is just one example for cucumber mosaic virus. We can see for each virus, on an average, we had 20 probes, 2 at the species level, 10 at the genus and specific species family level. So we had multiple probes. This is before hybridization, and after, this is after hybridization. And we developed the microarray in 2 formats. Earlier one was just 16 block, and here we had in a 32 blocks. It takes the different amount of time. So it takes around 11 hours of spotting to spot 10,000 probes. And this is comparison of demo chip and full version chip. So once we developed this microarray, so we wanted to validate how it works. So to validate, we contacted DSMG, German collection of microbes, and they gave us all the positive control samples, and we started hybridizing them using the Psi3 and Psi5 dye. And here we used only RNA here. So we know that plant viruses belong to both DNA as well as RNA genomes. But DNA viruses also make RNA during their transcription. So utilizing that feature, we extracted only RNA. Because in microarray, we don't know the plant sample, which virus is there. So we used RNA as a starting material, labeled it, and hybridized onto the slide, and scanned using the laser scanner, and we got the readings for those. Yeah. So this is example for cucumber mosaic virus positive sample, and as you can see here, the probes for cucumber mosaic virus are spread across all the 16 blocks. Even if there is a scratch or something missing here in one block, the result will not be affected. Okay, so this is the quantification of the result. So all 9,600 spots were scanned and were given the value from 0 to 65,000 value. So as you can see here, the probes which are marked in blue, the highest, the spot showing highest spot was the positive control. That is the Rubisco gene. And the next highest one is the probe. And in the top, you can see each probe is given a level. Either it belongs to species, genus, or family. The first one, the cucumovirus, is a genus level probe here. And similarly, here you can, below you can see from the bottom, the first blue probe, it is cucumber mosaic virus. It is a specific species level probe. And same here, Bromoviridae, family level probe. So this is the first time we have designed such a method. So all levels of probes are included in the array. So similarly, these are different samples. And we used another method. The first method was just visual method quantification. Second method is we used hierarchical clustering to remove some noisy probes, as well as to cluster. Can we cluster healthy samples from diseased samples? So we could separate those healthy samples. So this is, we validated with more than 300 plant viruses in different virus families. So we also used some more samples from SIP, Peru, SIAT, Colombia, and others, so ATCC, USA. And we used several samples like tissue culture samples in potato and all. And sometimes there was no detection. So we wondered, some samples were not, even though the virus was there, positive by PCR, but negative by microarray. So the negative in microarray doesn't mean the virus is not there. So we wanted to validate. So we tried to increase the sensitivity of the detection. For that, we did a random amplification first. So random amplification did not help much, because in the random amplification, everything was amplified using the random primers. And second method we did is we used the more of total RNA. In case of plants, there is no dearth of starting material. Whereas in case of humans and animals, we don't get enough samples like throat swab and all things. But in case of plants, we could get plenty of RNA. We increased RNA amount, and then also we got some more increase in the sensitivity. And third method was expensive and time consuming. We also tried that. That is the depletion of rRNA. We all know that total RNA has more than 90% of ribosomal RNA. So we tried to eliminate total RNA. and that increased the cost by $100. So we did not use again much for that. So these are the results with those enrichment methods. So this is another method of increasing the sensitivity was to identify the signals in the microarray. We used two-way clustering, and using that method, we detected many other viruses. So overall, this is the summary. On the left side, we see around 20 plant virus families. Along with viruses, we also used viroids probes and satellite probes, virus satellite probes. Okay. Now we know that in till 2006, the next generation sequencing was not there. Only DNA microarray was the universal method for virus detection. In 2006, NGS came, and based on the previous experience, it was shown that NGS is highly sensitive, and we don't need any prior nucleotide information. For microarray, initially we need micronucleotide information to spot on the slides. But in case of NGS, we don't need any prior information. And another advantage of NGS is we get complete, in most cases, complete genome sequence and also the information of variants. And finally, the multiple agents of etiology, especially in case of horticultural crops, for example, grapes. So in grapes, we find many viruses, not one viruses, many viruses are there, which are difficult to identify by PCR, even by microarray. So during this workshop, we have seen a lot of information about NGS, and there are several platforms for NGS. Most 90% of the cases we use Illumina samples, and we use either for virus diagnostics, single end reads are enough, of 50 base pair or more is enough. And PacBio sequencing method, it gives very long reads, even with some errors, but PacBio reads helps in assembly of virus genome, especially in case of Clostridioideae, Potiviridae, the viruses with long genome. So PacBio method helps in virus genome assembly. And another method is Oxford nanopore method that we can use in case of field also. That is portable. These are not portable. And another difference between microarray and NGS here I want to say is, it is, of course, it is very expensive. In practical cases, to get the results in NGS, it takes at least 1 week. Whereas in case of microarray, within a day, in 24 hours, we can know the result. Here also, just as in case of microarray, we used total RNA and single end or paired end library methods. And NGS method, the NGS type is RNA-seq basically. And once we isolate total RNA, treat with DNase random amplification. And after sequencing, we use the reads to blast against the virus RefSeq genome database. So this is some examples just to show that these 12 samples were sequenced on 1 lane of Illumina, 2500 high-seq. And as you can see here, all the 12 samples on an average 40 million reads were obtained. In the third column, you can see percentage of reads mapped from 1% to 77%. Depending on the virus and host, the percentage of reads vary. And this is just one example here. All the raw reads there are two methods. Once we do NGS, either we can remove the host sequences. That is not possible in most cases because most plants are not completely sequenced and it is bioinformatically very time consuming. So what we do is simply take all the reads and do a blast search using CLC Genomics Workbench. And we sort from highest to lowest. So there, in this case, we can see zucchini tigre mosaic virus, bitter gourd yellow mosaic virus, and like that we can see. It gives a very quick result, and we have to analyze to see whether it is true or not. So here is one example to show the information we get by NGS. We get complete genome distribution, and the ends can be filled with race also. And another thing we tried is we tried samples which are spotted on FTA cards, or and also from the dry leaves. So the RNA quality is not that much important here for diagnostic purpose. We can easily use any RNA present in a dried leaves or herbarium samples. So I did comparison with NGS and microarray. So the samples which were negative in microarray, we knew that the samples, we took positive samples only, like soybean mosaic virus, papaya mosaic virus, and other viruses, and more or less more than 70% there was a concurrence between two methods. But of course NGS is very superior method. And so the just presence of virus doesn't imply that that causes the disease, and we have to follow the standard rules, and including for viruses, modified postulates, as well as some more guidelines given by Fredericks and Rellman. So with this I will conclude my talk. So in this work we developed and validated a full version of universal plant virus microarray, and keeping the cost low, and a single hybridization protocol based on total RNA was developed for both DNA and RNA viruses. And so also enrichment for low titer viruses, enrichment methods were developed. So with this, I would like to thank all the collaborators and my PI in USA. So this work was funded by USDA and was carried out at Donald Danforth Plant Science Center, St. Louis, Missouri. Thank you. I'm happy to take any questions.
Basavaraj, for a good presentation on microarray and next generation sequencing detection of viruses. Are there any one or two specific questions or observations from the participants? One or two quick questions or observations. No? Yeah, I think Dr. Basavaraj made a presentation particularly on viruses, which are very important biotic stress nowadays. I think both plants, animals, as well as human beings. The detection at a very lower cost, and that too without much waste of time, is very important. Right now we are passing through COVID era, isn't it? That's why I think he showed he developed and demonstrated a universal plant virus microarray. He also showed and validated using cucumber mosaic virus through quantification of probes as well as hierarchical clustering. Am I right, Dr. Basavaraj?
Yes,
yeah.
sir.
Yeah, and also he showed how that next generation sequence also could be used, because they are very sensitive and can be used for developing and detection quickly. And he demonstrated using soybean mosaic as well as pepper viruses. So I think they are very important in the present era, because we are scared of viruses, whether it is a virus of human being, animal, or plant. Man is not secured. Food security is not there unless we are secure. So I think well done. All
Sir, you are not audible. Mike.
Yeah.
Okay, now okay.
Any questions?
No, you were not audible. That's what I want.
Not audible.
Now okay, no problem.
Okay, I think I was just concluding Dr. Basura's presentation.
Yeah, it's a good presentation. No.
Okay, thank you very much. May I now
Thank you, sir.
invite Dr. Basava Prabhu L. Patil, the principal scientist at IIHR Bangalore, who will be making a presentation on mRNA-induced gene silencing for control of multiple pests and pathogens. To give a brief introduction of Dr. Patil, he is presently at IIHR and he obtained his PhD from University of Delhi in plant molecular biology, worked in a European Union funded program on Gemini viruses in collaboration with John Innes Center, UK. He is also recipient of DARD scholarship at the University of Stuttgart, Germany. He is also postdoctoral fellow at Donald Danforth Plant Science Center, Louis, USA. He joined as scientist at National Agri-Food Biotechnology Institute at Mohali, Punjab, and Jan joined ICAR Institute at National Research Center Plant Biotechnology, where he worked on sterility mosaic of pigeon pea. I remember I was one of the advisory committee member in one of the students' thesis on sterility mosaic of pigeon pea. He received the funding from ICR and DBT, and he is also the recipient of Indian Virological Society Award, also recipient of European University Brave Project. With this brief introduction, I would now call upon Dr. Basava Prabhu L. Patil to make his presentation.
Yeah, thank you, sir, for your kind introduction. I hope I am audible to all. Am I audible?
Yeah.
Yes, sir. Yes, sir.
Okay, I'm glad. Yeah. Hello, everybody. Thanks for this opportunity, Dr. Fakhruddin and his team from UHS Bagalkot. Although the title I gave was microRNA based technologies mix, but I thought it would be too much narrow. So, I have modified it as RNA based technologies for management of plant pests and pathogens. Here, I'll be emphasizing on virus control broadly. And I'll take you through, the journey I did in my research career, starting from my postdoctoral days till date, whatever different RNA-based technologies we have used for control of plant viruses and other pests and pathogens. So you are all familiar about plant viral diseases. Just now, Dr. Bhagyavadi explained very elaborately, about different virus families, genera, how they are to be diagnosed and all. So I'll not take much time. All these viral diseases are caused by several humongous amount of diversity is there. They are either have DNA, RNA in different shapes and forms as the genome of this virus. Coming to the small regulatory RNAs, until last few years, people were not aware about the important role played by these small regulatory RNAs. They were considered as junk RNAs. It was mostly the transcription factors or different promoters that were thought to be playing a major role in gene regulation. However, studies have shown that these have a lot of huge amount of important role in gene regulation. There are only 30,000 DNA units that control gene expression. However, there are more than 400,000 endogenous small RNAs that regulate our gene expression. So there are different kinds of small RNAs, starting from siRNA, the ones that were discovered first, small interfering RNAs. We have then microRNAs, RACRNAs, piRNAs, STACRNAs. There are, every year we have a new terminology. I'll not make it much complicated, depending on their size and the way they are generated in different pathways, maybe different RNA, RDRs are involved, or dicer-like proteins are involved in cutting them, trimming them into different sizes. So based on that, the different classes of small RNAs are generated, and they have different roles in manipulating the gene regulation. So viruses can also generate small RNAs. Here in the picture you can see how when a virus gets into the plant cell, it starts replicating, and during the course of replication it transcribes and produces a lot of defective transcripts, mRNA. Viruses have very strong promoters. That is why some of the virus promoters are used in generating transgenic plants, such as cauliflower mosaic virus promoter. And when these strong promoters, they express the transcripts, a lot of defective RNAs are produced, which triggers RNAi machinery. And through this cycle, these small RNAs are generated, and the viral RNAs are degraded into small RNA, and eventually the plant recovers from infection. This is model host plant for virus studies. This is Nicotiana benthamina. You can see here at the bottom, the plant is severely infected, but in due course of time, because of generation of small RNA, the plant has recovered. So this kind of phenomenon can be seen in field scenarios also, depending on the environmental conditions and the time and other factors. This is not something which we discovered first, but way back in 1928, this was reported in Journal of Agricultural Research, when tobacco plants were infected by tobacco ring spot virus. Eventually the plants recovered through gene silencing or RNA silencing. For a long time it was not known, the mechanism of recovery was not known until Dr. David Balcom discovered gene silencing in plants. So later on, it was revealed that such kind of recovery occurs because of gene silencing, since virus infection results in generation of small RNAs that target proliferation of the virus itself. So this itself has been used in control of RNA, control of several plant diseases. And here broadly I term them as RNA-based strategies, such as RNA interference or double-stranded RNA-based strategy, or short hairpin or artificial microRNAs. And the latest one is microRNA-induced gene silencing, which is different from artificial microRNA, and genome editing, which also employs subgenomic RNAs as the key molecules for regulating gene expression or for modifying the genome. So although in 1986 Dr. Beatty, he discovered, he was the one first to report transgenic control of viruses using core protein mediated resistance, but the basic mechanism of disease resistance was not studied. There was no foolproof evidence as to how these transgenic plants are able to resist virus infection. So there are also hypotheses that it could also be because of RNA, since in these transgenic plants, very strong promoter like 35S promoter is used. A lot of aberrant RNAs are formed that may trigger gene silencing. So you know about this example wherein this is transgenic papaya in Hawaii. This is one of the best standing example till date. More than a decade, it is still standing healthy in Hawaiian fields. It's not only for virus resistance, for any transgenic technology. This is the best example that we have today. So you can see here the transgenic plants are standing in the field healthy and sturdy, whereas the non-transgenic have almost fallen down because of virus infection. So, however, the first transgenic plants that was based on RNA interference technology was in beans against bean golden mosaic virus. So this was done in Brazil, and beans is a staple food. Like for us, chana dal or tur dal is a staple diet for protein. So for them, South Americans, beans is a staple food, and they cultivate beans extensively. So bean golden mosaic is a very serious problem there. So they developed transgenic, and this is almost going to be commercialized, and it has already passed several regulatory hurdles. So this was the first RNAi based transgenic technology that was used in the case of against virus. So after returning back to India, in collaboration with IIT Guwahati, we also developed similar kind of strategy to control mung bean yellow mosaic virus, another Gemini virus that infects several legumes, including cowpea. So we developed transgenic cowpea, which was resistant to mung bean yellow mosaic virus. You can see these are transgenic cowpea free from virus infection, whereas the non-transgenics or the wild ones are heavily they are showing yellow symptoms because of virus infection. So not only viruses, RNAi also has been used for control of insect pests. And there are a lot of examples wherein transgenic bollworm resistant cotton have been developed using RNAi, not only BT genes, but also using RNA interference technology. However, RNAi for insect control has not been very successful because insect pests are not very comfortable in taking up the small RNAs, and somehow they get degraded and they are not able to silence the insect genes very effectively. However, transient assays have shown that by feeding long double-stranded RNA, insects are able to take up those double-stranded RNAs quite well, and small RNAs are generated. So, chloroplast-based transgenics were developed, and using chloroplast targeting, reporter genes. Since chloroplast, they lack the DCLs, and the dsRNAs that are produced, they are not converted into small RNA, unlike in the cytoplasm. So the dsRNAs are taken up as such when the plant tissue is consumed by insect, and they are able to give good resistance. You can see here a non-transgenic which is almost eaten up by these insects, whereas the transgenics are quite healthy. So this has been used for nematode control also. There are several studies. I will not really get into the details. But here, I would like to highlight our own work, which I did in Danforth Plant Science Center in a virus-resistant cassava project that was for African countries. It was a collaborative multi-institutional project, and cassava is a staple food for Africans and for some of the South Indians. And there are a lot of delicacies and industrial products from this crop, which many people don't consider it as a valuable crop. And in the era of climate change, this is going to be very promising plant since it is highly drought tolerant. So this is field pictures of this crop in African countries. And this crop was, its center of origin is in South America. However, it was introduced in Africa and India and other Southeast Asian countries. Though these plants were introduced in different countries, but the viruses were not introduced. It was the local viruses that evolved and started infecting these plants. So the cassava infecting viruses in Africa are significantly different from the cassava infecting viruses from Indian subcontinent. So you can see here, this is tapioca in China, which is quite healthy. However, in Africa, it is severely affected by mosaic disease. And this disease is caused by Gemini viruses because of their geminate structure, which are transmitted by whiteflies. So, we developed a huge number of RNAi constructs, and since cassava is very hardy crop, difficult to transform, recalcitrant for transformation, so we had to identify only few constructs that were really efficient. So we developed a transient technology wherein, by agroinfiltration in Nicotia benthamina, we analyzed the efficiency of production of small RNA and their ability to resist virus infection. So here is the result. We tried targeting different genes, and only few genes like AC1 and AC2 gave good amount of small RNA, and they gave good amount of resistance against the virus. However, other genes like port protein or genes that were located in DNA B segment coding for movement protein did not give good resistance. So these selected constructs were mobilized to generate transgenic cassava plants, and transgenic cassava plants were produced, and the first experiments were done in greenhouses, and later on they were shifted to African countries for field trials. And this work was done on an industrial scale from the funding from Monsanto, USAID, and several other funding agencies. And this was done at a commercial scale involving a large number of people from our lab as well as from Africa. The field trials were conducted, and in the field trial, confined field trial, the transgenic cassava gave decent amount of resistance against these mosaic viruses, whereas the non-transgenic succumbed to virus infection. By the time we found a solution again to this virus, a new virus emerged, which is called as brown streak virus. In contrast to the Gemini viruses, which have DNA as their genome, this virus has RNA as its genome, encoding for almost 10 proteins, which is similar to papaya ring spot virus and belongs to the same family but different genus. So this is even more worse, and in mosaic infected plants we could get some yield, but here, although we got some yield, when you cut open those tuberous roots, you would find such rotten tissues in the roots which were not edible at all and difficult to market them. So this was very difficult, and you can see the intensity of damage caused by different virus isolates of this brown streak in different places in African continent. So samples were collected to study the biodiversity of this virus. Knowing the target virus is very important. In the previous talk, you have heard, how important it is to diagnose properly what are the viruses involved in a disease complex. So, we took up the biodiversity study, and we found that there are 2 different species of viruses which cause the brown streak disease. And eventually, in due course of time, there are several mixed infections which have made the situation even more complicated, and there is no source of resistance for this virus even in the wild host plants. So initially, we took up experiments RNAi-based control strategy in model host, and we got good amount of resistance, and amount of resistance was positively correlated with the amount of small RNA that was produced via the double-stranded RNA through the RNAi constructs. And interestingly, many of the plants which were expressing high level of small RNAs were resistant to cross-infection by viruses that were not homologous to the target virus. So you can see here, for example, this particular plant which produced high level of small RNA shows 100% resistance to one virus species, and the same transgenic plant with high level of small RNA is resistant to another species involved in brown streak disease. So it is very important to have very high expression of small RNA to get good amount of long-lasting resistance. So similarly, we used transgenic technologies and developed transgenics, and field trials were taken up in Uganda and Kenya. You can see the transgenic plants are free from the necrosis because of this virus, whereas the non-transgenic control are severely infected by brown streaks. And we looked for small RNA expression from the field samples, and all these plants were expressing good amount of small RNA, which is the proof of concept that, and it works well even in the field scenario. So multiple cycles were vegetative cropping cycles were taken up to look at the resistance, and it was stable trait, and multiple field trials were taken up, and it worked well in several different locations. So all right, that's about brown streak virus. However, there are instances wherein both brown streak virus, that is similar to a potty virus, as well as the DNA genome Gemini virus co-infect these plants. So it is important to have dual protection against a DNA virus as well as an RNA virus. You can see the intensity of disease because of the synergism between the brown streak and Gemini virus. And when it is replicated in the model host, you can see how badly these plants look, and they are almost dying. So we tried to develop combo construct. However, this did not work well because there are certain limitations in using the sizes in RNAi generation. In generation of small RNA, we cannot go beyond certain level. So we had to find alternate technologies to stack these RNAs so that they are of limited size and they are expressing well and are able to resist different viruses. And we checked for artificial microRNA technology, and microRNAs are quite different from small interfering RNAs. The pathways, although they are similar, but there are certain key differences which result in different kinds of small RNA formation. And so using this technology, we developed transgenics, and by targeting different genes, we found, but the resistance obtained was good in model host, but not in in cassava across different generations. So genome editing is another technology which is also being used for virus control, and you can directly target the viral genome, either viral DNA or viral RNA, or you can modify the eukaryotic translational initiation factor. In the morning, a professor from TNU delivered a talk on how eukaryotic translation initiation factors were modified to get resistance against rice tumor virus. So this has been employed for getting resistance in brown streak virus also. And you can see by editing certain genes, these EIF4 like genes, they were able to get resistance against this virus. And you can see in the transgenics, these are free from the necrotic lesions. So, all right, I'll not get into. You can even target viral RNA by using a different kind of Cas13 along with the CRISPR technology, and you can even accomplish control of RNA viruses such as papaya ring spot virus, or even any other viruses like tospo viruses infecting tomato or other crops. All right, that was about transgenic technologies, but in India, we don't have very fertile ground for carrying out field trials for transgenics. So we had to find out an alternate technology, which is probably non-transgenic, but involves latest technology like biotechnology to control the viruses. So a lot of work is being done by wherein double-stranded RNA are used as sprays, foliar sprays on the plants, which eventually result in triggering of RNA interference and thus control of plant diseases. So a lot of work is done for control of viruses, and these double-stranded RNAs is mixed with carrier materials such as clay nanosheets or many other carrier materials to ensure that they are stable on the plant leaf surface when they are sprayed, and they are not affected by harsh weather conditions like high temperature.
Hello? What happened? Dr. Patil?
I think connection is gone.
You're not audible, Dr. Patil.
Rashid Yaman is contact.
Yes, sir, we are trying to contact him, sir.
Okay.
Yes, sir, we are calling him, sir.
Yeah?
Sir, we are calling, we are connecting to Basaprabhu.
Fine, fine. Go ahead.
He has muted himself, I think. He is,
No, sir.
it is showing mute for the select.
Can you hear me now?
Yeah.
Yes, sir.
Jayashree madam, you can unmute him.
Sir, he is unmute only. Unmuted, sir.
Can you hear me now?
Yeah.
I'm sorry, there was power outage here and I lost internet connection. I'm using the internet from my mobile phone. Are my slides visible? Probably not. Are my slides visible?
Yes, sir. We are able to see.
Yeah. I'm sorry for the interruption. Yeah, we have used this double-stranded RNA technology against papaya ring spot virus, and in greenhouse experiments, we have shown the control of papaya ring spot virus using these double-stranded RNAs. But yeah, we need to demonstrate this in field trials by using a very good carrier material. And yeah, since after I joined ICR, I was working on sterility mosaic disease, which is a major problem in pigeon pea cultivating areas, which is transmitted by areophid mite. This is called an MRA virus. We have used double-stranded RNA against this virus in collaboration with the US Bangalore. We did field trials, open field trials for the control of this MRA virus using double-stranded RNA. And you can see, all those plants that were supplemented by double-stranded RNA as foliar spray, they were able to give good amount of protection, whereas the control plant showed severe infection by this MRA virus. So this work has been recently accepted in this journal, and it's going to be online very soon. And we are currently at IIHR. I am using this double-stranded RNA technology for control of anthracnose called by Coltotrichum gliosporidis. I heard yesterday one talk wherein it is very difficult to get source of resistance against anthracnose. I'm sure this dsRNA can give good protection. You can see here, using dsRNA, we are able to protect these capsicum fruits against anthracnose. What you see here white is a secondary infection by other fungi as the tissue starts rotting because of anthracnose. And all right, coming to microRNA induced gene silencing, this is entirely different technology, different from artificial microRNA or small interfering RNAs. This was in collaboration with the IRI and an industry partner, and with technology support from Dr. Dinesh Kumar from UC Davis. Funding was from DBT BIRAC. So this is entirely different because a microRNA which is of 22 nucleotide size called as MIR173 triggers generation of tacrRNA, that is trans-acting siRNAs, and that is very efficient, and there are no size restrictions in the length of sequence that you want to use as target sequence. So that is one very good advantage, because in RNA interference there are limitations in using different lengths. So this is something very important, wherein the biogenesis of tacrRNA takes place through some specialized microRNAs which are of 22 nucleotide size, in contrast to the routine 21 nucleotide sized microRNAs. So this is how it works. You put different genes here, and once the MIR173 expression is triggered, it triggers the generation of tacrRNAs, and the target genes are silenced. So here, in this case, we targeted several different genes from different pests that infect cotton, such as nematode, root knot nematode, cotton leaf curl virus, and cotton leaf hoppers, which are major problem. Of course, you have for heliothis control, you have BT, and we thought probably if this construct could be pyramided along with the BT, this would be very good, since minor pests are emerging quite significantly in BT transgenics. So we developed this construct, and initially, we did experiments in the model host plant, since cotton transformation and all that would take a lot of time, and we were at least successful in showing resistance against nematodes, and all the genes that were stacked were expressing perfectly well. This is the expression level of different genes that target different pests of cotton, and you can see here they are efficient in controlling. This is the work done by the IRI collaborator, Dr. Umar Rao. and they are show very low galling intensity in the transgenic plants, and the number of galls, egg mass, eggs per egg mass, and the multiplication factor are very low compared to the wild type tobacco plants. So it's the proof of concept that microRNA induced gene silencing is a very good technology, can be used against multiple pests, and has the ability to pyramid several genes to target multiple genes or multiple pests. So you can see the female nematodes have grown up well in the wild type, where non-transgenics, whereas in the transgenics they are not able to grow well. So this is all the work done by my collaborator. Since I'm not a nematologist, I'll not really emphasize much. That's all I wanted to share. And this is a book which I recently edited. Somebody is interested can look into this. Thanks for this opportunity. Thank you, Professor Nayak, Vice Chancellor, UAHS Shimoga, and Co-Chairman, Dr. Kulkarni, for giving a nice introduction and time to speak. Thank you, UAHS Bagalkot.
Yes, Dr. Basuraj, well done. Very interesting and informative presentation. Are there any quick questions to Dr. Baso Prabhu? Quick observations, one or two. Nobody is asking any question in this session.
This is the last talk, last invited talk. So,
sir, can you hear me, sir? I have one question, sir.
Yeah.
Sir, you are mentioning about papaya ring spot virus, right?
Mm-hmm.
Whether the technology is commercially available and viable for field conditions, sir?
Using dsRNA?
Yes, sir.
Yeah, see, dsRNA, we have shown it on laboratory scale, but for field, we need to come up with a very good carrier molecule. Without that, that may involve people from other expertise like nanotechnology or others, wherein they can develop good carrier. We have similar problem like Moderna has come up with a very good vaccine wherein mRNA is used, but they need freezers of -70 or -80 degrees centigrade, which are not available in many developing countries. And they are looking for a carrier molecule which can keep them stable. So similar is the situation here. These dsRNAs may not be stable in field scenario. So we need to identify a carrier molecule, and that really needs a lot of work, and it's going to take years. What we have shown is only proof of concept.
Other than papaya,
thank you very much.
Hello.
Yes, please. Yeah.
Sir, other than papaya, is there any other crops commercially using by this miRNA?
No, as of now, none of these miRNA or dsRNA technologies are commercialized. They are still, being researched, and probably some of the industries are also working, but they are not revealing. Maybe one fine day you may get to know that it is commercially available. People are working in Australia, as well as Bayer Crop Science is also probably working on this technology.
Okay, thank you.
Dr. Basava Prabhu.
Yes, sir. Yeah.
Yeah, although very interesting presentation, but I have a small practical question. With so much of viruses being reported on every crop nowadays, take for example, you highlighted capsicum, chili, right? There are around 25 viruses.
Yes,
so,
sir. Yeah.
Technologies like you mentioned, RNAi, dsRNA, short hairpin, microRNA, good examples you have given in through bean golden mosaic virus, then mung bean yellow mosaic virus. But specifically, when you talk several viruses coming on a single host, how you could answer this? How you address this problem?
Yeah, that's why, you know, the last technology like microRNA induced gene silencing, wherein say there are 10 viruses infecting tomato, we'll have to pick up only the economically most important viruses, although there are more than 100 viruses infecting tomato. Say there are 10 and you can probably put them together, all these one, and this is Stasi RNA based technologies, which is whose expression is triggered by a specific microRNA, and probably use of such technologies may help in overcoming problems by multiple viruses. So I'm sure in chili or capsicum, such technology could be employed, and yeah, we can generate multiple virus resistance.
How is that you mentioned an anthracnose on capsicum managing? Will it be really showing in the field?
No, sir. What we have done is only in the laboratory scale, because you know, it's going to take a good amount of time to demonstrate this in field scenario, because if it rains or whatever, the dsRNA applied can be washed off. So we have to use a good binding material which can really stick to the foliage or the leaf tissue, any plant tissue pretty well, and cannot be washed off so easily, or if there is high heat, it cannot be degraded. So a lot of research and advancements are to be done in other areas also, which can support in employing this technology for virus control. Say we have shown it using one dsRNA. Probably you can mix up such 10 different dsRNAs against different diseases, say against 4 or 5 viruses, 4 or 5 fungal pathogens, and prepare a nice cocktail which probably can give resistance to multiple pathogens.
Yeah, thank you.
Sir, sir, we have lot many poly houses where people are growing capsicum. So what is your thought in that line, sir? Because you said if you grow in open condition, it may wash.
It could, yeah, it would be a very good idea, yes, especially at least in the polyhouse situation, wherein high-end vegetable production is done in polyhouses or greenhouses. Yeah, this technology could be used. Yeah, if we want to really take it to polyhouse, probably industry academia collaboration through funding from Biotech or that sort of thing can be planned. If you are from an industry or whatever, maybe we can make collaborative proposals.
Thank you, sir. Yeah, thank you very much.
That's the last question. I believe we have taken a lot of time. Thank you profusely, Dr. Basava Prabhu, for the very informative presentation. Only thing I hope the best way is that you mentioned virus-resistant cassava for Africa, and probably cassava brown stick disease. If they are working, they are going to be really useful. And the last concept that he mentioned was on multiple pest management through MIRN. That is going to be probably a good hope if it is really taken in future. Thank you for well done. Thank you very much. Let us give him a big hand. Thank you very much.
Yeah, thank you, sir. Thank you all for your patience.
Yeah. So that may I now invite Dr. Ajamada Kushalappa. Is he there? Dr. Kushalappa.
Yes.
Yeah. I welcome you, Dr. Kushalappa, professor at McGill University, Canada. Are you ready?
Yeah.
Yeah. A brief introduction of Professor Kushalappa. He is a PhD from University of Florida, and he was a professor at Titular Visitane at Brazil, and he's a recipient of CPS sponsored Dr. and Mrs. Bailey Award for his very exceptional and distinguished contribution in plant pathology. He was also invited speaker probably more than 16 number of times in national and international conferences dealing about plant biotic stress resistant genes and genome editing. He is currently interested in identification of plant biotic stress resistance genes through forward and reverse genetics, that is using systems biology. And then he has also been awarded quite a good many awards. He is also actively associated with Natural Science and Engineering Research Council, where he is a member for funding panel, and also associate editor of BMC Plant Pathology, currently guiding several graduate students and visiting scientists. I have heard of him when I was a postdoctoral fellow at ICRISAT. Dr. T.N. Raju used to tell about you. Now I have time to know we have an opportunity to listen to you. Dr. Kushalappa, please.
Thank you very much, Dr. Naik, and I hope he told you only good things about me. Okay, and also I take this opportunity to thank Dr. Kulkarni too. Okay, I had a good visit to Bagalkot and all. And at the outset, I would like to thank Dr. Fakhruddin and all the team who invited me to give this keynote speech for this theme. Since it's a thematic talk, I would like to cover a little more general grounds than specific areas. Today, I would like to talk about second green revolution, mainly based on gene editing to enhance multiple disease resistance. Okay. We have been domesticating plants for the last 10,000 years, and these nomadic people move from place to place, domesticating both animals and plants. And during these years, they selected excellent phenotypes mainly for yield and also for disease and pest resistance or general agronomic conditions. These became land races. Now, in nature, we have both self-fertilization and also cross-fertilization. This cross-fertilization leads to hybrid vigor, and the offspring can be more high-yielding or agronomically good traits than the parents. And this was used first to produce green revolution. Produce the hybrids, which led to the first green revolution. And they were able to produce dwarf plants, and which reduced the lodging and enabled mechanization, mechanical harvest. And also they selected mainly the varieties or the lines with the high yielding. These led to not only short dwarf plants, but also high yielding plants. And increased mechanization, increased disease resistance, diseases and pests. And to manage these diseases and pests, they applied a lot of pesticides. These pesticides led to environmental degradation. And increased fertilizer application led to degradation of soil. Well, if you look at nature, then the plants grow in nature, and they absorb minerals and organic matter from soil, take carbon dioxide from the atmosphere, and they biosynthesize carbohydrates and proteins, and also release oxygen. These are taken up by human beings and also other animals and microbes, and when they die, it gets incorporated into the soil. So it's a natural balance, and if we disrupt the balance, and a lot of these biological activities are not going to happen. And this is what the green revolution is causing in the agroecosystem. So this was realized, and a second green revolution was proposed. This was mainly to promote or mitigate problems associated with the first green revolution. It was first proposed in 2012 by the President of India, Pranab Mukherjee. And the intention was mainly to reduce soil degradation due to increased application of fertilizers, and environmental impact due to pesticides, and also hybrid seeds and equipment, which are expensive, where poor farmers cannot afford to grow. So they started molecular marker-assisted breeding, where markers are nothing but segments of chromosome linked to traits, and it was very good in pooling markers. Based on markers, they were able to pool complex traits and produce hundreds of these cultivars, which are agronomically good. And what are the limitations? They obtain disease resistance increase, but not satisfactory. And also it takes more than 10 years to breed one cultivar. And these markers or quantitative trait loci are now often not associated with traits, because most of these complex traits are controlled by hierarchies of genes, especially plant disease resistance. Good. Now, the second option, would be for us would be genome editing to improve plants. Okay. Today, I'm going to genome editing can be, as you have heard so far, in this e-conference, genome editing can be used to enhance, improve so many traits. Here today, I'm going to talk about mainly enhancement of multiple disease resistance. So genome editing is mainly a precise modification of genes. In the marker-resistant breeding, hundreds of cultivars have already been produced. Why to produce more? And these are good in certain yield traits and others where they are focused, but for plant disease resistance, generally these are quite poor. And these are poor because the genetic composition for resistance is poor. And we can increase this based on genome editing. Now, these are where the genes are located. Okay, these are plants and the cells and the nucleus and the chromosomes, and these are the double-stranded DNA helix, and with these ladders are nothing but the base pairs ATGC that we sequence, and the genes determine the phenotype. Now, where did the genes come from? Okay. Now, about 13 billion years ago, there was a big bang, and the cosmic rays joined together and produced protons and neutrons, which were enclosed in a nucleus, and electrons orbit around this nucleus. These atoms, several of these atoms join together and produce different planets. And also, later, about 3.7 billion years ago, the prokaryotic cell was evolved. And further down, and more complex cells with different organelles were evolved, and they are eukaryotic cells. And if you look at the genome, today we have genome sequence. This is the double helix, which is made up of pentose sugars, and they are linked by phosphoric acid. And these are connected to the ATGC, these nucleotides, these ladders. So all these things, whatever we have here, are nothing but atoms. So the genes evolved from atoms, and all the living organisms evolved from these genes. Now we have the whole genome sequencing, and, where we have ATGC, and we can identify the mutations, and these mutations lead to change in the phenotype. Now, how do these genes function? The DNA produces, transcribes into RNA, RNA translates into proteins and enzymes, and these enzymes catalyze reactions. They biosynthesize from glucose more than 200,000 metabolites. Okay, and these metabolites and proteins, make the plant, different organelles in the plant. Okay, now let us see how the plants defend against the pathogens. Both pathogens and hosts, they have evolved over years. And there is a balance between the host and the pathogen. So if you look at the mechanism, and there are mainly apparent resistance, which is mainly environmental control, and the true resistance is mainly biochemicals are controlled by genes. The resistance is mainly due to constitutive and induced by metabolites and structures, followed by, these are induced following pathogen invasion. And these are induced structures and biochemicals. Biochemicals are nothing but metabolites and proteins. And these metabolites and proteins are produced by genes. That is the global link, how resistance is controlled to genes and metabolites. Now, how to identify resistance to metabolites and genes? And several people, including researchers, including our own lab, did forward and reverse genetics to identify these things. We took plants with the contrasting levels of disease severity, and then did metabolomics, then related to metabolic pathway, and to identify the resistant proteins and resistant metabolites. And I mean, we identified resistant proteins and metabolites and related to metabolic pathway to identify the respective genes. When the genes are known, we did reverse genetics to silence this gene and produce the phenotype. Okay. Based on this is a metabolic pathway. Here is the glucose and the acetic acid cycle. It produces sugar, terpenes, fatty acids, alkaloids, and phenylpropanoids. These are the different metabolic pathways, and in these, we identified hundreds of the induced resistance-related metabolites following pathogen invasion, and these are called phytoalexins. Okay, now these, you have to produce a single metabolite. Okay, you need hierarchies of genes. Okay. We have biotrophs, necrotrophs, hemibiotrophs, and necrotrophs. They produce elicitors and effectors. These are recognized by membrane, host membrane located localized receptors, and they induce MAP kinase and also phytohormones. And these MAP kinase regulate the transcription factors. These transcription factors regulate downstream metabolite biosynthetic genes and also the protein, the code for protein genes. Okay. And these produce proteins and metabolites. These proteins and metabolites, they are deposited around the infection site to reinforce the cell wall. For example, if you look here, these are the phenylpropanoid pathway, flavonoid, fatty acid, and alkaloid pathways. The monomers of these are constitutive. Okay, they are combined with the glucose and they are phytoanticipins. They also can kill many pathogens. And most of the advanced pathogens, and the following pathogen invasion, more complex metabolites are produced. These are the from all different metabolic pathways, and they are phytoalexins. These phytoalexins polymerize, produce more complex metabolites, and also they conjugate with a different metabolic pathway to produce hundreds of conjugated metabolites, which are deposited around the cell, and most pathogens cannot produce enzymes to break down these. If these cells around the infected pathogen is contained, reinforce the cell wall, and the pathogen is contained to a small spot instead of a large lesion or a blight. We worked mainly on Fusarium head blight of wheat and potato. And in Fusarium, what happened? The host, this is the phenylalanine, the phenylpropanoid pathway. Phenylalanine generally produces lignin. When the pathogen comes, if it has these thioesters, the phenyl Querel-CoA, Cafel-CoA, and Ferrol-CoA. They take these, it combines with the amines, and they produce hydroxycinnamic acid amides. There are so many of them, and they are deposited to reinforce the cell wall. That way the pathogen is contained. Some of the higher forms, here in the potato, we identified co-compt, which produced the ferroil, which is diverted to more of lignin and related compounds. In wheat, we found lacase 4, which gives the race's resistance, because the pathogen Fusarium infects the florets and then travels through race's to other florets to infect other florets. And we found lacase 4, which produces lignin. And this is regulated by NAC32. So these are the different metabolites. This is the vascular bundles. This is the xylem vessels, the phloem vessels, and this RP is the resistant pathogen inoculated, and this is the resistant mark, and these are the susceptible ones. And this is increase in hydroxycinnamic acid amides following pathogen invasion. And here, similarly, the flavonoid combine with the glucose, glycosides, and they also are deposited around the cell to contain the pathogen. Okay. Now, how to enhance multiple disease resistance in cultivars? Okay, based on genome editing. So, as I told, we have already produced hundreds of cultivars for each crop, and these cultivars also have resistant genes similar to resistant cultivars or resistant wild plants, but some are mutated. And these mutated ones, genes, they disable the plant to biosynthesize these metabolites. Now, if you can cut and replace that mutated segment, it can gain the ability to produce this enzyme, which can catalyze and produce this metabolite, and thus the resistance. Now, what causes this genetic variability? Okay. Number one is sexual reproduction. When breeders cross two plants, hundreds of mutations are incorporated into the future selections, okay, because of chromosome, meiotic division. And then second one is in nature, genetic engineering occurs in nature. Several bacteria and also viruses take segments of these genes and transfer it to plants. And also, we have solar radiation and also chemical radiation and several other ways induce indels or insertions and deletions, and also base pair switch. Okay, this is the base pair switch we identified in co-compt in Pareto, phenylpropanol pathway gene. It is by mutation, it is converted, G was converted to T. Now, how to identify how to identify these mutated genes? Okay, we can do whole genome sequencing, QTL sequencing, individual gene sequencing, or RNA sequencing, and we can, based on the molecular functions, we can identify the detrimental mutations. And these candidate genes for genome editing, and the functions are, these are regulatory genes like immune receptor genes, MAP kinase, transcription factor, phytohormones, and microRNAs, and also metabolite biosynthetic genes and protein coding genes. These are the mutations in our cultivar. And the functional segments can be obtained from either resistant cultivars or land races or germplasm. And genome editing, there are several ways, and then I'm going to discuss mainly two, and this is the CRISPR-Cas9 system with the Gemini virus replicants. We have 3 modules. We can combine these, put in agrobacterium, and then inject it into embryo or stem to transform. Here what happened, this is our gene, this is a Cas9 and guide RNA. We select guide RNA, which is specific to make a specific cut in the gene where we want. And this causes a double-stranded break. And if it is this, the plant repairs it, non-homologous end joining, leading to silence. On the other hand, if you replace or you provide a donor template with homology arms, it will be replaced. So these are the 2 major ways. And also we can use base editing. Base editing is you use cytidine deaminase and adenosine deaminase, which replaces, switches CG to TA and AT to GC. And one of the advantages of this is this does not require double-stranded breaks. And how do you confirm after whether you have edited the gene or not? We can design a PCR and detect based on the PCR. Or we can sequence, these are the nucleotide, color-coded nucleotide sequence, and we can identify which nucleotide, whether it is replaced or not. For example, here in COCOMS, we change the T to G. And how to confirm the disease resistance? We can take to inoculate the non-edited plant and edited plant and produce disease severity. And also we can do the determine the pathogen biomass in the disease area. Okay, and this is the edited plant biomass significantly reduced, and this is the susceptible one. So this is the phenylpropanide, and we can prove the mechanism too based on metabolomics. Okay, this is the caffeine-CoA is converted into ferrule-CoA. That's what this CoComp, functional CoComp does, and this converts into monolignol, and it can lead to the biosynthesis of several downstream metabolites if those genes are functional, and this leads to suberin, wax layer, and cuticle. Okay, so now can we put this plant in the field? In Canada, no. Okay, they have, in Canada, we regulate based on plants with the novel traits. No novel genes, no novel proteins, no novel metabolites, then it is accepted. We generally, the cisgenic or the transfer of genes between sexually compatible plants similar to breeding is allowed, but others may be difficult. And it has to be transgene free. And no foreign DNA, we have to screen that. And also food and feed has to be tested. For example, how to screen out these transgenes? For example, in the case of clonally propagated plants, we can, the base editing may be better. Here, what happens on all these CRISPR-Cas or others, the agrobacterium backbone integrated can be screened out in the T0 generation. Okay, so because in the clonally propagated plants, it's difficult to produce segregating populations. If you can produce seeds, then we can use CRISPR-Cas with a higher potential, and we can use either T0 or T1 generator to screen out the mutations, or screen out the transgenes, or still not possible, we can back cross it to the original and select the segregating populations. Okay, now what are the challenges in genome editing? Number one is ploidy levels. To start with, it's better to select diploids because tetraploids and hexaploids are very complex. You have more genome to edit. And off targets is one of the concerns, but by careful or cautious selection of guide RNA using several software, it could be significantly reduced. And other one is callus production. It is kind of some plants are recalcitrant and it may be challenging, but more in-planta methods are coming up. Okay, so now how to register this gene-edited cultivar? These cultivars are already suited to local regions. They have been tried. They are being commercially produced. So all we have to produce is if they have more resistance. And then plants with novel traits, you have to meet this requirement. And regulatory agencies also require that you can identify your plant or cultivar based on gene edited, you have edited several genes, you can develop PCR. Ownership, if you don't know, if it's a new one, then you may have to, if it's already patented, you have to get permission. And the cultivar registration, each country, we have different mechanism to register. In Canada, it is through Variety Registration Office. Well, that's overall what I want to speak. If you have any, these are my graduate students and postdocs, and some of the previous people also worked, and some of these students are also working in India. So, and I would like to thank all the funding agencies, and I thank you all for attending. If you have any questions, I'll be very happy to answer.
Thank you, Professor Kushalappa, for a wonderful presentation on a thematic area, second green revolution, gene editing to enhance the multiple diseases, starting from, the background of the first green revolution, what our late President Pranab Mukherjee mentioned in his talk. Then he also mentioned about marker-assisted breeding. Then he narrated the genome editing. Before that, he also gave a origin of genes, origin of cells, genes, and how the plant started defending, how to identify the resistant metabolites and R genes, also reverse and forward genetics, how to make them silence, and RNA genes, R genes identified based on certain important tools. And he was more concerned about how to enhance the multiple disease resistance, especially through gene editing. And he also, the course, he mentioned the variability, how we can bring the variability on. So particularly CRISPR-Cas9, which is, and what are the challenges, particularly in genome editing. We gave a good account of how we can move about for the next green revolution using all these tools, particularly gene editing. If there are any quick observations, one or two questions for Professor Kushalappa, please.
Sir, can I ask you a question, please?
Please go ahead.
Yeah, Professor Kushalappa, sir, it was an excellent presentation. You delved deeper and deeper into evolution. So actually, if you recollect HH-Flask hypothesis, for every susceptible resistant gene in pathogen, there is a corresponding susceptible gene in host. What is your say on this at molecular level? Did he say this because they are co-evolving, or is it because the evolution of pathogens are dependent on evolution of host?
Well, that's a very good question. Okay, especially for biotrophs, they have identified several the receptor R genes. Okay, before all the R genes were receptor, but now we consider all the genes involved are resistant genes. These receptor R genes, they perceive the pathogen, and they are rather surveillance genes. They are not really the resistant genes. This is the HH4 hypothesis. Hypothesis is still correct, but it is only for recognition, not really for resistance. There are some exceptions and all people are proving, but in general, if you take, there are the resistance is mainly due to downstream genes, either the metabolites or the proteins. And I have covered mainly to answer your question specifically, in Pareto, they have pyramided a few receptor R genes. First year it was very resistant, then after that they broke down. Because these break down quite fast unless they are associated with the downstream genes. So the downstream genes, the metabolites, are not specific to races, not specific to pathogens. That's why they can give multiple disease resistance. It depends on the enzyme they can produce. Hope I answered your questions.
Yes, sir. Thank you very much.
Yeah. Any more questions? Well, if there are no observations, we will profusely thank Professor Kushalappa for his excellent presentation, making it very clear. Thank you very much, sir. Thank you.
Thank you all. And hope you all will keep safe.
Yeah.
I am also at home. We are not allowed to go to office. So I'm glad all the things worked. Yesterday we had the first snow, and I thought the electricity would go, and I would be in big trouble. Okay, so everything went very well. Thank you and good luck.
Thank you, Professor Kushalappa. You made it up so early in the morning for us. Thanks a lot.
Thank you.
Thank you so much.
Yeah.
Dr. Fakhruddin, you want to announce something?
No, sir. Please go ahead, sir.
Yeah. Now we have oral presentations. There are 7 speakers. Each of them have been given 10 minutes time. I would now call upon the first speaker, Dr. Sharda Chaudhary, to make a presentation on stem gall disease resistance associated genic SSR functional domain markers in coriander and its cross-species transferability. Dr. Sharda, please.
Hold on a second.
Kushalappa sir, kindly unshare your PPT, sir.
Dr. Kushalappa, please. Professor Kushalappa, please. If you can unshare your PPT.
Hello, sir. This is Dr. Sharda.
Yeah.
Okay.
Please go ahead.
It is okay. I deleted my. How do I do?
Yes, sir. Yes, sir. It is done, sir.
Done.
Okay. Yeah.
Thank you, sir.
Thank you.
Sharda, madam.
Yes. Yeah, ma'am.
Can you share your slides, madam?
Is it visible, ma'am? Hello?
Sir, yes, madam, it's uploading.
Okay.
Madam, we are not able to see, madam. Any problem in?
From here, it's still showing sharing.
Madam.
Still sharing.
Is this network is weak? Your network?
Maybe. But it's taking time.
Shall we go for the presentation?
It's okay, ma'am.
Yeah.
Is it visible?
Yes, madam. We got it, madam. Thank you.
Okay, thank you. Good afternoon, all. Shall I start?
Yeah.
Yeah. Good afternoon, all. This is Dr. Sharda Chaudhary, Scientist Biotechnology from NRC on Seed Spices, Tabiji, Ajmer. First of all, I want to thank all the organizers for providing me this opportunity to present my research work at this platform. As you can see, the title of my research is Stem Gall Disease Resistance Associated Gene SSR Functional Domain Markers in Coriander and its cross species transferability. As we all know that coriander is a popular seed spice crop cultivated worldwide. This crop suffers mainly from the stem gall disease and causes very huge loss in quality as well as the yield of the seeds and the quality of the seeds. The stem gall disease caused by the Protomyces macrosporus species co-dominant. So what we need to there are many options which we are okay. There are many approaches which we are applying to control these diseases. Some of them are by using the chemical methods which are not environmental friendly. So here we can go for the molecular markers and identify the disease at very initial stage. We are having very less reviews available. Please, yes. What we need today are the co-dominant genetic markers to investigate the disease in coriander based on the functional annotation, information, and domain contained in the SSRs obtained by bioinformatics analysis are termed as the SSR functional domain markers. The term was given by you et al. In 2010. So what strategy we have applied here in our coriander crop? First of all, we have synthesized the RNA sequencing, and then assembled data was used to identify the transcripts. And out of these transcripts, we have identified the functional annotation and the functional conserved domains. And based on the literature available on different sites, we have identified our functional domain markers as SSRs. After getting the forward and reverse primers, we have synthesized these SSR primers and screened the available 10 the most utilized varieties of the coriander as well as the germplasm, which are very dominating in particularly the Rajasthan area. And even like we are having a scarcity of the SSR markers in APSA family. So here we have tried these markers in phenol genotypes also. The material and methods applied here. The in-house transcriptome data was utilized for the study, which was generated 2 years back by us. 10 coriander accessions. CS6, which is the most susceptible variety for the stem gall. Pantharitama, AGCR1, Hisar Sugan, Hisar Surbi, ACR1. ACR1 is the most resistant variety for the stem gall disease. Hisar Anand and the Rajendra Swati and other varieties. And 5 fennel accessions collected from the germplasm unit of NRCSS were used for the validation of the FDM SSR's primers pairs. SSRs were detected from assembled transcripts using microsatellite identification, MISA tool. The link is given here in the slide. Functional annotation was performed using the BLAST2 Go tool. Functional domain marker, SSR identification and designing of the primers. Functional domain of the selected genes were predicted using NCBI conserved domain database tool, and SSR primers were designed using the sequences with SSR by using publicly available web-based web tools. And the parameters which we have applied here, the length of the primer should be between 18 to 24 base pairs, and annealing temperature was between 57 to 65, and the optimum GC content was approximately 50%, and the product size was 100 to 300 base pairs. Total 10 primers were synthesized, and the synthesis was done by GCC Biotech Private Limited. And validation of FDM SSR markers. The collected fresh leaves were grounded into fine powder. The leaves were collected from the fields where we have grown 25 varieties. Out of them, we have selected the 10 varieties. The CTAB method by Doyle and Doyle 1990 was used to isolate the DNA. The cluster analysis and transferability in fennel was checked. The successfully amplified SSR products were scored manually. For the presence, we have marked as 1, and for the absence, it was designated as 0. Genetic similarity matrix was obtained, and cluster analysis was performed using antisys software and by selecting Jacquard's coefficient and UPGMA with the bootstrap value of 1000. Here is the table, the total SSRs which were generated by the susceptible and resistant, the CS6 and the ACR1 varieties by using transcriptome. The statistics of SSRs identified in transcriptome are like this. The total size of examined sequences base pair in resistance, it was approx 8 crore, and in susceptible, it's 7. And here we can see we have received dinucleotide repeats, trinucleotides, tetra, penta, and hexa. The total detail is like as in table. The function. Hello?
Yes, madam. Madam, there was disturbance. Sorry for inconvenience. Please go ahead, madam. Yes, madam.
Functional annotation of resistance excision. These are the cellular component, molecular function, and biological process of the resistant excision, that is ACR1. And here is the functional annotation of CS6 variety showing their cellular component, molecular function, and the biological process. Here are the number of genes and percentage of the genes. Now, from the generated data, we have designed the 10 primers, but out of 10, only 9 primers generated the polymorphism. The last primer was not showing any polymorphism, and it was that's why I have not included that here in the list. The primers were named as Ajmer, coriander, stem gall disease 1 and 1 to 9. The genes which are used here are like this, and the primer, forward primer, reverse primers, and the generated primers were submitted to NCBI, and the NCBI IDs are like this. The gene names and the functions, the identified genes were searched on the already available literature, and we have found that all the genes are showing different level of stress resistance, abiotic resistant, biotic resistant in different crops, though it is not available in any horticulture crops, especially in seed spices. So this is the first study when we have used the specific genes, the specific SSR markers in coriander crop, and we have tried to transfer these SSR markers in the same the crops from the same family. Here are the examples of mostly these are from the rice and the other advanced crops. So it is the peptidyl prolyl cis trans isomerase CYP19-4 gene. Here we can say it is showing resistance in rice for showing the overexpression to tolerate the cold stress. And same like here we can see the UTP glucose 1-phosphate uridylyl transferase gene is showing this expression in these different crops. I have shown here the only 5 genes looking to the constraints of time. So here is the represented genic SSR markers with functional domain. Identified here we have identified the domain associated with the genes which are functional. Identified genes revealed their functional domain, which were submitted to NCBI. Here we have validated all the identified 9 genes in the coriander and the fennel excisions. The 4 gel picks which are shown with the green circle are showing the transferability from coriander to fennel. These genes are equally amplified in the fennel germplasm lines also, but these 2 primers were not showing the transferability with the fennel. They are specific to coriander only. And here the first primer, Ajmer coriander S1-1, here we can see it is showing the clear-cut absence of the marker in the most susceptible variety, that is CS6, and showing very good bend in the most resistant variety ACR1. So we can say that this is the most promising marker which can be used for the molecular characterization of the coriander germplasm, especially for the stem gall disease. On the basis of visible alleles, the dendrogram was constructed where genetic similarity metrics range from the 0.22 to 0.86, and 15 cultivars were divided into 2 major clusters. Cluster 1 is having 10 excisions of coriander, and cluster 2 is having 5 excisions of fennel. The result summary is like this. In the experiment, 9 microsatellites generated scoreable and polymorphic PCR product for coriander cultivars. The primer Ajmer coriander stem gall disease 1, a genic primer, SSR primer, and showed a good amplification in coriander, as well as this primer showed the 100% transferability with the fennel cultivars also. The primer showed amplification in all coriander varieties, excluding the variety CS6, which is considered as most susceptible variety of coriander for stem gall disease. The experiment was conducted in the field, and very visibly we can see that we were able to see that the CS6 was not able to survive when we inoculated, and the ACR1 survived very well. These other cultivars like Panth Haritima, Sar Subhi, Sar Anand, CO2, Rajendra Swati are homozygous and showing overlapping alleles, though they are having resistance, but not that that much resistance like in ACR1, which appears as single allele in agarose gel. The most resistant cultivar ACR1 is heterozygous, having 2 alleles for the primer 1, whereas all others were having homozygous primers. This primer can be considered as a good primer to identify the varieties for presence or absence of the stem gall disease in coriander. Primer pair is A coriander SGD1-2 and SGD3 showed monomorphic allele with different size in selected cultivars of coriander, but showed the 100% transferability in fennel. And the resistant cultivar ACR1 is showing the brightest allele intensity. Same intensity is noticed in cultivar Panth Haritima, Sar Anand, CO2, and Rajendra Swati. Primer 2 and 3 are showing same allele pattern in fennel cultivar and can be proved as good primer for screening of fennel cultivars too for the disease. It is noticeable that these SSR FDM might be linked to genes involved in controlling defense mechanism in coriander against stem gall disease, and it can be powerful tool for genetic mapping and marker assisted selection. Our advanced and interdisciplinary approach adopted in the study could be useful in speed up the molecular disease resistant breeding in coriander and other seed spices crops. Conclusion, in the experiment, 9 microsatellite generated scorable and polymorphic PCR products for coriander cultivars, a large data of FDM with a total of 116994 consensus sequences were generated using the RNA-seq method, aiming to provide the markers for deciphering the disease resistance defense mechanism in coriander. The coriander FDM SSR showed a high level of polymorphism and 50% transferability in related species. Therefore, these FDM SSRs will be a powerful resource for further studies such as taxonomy, QTL, molecular breeding, genetics, genetic diversity analysis, and defense mechanism against biotic stress in coriander and other members of Apiaceae family. Here I want to thank Dr. Mahantesha B.N. Naika from University of Horticulture Sciences, Bagalkot, Karnataka, for providing his valuable time and helping in research and doing the bioinformatic analysis of the given experiment. And I'm also thankful to our director for providing me the opportunity to conduct the project.
Okay, madam.
Thank you.
Thank you for your presentation. One quick question, if there is a time. Quick question, if there is anybody. Yes, she has identified SSR marker associated with stem gall resistance. Probably the primer is also useful in identifying the presence or absence of resistant gene and will be able to help assist in marker resistant breeding, I think. Okay. Thank you, madam.
Thank you, sir.
Thank you very much. If there are no questions, I will now call upon Dr. Raghu. Raghu S. will be making a presentation on synergistic effects of plant defense elicitors and Trichoderma harzianum on enhanced induction of antioxidant defense system in rice against sheath blight caused by Rhizoctonia solani. Dr. Raghu, please, you are given 10 minutes. Please note it.
Yeah, please let them adhere to the timings, please. Yes.
Yeah.
Sir, is my slides are visible, sir?
Yeah.
Okay, sir. Good evening, sir. Nayak sir and Kulkarni sir. And I thank Dr. Fakhruddin sir for giving me an opportunity to present my topic here. Sir, as we know, rice, it is called as a global cereal because India is most important. In India, rice is most important food crop, has been grown for more than 6,000 years. And it is a stable food crop for around 70% of the Indian population, and it holds the key for food security, and it is the source of livelihood for the millions. And we all know that rice is our culture and it is also our heritage. Coming to the Indian scenario of rice, and rice, it is growing in an area of 130 million hectares and producing about 170 million tons of paddy. And we are also producing 425 million tons of residues. And rice, it is being grown around 67 million farm families, and it is feeding around 80 crores of Indian families. And we harvest from around 43 million hectares every year, and annual value of around $53 billion will be coming from rice. So apart from this, rice is the production and productivity of rice is facing a lot of problems, especially the diseases. If you see here in 1945, only the brown spot disease was the predominant one, but as the time changes in 2017, there are around 13 diseases of rice which are of economic importance. Among them, sheath blight disease is one of the most important and primary disease of rice, which is causing huge economic losses in terms of yield and also the quality. Coming to the symptoms of this rice sheath blight, sir, and initially it will start as small lesions, and later these lesions will start coalescing each other. The symptoms can be seen both on the leaves and also on the stem, leading to the whole plant may die. The symptoms will typically look like this snakes, that is Brussels viper. To manage this disease now, it is very difficult as we are not having that much a stable resistant source in any of the cultivars, even in the wild rice, accessions and also the cultivated species. So only the chemical management is the commonly practiced management measure, especially the spraying of validamycin 3 at the rate of 4 ml per liter, or hexaconazole and triflutamide, and are also carbondazim is commonly practiced. Biocontrol agents are also being promised, but they are not long lasting. So in such cases, what we can do, can we combine some of the plant disease, that is chemical elicitors, along with biocontrol agents to manage this disease effectively? So with this, we all know that salicylic acid, chitosan, and they're all known inducers of systemic resistance and influence the plant defense responses, including reprogramming of the cellular mechanisms in response to number of biotic stress. So, however, the integration of these salicylic acid, chitosan, and trichoderma in the integrated disease management practice is not clearly understood. So with this rationale, the present study was carried out to understand the effect of these integration of these signaling molecules like salicylic acid, chitosan, and trichoderma harzianum to manage this most important disease of rice. So here is our work plan, sir. Here we followed three application, 3 times of application. The first application was given as seed treatment, where we did the seed priming of two varieties. One is Tapaswini, which is a highly susceptible variety for sheath blight, and CR1014, which is being identified as moderately resistant to sheath blight disease. So we treated these seeds at trichoderma talc-based formulation, 10 to the power 6 CFU per gram of talc base at 6 gram per kg of seeds. And we after that we took the observations on different plant growth parameters, whether these the treatment with the trichoderma and also elicitors seed treatment have any effect on plant growth promotion. And in the second application, we applied the salicylic acid and chitosan at this 100 milligram per liter after that 45 days after sowing. Here, it is a critical period for the sheath blight disease inoculation, where the crop will be in maximum tillering stage. And the third application was made by chemical, that is azoxystrobin 2 ml per liter at 65 days after sowing. And we have recorded both the disease and yield parameters. And we recorded the effect of seed priming with salicylic acid, chitosan, and trichoderma harzianum and plant growth promoters, plant growth parameters with a different concentration of chitosan, salicylic acid, and trichoderma and their combination. So among these treatments, we observed that the combination of the salicylic acid, trichoderma harzianum, and chitosan, they resulted in maximum germination in both resistant and also the highly susceptible variety. And we observed the enhanced plant growth promoters like activities like maximum root length, shoot length, seedling dry weight, and also the seedling vigor was observed when we combined all these three. So the same experiment we continued here. The experiment was we did the seed treatment with trichoderma harzianum, then salicylic acid. Seed treatment was done by trichoderma harzianum, followed by foliar spray with salicylic acid, after 45 days or 45 days after sowing. And another treatment was like this, sir. So seed treatment with chitosan and followed by foliar application like this we followed. And at the 65 days after sowing, we gave common foliar application of azoxystrobin. So here we observed that the reduced disease reduction, significant reduction in the disease parameters were observed. And in terms of number of days for the sclerotia formation, here the T3 was recorded, took maximum number of days, that is 45 days for the sclerotia formation in case of Tapaswini, and in case of resistant, moderate resistant CR1000, it was 50 days. And area and disease progress curve also, it was low in case of T3. And we also observed that the disease control over control, around 60% of the disease was controlled over the pathogen control in case of Tapaswini, and 64% in case of CR1014. So we recorded the yield parameters also in the same experiment. Same treatment, that is T3, it is recorded maximum panicle bearing tillers and average number of panicle, average length of the panicle, average panicle weight, number of field grains, and also number of grains per panicle. And here the yield was maximum, that is 62.83 gram per pot. We have recorded in T3 in case of Tapaswini, and in case of moderately resistant CR1014. To confirm this, we also recorded the different defense enzyme activities like polyphenol oxidase, peroxidase, and also chitinase in all the 10 treatments at 24 hours before pathogen inoculation, 24 hours after pathogen inoculation, and 48, 72, 96, 120, and 144 hours after inoculation. Here also we observed maximum activity of polyphenol oxidase in both Tapaswini and CR1014 in case of T3 at 120 hours after the inoculation. Thereafter, the enzyme activity started declining. Similarly, in case of chitinase activity also showed maximum activity of chitinase in case at T3 in and also CR1014. And similarly, peroxidase activity was also in maximum in case of T3 in both in Tapaswini and moderately resistant CR1014. To confirm this, sir, we undergone both fluorescent microscopy and scanning electron microscopy. In case of control leaf sheath, there was profuse growth of Rhizotonia solani, mycelium was observed. But in case of treated, that is T3, we could not see any mycelial growth. And scanning electron microscope also give us the similar results, where the resistant structure like papilla were more in case of T3, sir, when compared to the control, sir. So the summary of this experiment was like this, where we treated the surface sterilized rice seeds with Trichoderma harzianum and helicitors, which allowed for the sprouting, enhanced the both plant growth promotion in both resistant and also susceptible variety. And the spraying with the salicylic acid and chitosan at 45 days after sowing, followed by challenge inoculation with Rhizotonia solani, will enhance the defense enzyme activities like peroxidase, polyphenol oxidase, and chitinase, which resulted in disease control, sheath blight reduction when compared to the control. And in future line of work of this is we are now designing some of the markers for to study the expression analysis of different defense genes in both in the different treated plants. And this will be helpful in deep understanding the host pathogen interaction in elicitor and non-elicitor treated plants. And with this, I'm concluding. I want to thank my director, Dr. D. Maiti sir, for giving me an opportunity, and also my mentor, Dr. Fakhruddin sir, for giving me this opportunity. Thank you, sir.
Thank you, Dr. Raghu. So, is there any question for Dr. Raghu? Anybody? Dr. Raghu, since you are going to take up the spray after 45 days,
Yes, sir. Yes.
practically sheath blight starts later in the season.
Yes, sir.
Maybe in a crop like 130, 140 days, 120 days.
Yes, sir.
After at the grain filling stage, sheath blight reaches its peak. How do whether the only one spray of salicylic acid and seed priming is good enough to take care of the disease towards the terminal end of the crop?
Sir, actually here the treatments which were given the seed priming were maintained, sir. The same treatment were given, the elicitor spray, and one spray was given with the chemical also, that is azoxystrobin at 65 days after sowing. So I think this was taken up in the control experiment, sir.
Okay.
So it will be
I'm talking
repeated.
about the field level. Field level.
Yes, sir. Yes, sir. Yes. Field. I am repeating this experiment in the.
Okay. Right, Dr. Raghu. Thank you very much. Are there any questions to Dr. Raghu? Thank you very much for a nice presentation. Thank you.
Thank you, sir.
Now I will invite Dr. Yogesh Vikal. Will be making a presentation on mapping of novel bacterial blight resistant gene XA47 identified from Oryza rufipogana and its transfer into Oryza sativa. Dr. Yogesh, please.
Yes, sir.
Yeah.
Yeah. A very cheerful evening to chairperson, co-chairperson, and the all participants. Well, today is the scenario of climate change that hampers crop productivity due to emerging biotypes or pathotypes for biotic stresses and aggravation of abiotic stresses. For sustainable crop production, there is need to develop climate resilience crops. Therefore, there is need to explore and identify new genetic resources to combat these stresses. In lieu of that, I would like to share my work on mapping of a novel bacterial blight resistance gene XA47 identified from Oryza sativa, Oryza rufibogone accession CR10098A and it's transferred into Oryza sativa. Bacterial blight is one of the most devastating diseases in rice caused by zoo, resulting in yield losses of up to 20 to 30% and in severe epidemics up to 80%. The zoo pathogen actually invades the xylem tissue either through wounds or water pores, leading to systemic infection. Host plant resistance is the eco-friendly and economical approach to manage this bacterial blight disease. Till date, 46 bacterial blight resistance genes have been identified and mapped on the rice chromosomes. And out of these 46, 40% of them are recessive in nature and 11 of the bacterial blight resistance genes have been cloned and characterized. Though many bacterial blight resistant genes have been deployed in resistance breeding programs, breakdown of resistance in many rice cultivars has been observed. This is due to rapid evolution in the virulence behavior of U pathotypes. So to manage co-evolutionary arm race between plant and pathogen, there is need to explore novel sources of resistance and utilize them in rice bacterial blight resistance breeding program. With this, wild species are rich a reservoir of genetic diversity. At our PAU, already we have mapped and already bacterial blight resistance gene XA38 from the Oryza nivara on chromosome 4, and recently we have mapped a gene XA45 from Oryza glabrima accession on chromosome 8, and this Oryza longistaminata has been explored for the productivity traits. And now we during screening, because this PAU has the active collection and maintaining the around 1600 wild species which are procured from the IRI Philippines and NRI Biotech. So during the screening against bacterial blight pathotypes, Oryza rufipagon has been found to be resistant to 9 of the pathotypes which are prevalent in the Punjab and susceptible to one of the pathotype, that is 1. Whereas from the Oryza rufipagon, another gene XA33 has already been identified and mapped. So this we explored it also against the Punjab pathotypes, and we found that it was resistant to all pathotype except pathotype 10. So both these Oryza rufipagon and XA3 have their contrasting phenotype with respect to pathotype 1 and pathotype 10. This makes us to see whether this gene is different from the XA23 or it is the allelic of that. So we have exploited these assertions and generated a mapping population by crossing it with the PR114. This is the actually experimental design and the screening genotyping of the mapping population. First of all, this Oryza rufipagon assertion CR11098A was crossed to PUSA 44 initially, and then the F1 generated was crossed to PR114, which is a semi-dwarf high-yielding variety. The F1 generated from this cross was back crossed to PR114, and the BC1F1 population was generated. The BC1F1 population and the F1 ones were moderately susceptible to the zoo pathotype 7. From the BC1F1, 5 of the randomly plants were selected and self to generate the BC1F2 populations. The BC1F2 population was screened against the zoo pathotype 7, and out of 5 BC1F2 population, 3 of the populations showed susceptible to the pathotype 7, whereas 2 of the populations showed segregation for the resistance and susceptibility. And then this BC1F2, the nature of the gene present in this Oryza rufipagon was confirmed in BC1F3 population using the same pathotype, and these two are used for the inheritance study. Then the BC1F3 population progenies were carried forward with single seed descent method to generate the backcross rails lines. This BC rails population was then used for tagging and mapping of the bacterial blight resistance gene, and this genotyping is done with both SSR and the Cas markers. Then both genotypic and phenotypic data was analyzed with MapMaker software for mapping of the bacterial blight resistance gene. So first of all, in inheritance studies, we observed that 463 BC1F2 population was segregated into 105 resistant and 358 susceptible individuals. So that showed the segregation ratio of 1:3, and that is which is recessive in nature. And the genetic ratio was confirmed in nature of the gene was confirmed in BC1F3 progenies and also in 388 BC rails, where they show the segregation ratio of 1:1. So that confirmed that the resistance gene present in Oryza rufipagon is conditioned by a single recessive gene. So this is the showing this peak is showing the distribution of average layer length of the backcross rails. Oryza rufipagon has an average layer length of 2.3 centimeter, whereas PR114 showed the average layer length of 25.4 centimeter, whereas this BC rail population fall in the range of 1.1 to 30.4 centimeter of layer length. This peak with this number one is showing the layer length on the PUSA 44, and second is for the PR114, and the 3 to 8 are the homozygous resistant BC rails, and 9 to 13 are the homozygous susceptible BC rails. Now we did simultaneously center polymorphism survey and bulk segregant analysis. So a total of 203 SSR markers spanning all the 12 rice linkage chromosomes were analyzed on this parents PR114 Oryza rufipagon and the bulks, and 62 were polymorphic. That is 30% polymorphism was observed between the parents, and 2 of the SSR markers which are present on chromosome 11 that showed co-segregation with the resistance. These 2 markers are RM27235, RM21365. This clearly show that the resistance allele which is present, Oryza rufipogon, that is present in resistant bulk with both the marker, and the susceptible allele which is present in PR114 cultivar is present in the susceptible bulk. And these 2 markers were to confirm their association were then employed on the open bulk. So in the open bulk peak, you see clearly the resistant allele is registered in all the homozygous resistant BC rails, and the susceptible allele is present in all the homozygous susceptible rails. So within this bracketed region of 2 markers, from the SNP, we have the GBS data available for the PR114 and Oryza rufipogon this session. So SNPs were SNP-based markers, that is Cas markers, were designed between these 2 markers, that is bracketed region. 86 Cas markers were designed, and these 86 Cas markers were then employed on the bulk segregant analysis. And from this 86, 3 of them showed the association with the gene. And this picture is showing that this is the rectangular shape is the parent rufipogon, and the oval shape is the parent, resistant bulk, and this on the blue dots that are for the susceptible parent and the susceptible bulk and the open bulk dots. And some of the resistant lines, open homozygous resistant, they showed the heterozygous SNP. Now, within that bracketed region, we again synthesized more 77 new SSR markers, and these out of these 77 new SSR markers, 26 were polymorphic, and this 26 marker along with these 2 markers, associated marker and the Cas markers, were genotype of the whole of the 388 BC rail population. This peak is showing with one of the marker. And after genotyping, the gene was placed between the 2 markers, that is PAU 1165 and PAU 44, with the region of 2.8 centimorgan. And we also generated the graphical genotyping for all the 388 BC rails within the region of 7.8 centimorgan, and we observed 30 of the recombinants, whereas 11 recombinants were observed within the linked marker, and one of the recombinant showed the smallest introgression with respect to these linked markers. Now, this gene was, physical map was constructed bioinformatically locating the coordinates. So we use the start coordinate of the marker, the 65, and the end coordinate of the marker 44 to locate this gene into the physical distance of 376.3 kb. And this gene, this region actually contains the candidate genes, about 59 candidate genes. And this chromosome 11 already, and we located this gene on the subtelomeric region of the chromosome 11. Chromosome 11, 15 of the BB genes are clustered on chromosome 11, showing that it is a hotspot region for the resistant gene. And these 6 of the genes, XA3, XA40, XA44, 36, 43, and 4 are present on the right hand side of our gene, and whereas rest of the genes, this XA23, are present on the left side of the region. So as all the genes are dominant in nature except XA44, that confirms that our gene is non-allelic to this dominant gene. This XA44 has been identified from the Japonica cultivar, and it is avirulent to the Korean races, whereas our identified gene has been the collection of from the Indian accession Oryza rufipogon, and it is avirulent to the Punjab pathotype. So that shows it is non-allele to this, but still we have further confirmed the uniqueness of our allele. We employed the linked markers of the genes which are present on chromosome 11, like for IRBB21, this is XA21 gene. It has a different allele from our parental alleles. This resistant allele, these are the susceptible alleles which are present in the Oryza rufipogon and the PR114. Similarly, for IRBB23, because this XA23 is also derived from the Oryza rufipogon from different accession, so it is also not an allele which is present in our accession, that is Oryza rufipogon CR10098A. And this XA44 is showing non-polymorphism between our parental line, that confirms that it is non-allelic to XA44 also. Similarly with the other markers. Next, we have also, you can
you already taken more time.
Okay, this is the last one. We have studied our linked marker, that is they are suitable to perform the mass. So we have employed our linked markers to the released rice varieties from the Punjab, and it showed a different alleles. So we can say this marker, linked marker, can be successfully employed for the mass for either singly or stacking with other BB resistance genes. So these are the putative candidate gene which we have identified, and we have now in a process of development of gene-based markers to further narrow down the region. This is a general mechanism for the BB genes already available. And in conclusion, we can say there is a need to further saturate the region with more markers to facilitate the isolation of XA47 gene. Its characterization and cloning is an important step in understanding its mode of action to provide resistance, and will provide further insight into the molecular mechanism of resistance. And these are some of the varieties in which we have deployed the genes, BB genes and the varieties are released as a mass product. Punjab Basmati 3, 4, 5, and this PR1212. This is the recent released variety PR127 in which XA45 have been deployed. With this, I would like to thank the coordinates giving me an opportunity to share my work on this platform. And thank you all to the chairperson and the co-chair persons.
Thank you, madam. Thank you very much.
Thank you.
I think you have not left any time for question.
Okay, sir.
So maybe all of you may contact or email for any queries, right? Thank you, madam.
Thank you, sir. Thank you.
Move to the next speaker, Dr. Anuradha Chellai. She will be making a presentation on identification of Physarum oxysporum forma species cubens TR4 resistant gene candidates in bananas. Dr. Anuradha, please. 10 minutes time.
Madam, Anuradha madam, kindly unmute your speaker. Madam. Madinabri.
It's unmute only.
Yes, madam. Now we can hear you, madam. Share your slides, madam.
Okay. Okay. Now it's okay, I think, ma'am.
Yes, yes.
Okay. Thank you. Thank you. Thank you, ma'am. Respected chair and co-chair. And all the participants, a good evening to all. Today I'll be presenting the topic on identification of Fusarium oxysporum form of species cubensis TR4 resistant gene candidates in bananas. As you all know, banana is one of the major food crop, and its production is seriously threatened by Fusarium wilt, which is caused by a soil-borne fungus, Fusarium oxysporum cubensis. There are many races in case of Fusarium oxysporum, of which FOC TR4 attacks Cavendish clones, and and it causes which are the source of 99% of the banana exports, and it also infects various diverse range of other banana cultivars, including the cultivars which are susceptible to race 1. FOCTF also threatens the multi-million dollar banana industry, but also it affects people in developing countries whose main source of calorific intake is banana. In India, FOCTR4 was reported in 2015 in cultivar Grand 9, which occupies an area of 63% of banana growing area in India. The FOCTR4 incidence is up to 35%, and this may lead to incur a heavy loss to Indian banana industry. So coming to the need for resistant genes, once infected with FOCTR4, it is successful to replant the susceptible cultivars because the chlamydospore remain in the soil to a span of 30 years. Till date, there are no effective control strategies, and the only practical strategy is to cultivate resistant cultivars. Resistant sources has been identified at NRC banana, and but the major problem is introgressing these resistant genes in commercial cultivars like Grand 9 through conventional breeding, which is not possible due to polyploidy, sterility, and low fertility. An alternate strategy to this is to incorporate resistance through non-conventional breeding, for which resistant genes is a prerequisite. So there are recently reliable banana transformation protocols is available, and this makes molecular breeding quite easy, and we will be able to generate disease-resistant banana. So coming to the structures of major plant genes and how the pathogen interacts with the plant. So there are two major class of R genes in case of plants. One is the nucleotide binding site leucine-rich repeat, and another is the transmembrane domain leucine-rich repeat sequences. NBS LLRs are further classified into toll interleukin-1 receptor like NBS LLR genes and coiled NBS LLR genes. These genes, NBS LLR genes, they are usually present in the cytoplasm. And the transmembrane LLR proteins, genes, they are of two types, that is receptor-like kinase and receptor-like proteins. Both of these genes are genes, they have the same domains, that is signal peptide, leucine-rich repeats, and transmembrane domains, but there is a extra kinase domain in case of LRR-RL case. So coming to the mode of interaction in pathogen and the plant, once the pathogen infects the plant, the pathogen produces the toxins as well as the effectors. Once the toxin is released, the toxins have microbe-associated molecular patterns which are recognized by the pathogen recognition receptors. These pathogen recognition receptors are nothing but the LRR, RLKs, and RLP genes. This in turn will trigger the PAM-triggered immunity, which in turn will switches on all the defense-related genes. So when the primary defense mechanism is not working, or when the pathogen overtakes this primary defense mechanism, the effectors enter the cytoplasm. These effectors will then interact with the R proteins, and they will trigger the effector-triggered immunity. In this way, the plant and the pathogen is able to interact. So coming to the methodology, based on the mapping population and inheritance studies made by Elizabeth Atkin, it has been reported that a TR4 resistant gene is confirmed by a single dominant gene. So based on their studies, actually they have identified the SNPs, and they have made assessment in the Musa-acuminata malacensis population by comparing the bulk of resistant with that of the bulk of the segregant. And finally, they were able to study the distribution of the SNPs in each chromosome, thereby revealing the hotspot where R genes could be, probably the R genes could be present for TR4. So in silico analysis was carried out using publicly available Cavendish and DH Paghang transcriptome data, and these transcriptome data were then mapped to the R gene loci present in the hotspot region of the banana genome. So from this in silico studies, we have selected a few candidate, we have selected few genes which were upregulated upon TR4 infection, and we tried to study its expression in resistant and susceptible cultivars upon TR4 infection. So based on this QRT-PCR results, we have narrowed down with the 3 candidate genes, and these candidate genes will be further validated for its resistance against the TR4 through a knockdown approach by CRISPR. Next, so this slide you can clearly see that the transcriptome data of the resistant and the susceptible cultivar is mapped against the banana genome. So this is the banana genome, and here you can clearly see the number of reads in resistant cultivar is higher than that of the susceptible cultivars upon TR4 inoculation. So this is in case of Cavendish cultivar, you can see the number of reads with that of with respect to this particular R gene is higher in case of TR4 inoculation than that of the mock inoculated. So similar results have been observed in case of Pahang upon TR4 infection. So here also you can clearly see the number of reads is higher more when compared to the mock inoculated. So based on these studies, we have we have narrowed down to 20 R genes, and we validated these R genes through QRT-PCR in resistant as well as susceptible cultivars upon TR4 infection at various time intervals, that is 0, 24, and 48 hours. So here you can see the expression of this particular R gene in case of cultivar rose. Cultivar rose is a resistant cultivar, and Matti, Namare, and Grand 9 are susceptible to TR4. So clearly you can see there is a fourfold upregulation in case of resistant cultivars upon TR4 infection at 24 and 48 hours. Similar is the case with this gene as well. So similarly, we have carried out expression analysis for all the genes. R genes present in that particular hotspot loci. So here also you can see this particular gene is expressed fourfold at 24 hours in case of resistant cultivar. So based on this validation, we have narrowed out to 3 candidate genes against FOC TR4. So they are named as RGA2, RGA3, and RGA4. RGA2 is an NBS LLR type of R gene. So this type of R gene will usually interact with the effectors of the FOC TR4. So here you can see fourfold upregulation at 48 hours in case of cultivar rose, whereas it is downregulated in case of susceptible cultivars. And RGA3 is a transmembrane type of R gene. That is, it is a LBS LRR RLP. So here also you can see fourfold upregulation at 24 and 48 hours in case of resistant cultivars. And RGA4, this is a receptor-like kinase type of R genes. So these 2 R genes, they are pathogen recognition receptor like that is transmembrane LRR type of R genes. So here also it is upregulated at 24 and 48 hours. Here you can see it is more or less uniformly its expression is maintained. So all these 3 R genes were cloned, amplified, cloned, and sequenced from resistant and susceptible cultivars. So gene, you can clearly see the amplification, and they were sequenced. And so motifs and domains of these R genes were studied. As I have mentioned, RGA2 is an NBS LLR type of R gene. So it has and mostly in monocots, coil type of NBS only is present. And this NBS also belongs to the coil type of NBS LLR. And here you can see NBR domain and leucine rich repeats, 18 to 21 repeats are present. So RGA3, this belongs to RLP. So here also a signal peptide is there, a transmembrane domain is there, and 14 to 18 repeats of LRR is there. So RGA4, it has an extra kinase domain as compared to the 3. Next, so finally, to validate three R genes, guide RNA has been designed, and we have cloned that in a CRISPR vector, and we are in the process of transforming so further, we were able to identify a genic SSR marker in case of one of the 3 candidate R genes. So this genic SSR marker was able to clearly differentiate the resistant from that of the susceptible cultivars. So here you see cultivar rows, we are able to get only was a single band, whereas in susceptible there are 2 bands. So this was further validated in all the reported internationally reported cultivars under various genomic groups, and you can see clearly a single band in case of resistant cultivars and double band in case of susceptible cultivars. And this SR markers further validated in the mapping population of matti into cultivar rows. So here also you can see different banding patterns among 18 progenies of the mapping population. So to conclude, 3 candidate genes has been, the gene SR marker has been identified from one of the candidate gene, and both candidate gene as well as the marker has to be further validated through gene has to be validated through CRISPR, and SSR marker has to be validated in further mapping populations. And I wish to acknowledge our director, Dr. S. Uma, who helped me in facilitating all the work related to this project. And thank you.
Thank you, Dr. Anuradha, for a nice presentation, also in identification of 3 R genes against Fusarium wilt race 4. Right now, not a problem in South India, right?
No, it's not it.
Southern parts.
Southern parts not in Karnataka, Andhra Pradesh or Tamil Nadu, Kerala, but in Maharashtra and Surat, means Gujarat and UP and Bihar parts, Bihar it has been reported.
So
not in southern 4 states.
Maybe from the rhizomes and suckers, it might be introduced. That is the apprehension, right?
Yes, sir. Yes, sir. Possibilities are there, sir.
So, are there any resistant varieties in the already available cultivars? Under the not through the
commercial cultivars, it is not there, sir. But for race 1, it is available. Red banana, Neintran and all. But for TR4, commercial cultivar is not there. But few of Cavendish we have identified, but not yet reported. So soon publication will be there from resistant sources. Actually, few we have identified which are resistant to.
So you will have a lot of way in identifying this gene and put it into maybe marker-assisted breeding, right? Could be another source.
Yes, sir. Yes, sir.
Thank you very much.
Yeah.
Is there any question? Quick question to Dr. Anuradha.
Madam, what about in case of PU1 types? This TR4 affecting PU1 types?
It is affecting, but actually it is there, but mostly in Cavendish gram only it has been reported. Isolate from PU1, I don't know. Maybe it is there. It is susceptible as far as screening was done upon TR4 inoculation. PU1 was susceptible.
No, Dr. Anuradha.
Yes,
Dr.
madam.
Anuradha, you can say there are certain inputs of PU1 are resistant.
Thank you, madam.
Okay.
Okay.
Thank you.
Because it is a major cultivar in Karnataka. That's why I asked, madam.
Okay.
Thank you,
not
madam.
all PU1, I think.
Thank you very much.
Thank you.
Yeah.
Now I will invite Dr. Manjunath G. Will be making a presentation on comparative genome sequence analysis of Ceratocystis pimbria with genomes of Ceratocystis family. Dr. Manjunath, please. 10 minutes time.
Sir, Manjunath G sir is not there.
Okay.
I request you to go for next speaker, sir.
Thank you, madam. I'll now call upon Dr. Ramya. She will have her presentation on molecular. Is she there?
Yeah, she is there.
Okay, if she is there, she will be making a presentation on molecular detection of spine resistance in population of rust red core beetle, Tribolium castaneum. Dr. Ramya, please.
Hello. Good evening, sir.
Yeah.
I hope I'm audible and my screens are visible.
Yeah.
Yeah. Good evening. Good evening, good evening, respected chairperson, respected dignitaries, and the various delegates attending this conference. I'm first of all thankful to the organizers for giving me this opportunity to present my work in front of you all. I welcome everyone to my presentation. I'm Dr. Remy RS. I'm a scientist in the Division of Genomic Resources in ICR NBR Bangalore. And I am here to present my work on molecular detection of phosphine resistance in populations of rust red flower beetle Tribolium castaneum. So I will just give a brief about why the study was undertaken. As you all know, India is one of the largest grain consumers in the world with large-scale reserves of food grains by the government on a long-term basis. And wheat is a chief cereal crop of the country. And out of the total produce, around 30% of the various storage agencies collect the wheat and store them on a long-term basis so that it can be given to public distribution. So 30% of the food which has been produced goes to the storage. And rust red flower beetle comes to the scenario in this stage. Rust red flower beetle shows global distribution as a tropical and a subtropical pest in wheat and wheat products, and it causes damage up to even 15%, and there are reports of causing damage even to 30%. And fumigation is the most effective method since the pest comes in the storage. And phosphine is a major fumigant which is used worldwide due to various reasons like quick dispersion, low residual activity, and also cost effectiveness. But the Montreal Protocol, which came in 1997, which was a agreement which was made between the industrialist nation to phase out one of the major front runner of the fumigant, that is the ozone depleting methyl bromide, by 2005 in developed countries and 2015 in developing countries. This made excess pressure on phosphine, and there has been over dependence on phosphine to control the storage pests in wheat and also other crops. So this scenario caused the phosphine to be used repeatedly, and this along with non-proper sealing of the storage condition led to underdosage and lead to repeated control failures, and this resulted in developing resistance. That means the phosphine resistance has been developed by several pests and has been reported in different regions of the world by several workers. So this phosphine resistance has to be monitored repeatedly and on a long-term basis so that appropriate resistant management strategies can be initiated. So one of the most important one of the most important method for monitoring resistance is FAO type bioassay. But is it the best option? There are various drawbacks for this FAO type bioassays, which are this bioassay requires living adults, which has been recently collected from the population of interest, and this need access to quantitative analytical chemistry like gas chromatography, which is needed for measuring the concentration of phosphine which has been generated, and this needs handling of dangerous phosphine gas, which is extremely dangerous to the handler who is handling the phosphine gas for doing the bioassay. And also the bioassay will not give you specific information on the genetic base of the resistance, will only show you the presence or absence of resistance, and it takes more time and even the assessing the mortality takes 7 days along with the other time involved in exposure and all. So ours is a collaborative work which has been done with collaborators from University of Queensland, Australia. So our collaborators have done the initial work which has been published in a reputed journal Science. They have discovered that a core metabolic enzyme mediates resistance to phosphine gas. They have discovered that there are two genes involved in causing phosphine resistance in Tribolium and other storagens also, but I am dealing with Tribolium here. So there are two resistant genes, RPH1, which is responsible for weak resistance, and RPH2, which is responsible for strong resistance. RPH2 codes for the enzyme dihydrolipamide dehydrogenase, and this is the DLD gene in Tribolium castaneum along with the predicted domains. And this DLD gene is a monodimer of consisting of four enzyme complexes. These are present in the mitochondria like glycine cleavage system, pyruvate dehydrogenases, branch chain keto acid dehydrogenase, and alpha ketoglutarate dehydrogenase. So in this DLD gene, amino acid substitutions happen, and this happen in the catalytic disulfide center and the cavity relating leading to it. So the amino acid substitution, as you can see, proline to serine substitution happens due to a single nucleotide change of cytosine to thymine, and this nucleotide change results in the generation of a recognition site of the restriction enzyme MBO1, which helps in generation of markers, which I'll be dealing later. So this is a main thing which happens in DLD gene, which leads to phosphine resistance in tripolium. So based on this, we have collected population from 15 locations that lead around 5 states like Uttar Pradesh, Punjab, Haryana, Madhya Pradesh, Rajasthan, and also we got a susceptible strain which has been repeatedly reared in TNAU without in the lab condition TNAU without exposure to phosphine. So we have taken that as a susceptible strain, and other 15 field population which we assume may be having resistance. So we did the normal conventional phosphine biopsy with this 15 population. For that, we have taken the adult stage of the insects inside the gas tight desiccators, and then we generated phosphine by submerging a sulfose, that is an aluminum phosphate tablet, in 5% sulfuric acid, and if the phosphine gas which gets generated, we sucked in using a Hamilton syringe, and with this Hamilton syringe we have injected the phosphine into the desiccator so that the insects gets exposed to the phosphine. And then we gave an exposure period of 20 hours, and after that the desiccators were opened, and then the mortality was assessed after 7 days so that endpoint mortality will be achieved. After this biopsy, we got the LC50 values, and so as you know, higher the LC50 values means higher the resistance of the population. So we got the lowest LC50 value for the susceptible strain because it has not been exposed to phosphine. So we got 0.07 LC50 values. But along in that line, we got one population Haryana, which is also having low LC50 value. So we thought we took this as susceptible strains, and also resistance was detected in Ajmer population, which show a higher LC50 value of 1.901, and other populations are also which also showed similar high LC50 values were together assumed to be detected to be resistant population. So we calculated the resistance ratio, which is actually a ratio between the LC50 of the resistant population with the LC50 of the susceptible population. So higher the resistance ratio, higher will be the resistance of the particular population. So we got a higher resistance ratio of up to 26.77, which is detected in Ajmer when compared to the susceptible population that isn't that's a TNA computer population. So based on our previous study which has been done in University of Queensland, we tried a marker that is MPO1 CAPS marker, that is cleaved amplified polymorphic DNA marker, so that we can detect the resistance molecularly, so that hence the quick detection of the resistance will be possible, and thus appropriate resistance management strategies can be initiated. So as a normal protocol, we have isolated genomic DNA from all the 16 population, and the DLD gene, which is responsible for resistance, was amplified using PCR, using the gene specific primers, and the PCR was performed using with the conditions, predetermined conditions. And then we got a PCR product which is of 368 base pair length, and because of, as I have already told, the nucleotide change makes the recognition site for the restriction enzyme MBO1. So after the PCR product, we got it digested with MBO1, and after the restriction digestion of MBO1, we got three types of products. If we got an original uncut PCR product, it is susceptible because it doesn't have the recognition site, and two fragments 72 and 296 base pair if it is a homozygous resistant one, and three fragments 72, 296 and 368 base pair if it is a heterozygous resistant one. So after this, we have estimated the allele frequency using this formula, whereas RR is homozygous resistant, RS is heterozygous, and SS is homozygous susceptible. So this is about the VLD gene fragment which got amplified after the PCR. We got a 368 base product, and this is what I have told. Like if it is two bands, that is 296 and 72, that is homozygous resistant. If there are three bands, 368, 296 and 72, that is heterozygous resistant. And if there is only one band, that is 368 base pair without the cut, without the MBO1 product, that is susceptible. So we have done for all the 15 population plus the susceptible population, and we got same correlated with our bioassay studies. We got the same results when in the molecular one also. So the Coimbatore population, which have taken as susceptible population, we got R allele frequency as 29.31%, whereas in the highly susceptible, which we got in the bioassay population, bioassay studies, that is Ajmer, we got R allele frequency of even 83.93%, and the other population sandwiched between these two. So we got a clear relationship between RLN frequency and the LC50 values, which we have obtained using the FAO type bioassays. And so as conclusion, I'll tell that population from India have developed significant level of resistance to phosphine, the populations of Tribolium castaneum. And we have seen that population from locations like Ajmer, Indore, Jaipur, Hapur, Jhansi, Ludhiana, and Narwana has exhibited high degree of resistance, while Gohana population showed relatively less resistance. And this MBO1 CAPS marker screening has confirmed phosphine resistance in relation as same as that of the FAO type bioassay studies. And that's what I told. Close correlation between phosphine resistance and RLA frequency was observed. And the major takeaways from our study was CAPS marker analysis is comparatively simple to carry out and economical. It can be applied to living, dead, preserved, or dried specimens of beetle, whereas the FAO type bioassay cannot be done. So this makes it possible so that data can be obtained for collections made over large geographical areas and at many sampling times. And it enables us to understand the genotype, whether it is heterozygous, homozygous, or susceptible of the resistance, unlike bioassay, which will show only the presence or absence of resistance. And it gives even the information of precise allele frequency, even when the frequency of resistant allele is low, and the phenotype will be mostly unnoticeable in bioassay. That also we can pick up in this method, and also gives a possibility for evolution of phosphine resistance in a pest population, and provides a forewarning so that appropriate management action can be initiated. So if after the CAPS marker screening, if no strong resistant alleles have been observed in a particular pest population, we can manage the pest by this appropriate management action, that is by increasing the gas concentration of phosphine, and with longer exposure times, warmer temperature, and in well sealed structure. This will eliminate the weak resistant insects and will prevent the development of more resistance. And if strong resistant alleles are present after the CAPS marker screening, we have to go for alternative control methods other than phosphine, and thus we can prevent the selection for even stronger resistance to phosphine. So I acknowledge University of Queensland, Australia, our collaborators in University of Queensland, Australia, DST, DST for providing support, ICR, IR in New Delhi, and NPR Bangalore. Thank you. Thank you for listening.
Thank you, Dr. Ramya, for a good piece of work presentation. If there is any question, quick question, one question. Anybody? That means major populations in northern India, except you feel it is a resistance. So what is the alternative? If you go on increasing the dosage, you might eliminate the weaker ones, but the strong resistance can also be obtained when you go on giving the higher dosage and longer exposure. That is also very deadly.
Yeah. So what is the problem in India is mostly our sealing conditions are not at all accurate. So it leads to leaking of the gas, and almost underdosage is happening in our godowns. So in Indian condition particularly, if we get the sealing done properly and the gas is maintained in the fumigations thing, and with exposure period we can manage. Since India we have this problem, but in we have collaborators in Australia, they don't have this much leakage in their godowns, so better maybe
Even underdoses also can lead to such.
yeah.
Yeah, thank you for your good presentation. Well done.
Madam, any organic fumigation alternatives?
Yeah, organic fumigants, there are researches done based on you may be knowing like our household things like neem and all, but in a large level in go downs it may not be practical all every time, but there are studies undergoing even in our institute to get the like essential oil extract and get it done in a higher like in a larger way, but it has not reached up to the commercial level still, since still researches are going on.
Thank you, madam.
Important point. You need to focus on organic soil.
Yes.
Okay, thank you, madam. Thank you very much. May I now call upon the last speaker of the day, Richita Saikia, to make her presentation on understanding the viral complex associated with the leaf curl disease of Butylokia, that is Capsicum chinense. Dr. Richita, please.
Sir, am I audible?
Yeah.
Just a second, sir. 1 second, sir. Sir, are my slides visible?
Yes. You can go ahead.
Yes, sir. Okay, sir. 1 second, sir.
Yeah, 1 second.
Okay, sir. Good evening, everyone. Respected Chairman, sir, and Co-Chairman, sir, Dr. Nayak and Dr. Kulkarni. My name is Richita Saikia, and I'm currently a PhD scholar doing my research to understand the viral complex associated with the leaf curl disease of Butylokia, Capsicum sinensis. And I'm doing my PhD from Assam Agricultural University, Jorhat.
Can you make it in a slideshow presenter? Yeah.
Yes, sir. Okay, sir. Firstly, I would like to thank all the organizing members and committee of the University of Horticultural Sciences, Bagalkot, to give me the opportunity to present my current work in this international e-conference. So first, I would like to start with the background of my current study. So Capsicum sinensis Jack, commonly known as the Butylokia, it is an cash earning crop in Assam and also in various regions of Northeast India. And it is one of the hottest chilies, ranking world number 7 around the globe. And it has a high capsaicin content of 3 to 5%. And Butylokia is the interspecific hybrid of two species, that is Capsicum sinensis and Capsicum frutescens. So lately, these species has been facing a constant threat due to the infection of the viral leaf curl disease, and due to which the viral complex is not been understood so well till date. So coming to the rationale of my study, I would like to say that the molecular mechanism behind this disease development is still not understood. And several viruses have been reported in Butylokia, but only at least 5 viruses have been reported in association with this leaf curl viral disease in Butylokia, namely cucumber mosaic virus, PV potato virus Y, then chili leaf curl virus, then groundnut bud necrosis virus, and tomato spotted wilt virus. However, as I mentioned, the molecular mechanism is still not understood. Which of these viruses are actually responsible for the disease is still not clear, along with the symptom manifestation. So in this study, I will try to identify the actual pathogen or the combination of the pathogens associated with the disease, that is the leaf curl disease. So coming to my work, firstly disease sampling was done from different parts of Assam, and as you can see, these are the leaf curl symptoms showing upward curling and stunted growth of the Bhut Jolokia plant. And these leaf samples were collected from 3 districts of Assam, the Golaghat, Sibsagar and Jorhat. Then from this disease sample, we had performed DAS ELISA and PCR. DAS ELISA was done for the detection of the particular viruses, the particular DNA and RNA viruses, and for this TYLCV screening of the samples were found negative for all the samples, while one second, while CMV screening from Jorhat and that too from the samples of Golaghat were found to be positive in case of ELISA. And for PCR analysis, all the collected DNA samples were done using the chili leaf curl and the tomato yellow leaf curl primer pairs. So finally, the samples from Tiok Jorhat had shown positive results for both the primer pairs, and the other samples from the other districts were found to be negative for both set of primers. So what we did was that the results from both PCR and DAS ELISA showed that the chili species from Tiok were found positive for 3 different viruses, that is tomato yellow leaf curl virus, cucumber mosaic virus, and chili leaf curl virus. So these positive samples were further used to check the transmission in different host plants, namely Nicotiana tabacum, Benthamiana, Amaranthus, and chili. So these were the model host plants for the transmission, and mechanical inoculation of viruses were performed in this by giving a minor injury using carborundum powder, where the samples, that is the positive samples from Tiok, were homogenized in sodium phosphate buffer. So after this, after the mechanical transmission, disease scoring was achieved. You can see here the leaves were regularly observed for lesion formation, and like after 3 weeks of prior inoculation, disease symptoms of upward curling were seen. The first picture and the second are for the plants of Amaranthus. The third picture is for the chili plant, and the fourth is for the Nicotiana tabacum plant. So these were the PCR results for the DNA and RNA viruses of the disease code plants. Though the results were not very satisfying, I shall be repeating the experiment again, because I'm still my research is going on. The first picture is for the CMV. You can see a bit faintish bands with an expected amplicon size of 600 base pair. The next with the chili venal mottle virus having an expected amplicon size of 720 base pair. And the third gel picture is for the PVY primer having an expected amplicon size of 689 base pair. And the third one for the chili leaf curl virus. So this experiment will be repeated again. Then again, we had done sampling for the second time for this year during the month of February. And then these were the PCR results for the second sampling, disease sampling, which we had done. And these are the PCR for the respective DNA viruses. This one is for the first gel picture, though it is showing multiple bands, but still it confirmed confirm positive result for the particular virus, that is for the tomato leaf curl New Delhi virus. The second gel picture is for the tomato yellow leaf curl virus, and the third was for the chili leaf curl virus. Then coming to another this thing, we had I had done association of tomato leaf curl Joydepur virus was seen with that in association with Bhut Jolokia, showing 96.63% identity with the help of degenerate primers targeting the begomovirus viral replicative form. which were like used for PCR amplification of the DNA samples, that too infected chili samples from Jorhat. And as you can see, this is the top 21 blast hits of those PCR positive chili samples. Then coming to that of the PCR results of those positive RNA viruses, the first gel picture is for that of cucumber mosaic virus. The second one is for the chili venule motile virus, and that of the third one is of the PVY. As you can see here also, the bands are very faintish. So I shall be repeating the experiment again. And further work to be carried out is like deep sequencing would be done with the help of NGS platform after isolating the total DNA and RNA. Then with the help of those of the deep sequencing data, I would be identifying conserved microRNAs and also their target genes, along with the hot and cold identification of the hot and cold viral spots and their possible target regions. I would like to lastly, I would like to conclude that from my present study that I'm doing, I will investigate the host pathogen interaction and try to identify the tolerant source, and also to like develop an easy diagnostic in order to like lessen the leaf curl disease in chili leaf curl disease in case of commercial cultivation. And lastly, I would like to thank my supervisor, Dr. Bokhanto Kumar Bora, and also the organizing committee of University of Horticultural Sciences, Bagalkot, for giving me this opportunity to present in front of you people. Thank you.
Yes, sir. Dr. Ruchita.
Sir, I am not yet a doctorate. I'm still a current PhD student.
Okay. Hopefully, no problem. Shortly.
Yes, sir.
Okay.
Yes, sir.
May I now request anyone to have observation, quick observation?
Sorry.
Sir,
Yeah.
she is working on a chili leaf curl, associated chili leaf curl, and also she's other viruses she has used PCR, RNA virus PCR, but should be RT-PCR. As per my knowledge, there is no tomato leaf curl, yellow leaf curl virus in India, in a chili or in a tomato.
Yeah. Chita, please.
Yes, sir.
You want to respond?
We are still working on this, sir, because this, as I told, no, there are several viruses affecting this in association with this leaf curl virus, but it is still not understood, like which is the actual pathogen, like associated with the disease. So through the help of NGS platform, after we do the deep sequencing, we will be able to know that what is the actual like virome related to this particular leaf curl disease.
Are you satisfied?
Yes, sir. Virome or NGS you can go, you can identify.
Hello?
Yes, sir. Okay, sir.
Yeah. I think.
Sir, madam, is there, sir, I have a small question.
Yeah.
Hello.
Yeah.
Sir, is there any relationship between pungency and resistance against viral diseases in particular?
Pungency, I'm not that sure, but the particular disease I'm working with, it is mainly due to the insect vector known as Bemisia tabaci, commonly known as whitefly.
Okay, thank you.
Okay, in fact, there are a lot of
Yes,
leaf curl,
excuse me.
leaf curl.
Yes, yes.
Excuse me.
Yeah.
Mr. What are the other symptoms you look for the leaf curl virus?
We generally see upward curling, mainly upward curling is there because and then puckering, we see puckering, and also like stunted growth of that particular chili species.
So the two symptoms only, the puckering and the leaf curling.
Yeah.
Okay. Did you try it with the vein yellowing or pale yellowing or reduction in the leaf size?
Can you see here?
Okay.
You can see here upward curling is there, and then the like growth remains stunted. They do not grow further.
Pardon me, but okay. Fine, fine.
Okay.
Okay, I think we have been discussing so much on chili complex ever since the chili plant is here. I'm sure one day we will have the clarification. Okay. Yeah. With that, we will thank Richita for her good piece of work. Thank you, madam.
Thank you, sir. Thank you so much, sir.
Yeah, coming to the end of all presentations. Dr. Kulkarni, you have any concluding remarks?
Just I want to interact here. Totally, we had 4 invited talks and 1 keynote address. Among them, except R.L. Ravikumar, all have presented. And it was a very good source of information for all of us. Actually, I had a refresher course in virology actually today. Actually, we learned a lot of new things in this molecular biology of the biotic stress resistance. And among the oral presentations, of course, except 1 or 2, all are up to mark. They have to improve. And we have in India or over the globe now a lot of work has still to be done on practical aspects of viral management, because any amount of our genic level or molecular level, it should be of immediate practical use. Otherwise, it may take another 4 or 5 years to confirm resistance, some of them told. But we have to work on the immediate result-oriented aspects. That is my opinion. Overall, the session was very fruitful, and all were very actively involved in their work. And I think it's a very good contribution what we are giving to the faculty of science to improve upon the understanding about the molecular aspects for a biotic stress resistance. I thank all the presenters as well as the participants.
Yes, sir. Thank you, Dr. Kulkarni. We are coming to the end of this particular session. Let me take one minute of your time. Let me first express congratulations to the team of UHS Bagalkot, particularly led by Professor Fakhruddin and his team, who have done a very efficient job of conducting an international e-conference on outlook in biotechnology and crop improvement. Most of those talks which are drawn here are well thought out, very distinguished speakers. And tomorrow also you have one more session to go. And as it comes to our session, I think you have taken care of most of the aspects, including the gender issue. There were 5 female speakers and probably 3 male speakers. Only 2 were absent, probably Dr. Ravi Kumar and Dr. Manjunath. So covering most of the aspects of biotic stress. Of course, only one aspect in terms of insect pest, but otherwise mostly confined to virus. There were as many as 3 or 4, I believe. And good coverage has been given. I think this particular conference and the session on advances in molecular biology, particularly against biotic stresses, has given some new insights, including information on gene networks, chromosomal location of relevant gene, particularly that particular paper on BLB on chromosome number 11, large number of genes being clustered there. Of course, I'm sorry that I could not give time to her, but it was a very good presentation. And also, in the course of several talks, Dr. Kushalappa also mentioned the inferred the evolutionary history, and also some of those previously not undescribed phenomena also came into picture here. I think by and large an unparalleled perception it has given. I think transcriptome analysis has been dealt very nicely in saying that the genes involved, particularly in pathogenesis, and an insight on host pathogen interaction, very complex interaction that has been also interplayed between the host and the pathogen, its metabolism and biological stewards have been dealt very nicely, and the genetic and molecular mechanism in one or two presentations, and race-specific resistance. I think I foresee elucidating structural and molecular mechanism in future, specificity of defense mechanism, and also parasitic ability, interactive resistance, and some more insight on the durability of resistance, because after all, we see that so far the conventional breeding has been successful in giving resistance to only biotrophs, mostly successful resistance. But as it comes to necrotrophs, probably we may have to look for unconventional breeding methods, and I'm sure these will be taken care in future. Let me once again congratulate all the speakers for having done a wonderful job. Professor Kushalappa is still early morning, he got up and still waiting for listening to the speakers. Thank you very much, sir. Thank you all. And Professor Fakhruddin, especially our appreciation and thankful to you. All the best. And I thank the reporters. Probably I just forgot their names. Dr. Shankarappa Yogi, please, on my behalf and my co-chair behalf, I'm requesting you to, I'm sure you have documented well, and please submit it to the organizers.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir. Thank you. We thank you profusely on behalf of our organizing committee. Sir, amidst your busy schedule, you are here involving actively with your valuable suggestions and inputs, and giving your valuable inputs in between the sessions with this for this excellent session on molecular biology of biotech stress resistance. Sir, we thank you. We also thank you, sir, MS Kulkarni, sir, for being here and chairing such a lengthy session with your guidance. Thank you. Thank you, sir.
Very much. Thank you.
I would also like to thank the reporters and the distinguished guests, participants. Thank you, one and all the speakers, all the oral presenters of all the 3 sessions of the day 2 of this international e-conference. So I request all the participants to join well before 9 o'clock, because tomorrow we'll be starting the session sharp by 9 o'clock. So be online before 9. So by 8:45, be online. Tomorrow also we have 3 different sessions with the brainstorming sessions. Thank you, one and all. We'll be ending this session. Thank you, sir.
Thank you, Raj.
Thank you, one and all again.
Sir, we would like to thank Dr. Kushalappa, sir. Kushalappa, sir. It's our privilege, it's our pride. We have here one of the eminent professor of McGill University, Canada. Sir, thank you, sir. Thank you for your valuable lecture, especially second green revolution with multiple disease resistance. Thank you, sir.
Yeah, my mom told me, I educated you. What did you do for India?
India. Sir.
So this is the minimum I could do.
Thank you, sir. Thank you so much.